co-host, and she will share the screen. I'm on sharing this one. She wants to try it. So,
Sir, sir.
Stop sharing, right. Make Dr. Subramanti as the co-host and 4.5.
Good morning, Vishwa sir.
Good morning, good morning. Namaskar.
Kai piki palai aisilu to. Now to cha milibani, kichi milibani. Kali.
Now I have made you host. You can share your slide.
Okay. Is it visible now?
Not yet.
No,
Share it.
not yet.
You have to share your screen on the bottom of this. Yes.
Yes, it will come.
Now it started.
Now it will come. Right. Yes. Perfect. Yes. You can
It is coming.
Yes.
Yeah, make it slide slow. Slide slow.
Go to the full screen mode. Yes. And then yes. Perfect.
Yeah.
Now it is coming, right?
Yes.
Yeah.
Perfect. Perfect. Very good. And you are there in the panel, and there will be several kind of questions.
Okay.
Like, what is the cause of the situation, the teaching and research situation in Odisha? Why Odia students are not competitive in national scenario, and why they are not employable? So, cause is not very important. What are the possible remedies? You can
Right. I have some thoughts on that. Okay, because I also go to this KJ Somay. Yeah, I also go to this KJ Somay as a visiting faculty. So I see like there are certain differences between the education system of Odisha as a whole and other parts of India, specifically in Mumbai or Maharashtra, Gujarat, something. Even if they are not intelligent, they get better opportunities.
Yes.
So that is what I see Odia students when I think about ourselves. At least our basic understanding is much better. They don't have that, but still they are employable. That is a bigger challenge for us actually. We need to see that. Maybe I have some thought process we can discuss during our meeting.
Yes. Dr. Biswal will be chairing the session.
Okay.
S.K. Biswal is chief scientist in IMMT.
Yes.
RRL Bhubaneswar. So
Okay.
he is also the president of 4MI Minerals, Materials, and other MSA.
Mining Mineral Metals and Metal Society of India.
Okay.
Okay.
Yeah,
So we have joined hands. OCS has joined hands with them, and so there is a very specific reason, because they will deal with engineering sciences, chemical engineering to be specific, and we deal with fundamental chemistry. So we want to improve very sincerely the situation in Odisha, and all the panel members will be requested to give very thoughtful their ideas, and we shall record them and then pursue them, and maybe we shall have a very specific webinar on that thing. What are the possible solutions, and more debates on that, and then how to achieve that target. So that will be again probably.
Yeah, that is actually best solution what you have even said yesterday, that only students and only few things to discuss their problems are now it is all webinar, so there is not much problem, not traveling, nothing is there. So that is really helpful in this today's digital world. So it will be much easier. Yes, we want our students. I see very few ODS students coming out.
Good morning. Good morning, sir.
Morning, Subrat.
Good morning. Good morning.
Am I audible?
Yes.
Thank you. Hi, everybody.
Right, good morning.
How are you?
Okay. Sunday morning, watching cricket match. India, Australia going on.
Good.
Number has gone to 45. Okay.
Then we'll start on 10. But exact.
Oh, absolutely, dot in time. And today, yesterday, I got several calls that why the timings are not being adhered to. And I said, that happens, when somebody is emotionally speaking, uh, uh, doesn't look at the time. It happens. But it should not be too much, like 10, 20 minutes, this, that side is okay. But
The
1 hour,
5 minutes, sir.
I mean, 20 plus 5 is there, so another 5 minutes is okay.
Yeah.
This is okay, but it should not be like 45 minutes. That happened yesterday. Certain lectures went on till 45 minutes. So the chairpersons, they also become helpless at certain time because the debate is so strong. So, that shows people are interested. They want to ventilate their feelings in many ways. So today we shall try to stick to time, and we shall start the panel discussion exactly at 1 o'clock. Shall try.
Maybe some discussions, if it is required, we can do offline
Sure, absolutely. We can
so
continue.
that we don't have much delay in the discussions.
The chairperson is the anchor person to whom you can write email and stating all the details, whatever your thought is, and all such emails will be compiled. And in fact, these materials are not like going to dust. They will be put in certain, in this meeting, proceedings will be prepared. And in that proceedings, after the abstracts and all that, we shall put this panel discussion summary, and we shall try to follow it up. It's not like that we just leave it there. So basically, say for example, nobody is asking to teach thermodynamics or chemical kinetics,
Right.
but problem-oriented questions like solubility product, common ion effect, problem-oriented questions at undergraduate and postgraduate level. That is theory part. And then for experimental skills, experimentation training, that real experiment conducted, like there will be several lab visits, virtual labs. I mean, we shall actually visit the lab and record some experiments. So that is what we shall do. We shall contact several group leaders. Like had you been here, would have contacted you. Otherwise, you yourself will plan it out and prepare a video and send it to us. And dealing with any instrument that you are handling. So any principal investigator dealing with any instrument can send the video of its uses, and we shall put it up in the OCS website. So I think these are the things which are doable at no cost. So we can do it. Let us try. Let us try to help the students as much as we can.
Yes.
So Devasis, can I share my screen now? Am I still the host? Are you there, Devasis?
Yeah.
Yeah, I can share. Okay.
You can share, sir.
Okay.
My post.
Yes.
I have one more suggestion, Professor
Yes,
Samal.
Go ahead.
We should come out of this Zoom. We should have our own Geo Meet or something. Zoom is a Chinese this thing.
I know. I mean, they keep all records of everything. That is a
Yeah.
problem. But just like TikTok, this is the same thing. So
We can have a Geo Meet. Geo Meet is working pretty good.
Geo Meet is produced is Indian.
This is Reliance. This belongs to the Reliance.
Okay.
Give a demo. Give a demo at this meeting.
That I cannot do right now, but yes, whoever is there, I can share. You can download in your mobile itself also.
Okay, yeah, same thing as Zoom.
Same
We shall do
Indian. Yeah.
We shall keep it in mind. What is the bandwidth? Bandwidth is good.
It is very good because Reliance this year's this annual general body meeting was conducted through GeoMeet. I think 3 lakhs people are participated in that. It is such a big one, so it will be pretty good.
So we shall come out of this Zoom.
Zoom.
That's good advice. We shall keep it. I'll download it and I'll ask everybody. Okay.
That free? GeoMeet is free, or you have to pay for that?
It is free. It is free.
Oh, good question.
Maybe I'm not
Subhra
will pay for us.
When the number of participants is more than 100, we are paying around 7,000 something per month. Is it right? The bosses can say. So since the number is going to somewhere around 500 sometimes, so we are paying money.
Maybe sometimes, yes. I'm not sure about those
there must
money
be some
parts.
payment component. Up to 100, beyond that there has to be some payment.
Yeah.
It is not sustainable.
Free meeting is only for 40 minutes. If you conduct a seminar, you have to purchase.
Oh, it's a time, not number.
Also number.
3
people and 40 minutes.
40 minutes is free. After 40 minutes, you will be. If you have to conduct a seminar, you have to purchase the pro.
Okay.
Maybe there, but it is GeoMeet belongs to India. That is only the point.
Yeah, these videos should not be.
Madam, can I interfere?
Yes, please.
Sometimes see, we are telling about the China, how much Chinese people are doing work. Is it possible in India?
Why not? We have the similar population.
No question of not. I am not telling this way because first of all we should develop our mentality to be a competitor, and I ban everything, but at least we should develop our mentality in such a way that we are at par with them, and then we should discuss this one. We should ban everything, but we should develop our mentality in such a way that we should be at par with them. Please carry on.
Yeah, yes.
Thank you.
True. You said rightly we should develop. That's what all faculty members are there who are building the students. Who are building the students of the future generation. Definitely.
We are always talking about the future. And after these ones, how many professors are doing science, technology, application and modification? We are always within the university. We should come out of these ones and we should develop that mentality. Same things we are doing. We should ban China. We should ban everything, but we should stand in our own leg. That students, how many students will come if we take a class of characterizations, and which is the prime focus of the development of anything. So my request to all of you, we should develop our mentality in such a way that we should focus with certain goals, and that goal should be taken care of by the professors, and these professors also they should improve their selves. So professor, scientist, and everything.
Okay. May I interfere, please?
Yes, sir. Please.
This is beyond our discussion because there are these fundamental differences between India and China. India is a democratic country and China is.
Sir, a question is not democratic. Question is not.
Purusartha.
Question is our mental attitudes to develop ourselves.
काम करिवार attitude.
काम करिवार attitude टा हमें रखिबा. हमें develop करिबा. We should stand in our own leg. खाली talking माने कही हम केते वर्ष बंचु. That sorry, I am interfering with this one in a different way. I should not tell you because I
No,
know
ठीक
I
ठीक.
have,
This is the requirement.
and then I have traveled about 11 countries of the world, and I was working in a MNC also. So I know about the difference between Indian attitudes and Europeans or Chinese attitudes.
Our is better.
Huh? Our is better. Always we are talking better.
No, we
Because
are
always,
better actually. We are better.
no,
We know
no,
how to
sir.
live, how to enjoy. Yes.
Oh yes, yes. That way we are better. You are absolutely correct in that way.
I don't need this materialistic world. Right?
This
No, no,
particular
no. We are
question,
always talking about the spiritualism, and materialistic. Okay, sorry, sorry to interfere with this matter. And I think that all the intellectual persons should develop in such a way that we should stand on our own leg. Okay, sorry to interfere with this matter.
no, it's all right. But see,
Yeah.
the point you are raising is such a complex point. There is no easy answer to it. So
Yes,
it
sir.
was itself a huge debate. Why India is spending such a small amount of our GDP on higher education?
The question is
Why
not GDP,
South
sir.
Korea and
Question is that we are always talking about the GDP, and whatever facility we have, we are not facilitating that once. I am telling you one of my own life story, and we are working. I am working in IMMT Bhubaneswar, and sometimes from Utkala University, some students has come. And when we are talking about the characterizations, a professor of that university is telling that you finish within 15-20 minutes. So in 15-20 minutes, what they will know about the instrumentations. So if you will not improve that once, and we should educate them. We are talking about the at par with the Europeans or Australians or other things. At first, we should develop in such a way that we are at par, and we should have that one. So that's why I am requesting all the intellectual mass, especially Chemical Society of India, they should develop in such a way, because without chemistry nothing is there, research fields. So all fields are related, and without characterization nothing is possible. And everything you intellectual Professor Samuel, I am requesting you. I have met you in OCS, and as a learned person, I am requesting you that you develop in such a way that we should have an attitude to achieve something. That is my request to all of you. My objective is not to hurt anybody. If I was hurting anybody, then I am apologizing myself. But while I am going to other country, whatever I am facing, because we are telling that we are intelligent. Yes, we are intelligent, but our intelligence remains with the certain qualifications and a certain university. But we should develop these ones. We are talking about Mumbai. They are industrialists. They are working hard. So we should work hard, and we should think about others. Sorry to interfere with these matters, and I am apologizing if anybody is hurted by this one.
okay.
And I am
record, so nothing.
Okay, sorry to interfere.
Mr. Pallai.
Sir.
Madam already told that why the Gujarat and Maharashtra students are getting employable, why our students are employable. We should make debate on that. Not China, India, that is a different issue.
नहीं सर आमे जो जूम को आपण जोटा कहउचंदी आपण हमें तो हम जानि छ समस्ते किंतु आमे जो बैन करिवा कथा इवन इफ नेशनल मूवमेंट रे हम बैन कर दबा किंतु आमे ए पर्यंत इंप्रूव करछे
Mr. Pallai, IT sector is possible to ban this thing, but in industry sector you cannot ban because we depend lot of raw materials product on China.
Yeah. Entire power industry now depends upon China now.
नहीं वो कहबा कथा सर
we understood that, but that has to come by a decision that Dr. Samuel told. This is a actually attitude, not decision. We will take decision. We cannot ban immediately. We can slowly we can.
Ban कले किछ असुविधा नाही मो कहबाटा सर बैन
कले मैन्युफैक्चरिंग हबनी सर ना संगे संगे बैन कर
नहीं मो कहबा कथा हम प्रथमे से
गुड मॉर्निंग
गुड मॉर्निंग
गुड मॉर्निंग
गुड मॉर्निंग तपन
ସାର୍ କାହା ହାତରେ ପାୱାର ନାହିଁ ଆମେ କଣ ବ୍ୟାନ୍ କରିବା?
ଅଛି ଅଛି ଆପଣଙ୍କ ହାତରେ ଅଛି ସବୁ Tata Steel, Reliance and Aditya, they can do whatever they like.
କିଛି ନାହିଁ, they are highly political ହେଲା କି ସାର୍, ସବୁ ପଲିଟିସିଆନ କରୁଛନ୍ତି, ଆମ ହାତ ସାର୍ କିଛି ନାହିଁ। ଇଣ୍ଟେଲେକ୍ଚୁଆଲ ମାସ୍ ଯେବେ ଜାଗିବେ ସେବେ ଚେଞ୍ଜ ହବ।
You are right, Tapan. Intellectual mass, we should be, our motivation should be go in positive, motivation should go in positive direction with certain objectives. Okay, ठीक है भाई। तपन आपण करंतु, sorry for interference।
आपण रोज जिबे कोडे
कुआडे जाउछन्ती?
नहीं कुआडे जाउनी, अछि आपण पे वेट कर।
हां काली तो जॉइन करनथले, आज जॉइन करछन्ती।
आई भाई काली से आपण जानछन त म कहि छी आपण को घटना हैला, सॉरी। ओके।
One day,
students are देखा होना दिखाई, but
a king said, why
आज के स्टूडेंट बहुत कम होछन्ती।
काली भी नथले
के नरेंद्र जने राजा दिने कहले मो प्रजा माने कोन बिरयानी खाइयो पाउ नांदी। एटा गोटे बड़ लेसन जानी था जे आमे पिला माननकु किबा टीचर माननकु कि साइंटिस्ट माननकु को ये ताको ब्लेम करबी, किंतु प्रकृत रीजन टा आमे केही जानती नै। आरआरएल ले आपण माने आईआईएम रे जे माने अछंती, यू आर एक्सपोज टू ह्यूज इंस्ट्रूमेंट्स, you are like a king sitting there, you do not know what is the situation in Bhadrak College. Sanction strength is 18 faculty members, there are only 2, no money to buy certificate. Understand? So
sir reason जानी चंदे, reason के कहूना सर, reason
that is why Dr. Bishwal is here today. We shall not focus on reason. Reasons are very complex, and we are not here to find out the reason. We will be focusing on solution.
Yes.
Concrete suggestions. Don't blame teacher is not doing, student is not reading, or this is not happening, that is not happening.
Sir, that should be the attitudes of everybody.
No.
Cannot be changed, sir. Attitude is a very complex question. Attitude is a very complex question.
I said I should not ask any question.
We can define attitude. Attitude is a national character.
Yes, you are absolutely correct.
A national character comes by
nationality.
It can undergo modification if the leadership wants. If the leaders are not interested in building national character, then come what may, or whatever hard or whichever way you try, it is not going to change the situation. Se gote छुआ बेले गोटे सिनेमा ना गोटे ड्रामा करथलु, से कहउतला जे हीरो स्तर कहउची भोकिला पेट आदर्श सदाल के होसियो भोकिला पेट आदर्श। So जार स्टोमक, जार पॉकेट वॉलेट इज एम्प्टी, ता पगर आपण आदर्श कोन देखेबे?
हां से
सर मेन हला सर इजी जोटा लोक सेटा करूचंती, ब्लेम करिया का ना इजी।
Faculty members who are getting 1.5 lakhs, 2 lakhs salary, but there are faculty members who are getting 5,000 rupees salary. Highly heterogeneous, complex situation. So we shall, we have only 4 minutes to go, so let's move.
Yes. नहीं बाद में करना, मैं टेलीकॉन में। थैंक यू। Sorry about it.
Sir, Dr. Bishwal sir, हमरो membership fee is only 500 rupees, life membership. But we are not interested in that money component. We are interested that some persons from industry or engineering stream join OCS, so that they can know through several meetings what is our problem. We shall continue this collaboration or these meetings, but some members, it's just a very small amount of money, but if they join, then they can know What is the problem?
ସାର୍ ଭଲ ପଏଣ୍ଟ ସାର୍ କହିଲେ, କାରଣ ଆମର ଯୋଉ 4MSI ର every month we used to do the brainstorming session on the specific industrial issues and problems. And
So we can always
yes,
that
you are
connectivity,
yeah,
Tapan,
sir.
when we are doing the brainstorming session, the linkage you can give to the OCS secretary,
yes,
that they can also attend.
yes.
Okay, sir.
Dr. Raut, are you ready?
Yeah.
Not at 10 o'clock.
Good morning.
You ensure if Dr. Jena, Professor Jena is there.
That's Devasis. 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 Professor Jana sir.
Sir, hello.
Devasis sir ne jo Satyan sir asili
हाँ ମୁଁ ଅଛି ଲାଇନ ରେ
let us start sir.
Yes,
Dear participants,
over to Dr. Ramachandra Raut.
good morning to all. Dear participants, welcome to day 2 of the international conference. Let us get started with technical session 4. Professor Satyavan Jana will chair the session. Before Professor Jana takes over, let me introduce him to the audience. Professor Dr. Satyavan Jana is university topper both in Bachelor in Science and Master in Science. He obtained his PhD degree in chemistry from Utkal University in the year 1984. He was a postdoctoral fellow in 1989 at the University of Leeds, the UK, and Maxwell's Institute, UK. Professor Jana has specialized in organic chemistry as the major subject. Professor Jana is well known as a wizard of the organic reaction mechanism. He was a professor of chemistry at Utkal University. 11 students got their PhD degrees working under his supervision. Professor Jana was president of Odisha Chemical Society for the year 2016. Now I will request Professor Jana to chair the session.
Okay, thank you, Dr. Raut. It is my pleasure to start the session, technical session of the second day. I think there are 3 speakers in this session. The first speaker is Narendra Tripathi. Narendra Tripathi was our student. He passed MSc in the year 1992 from Utkal University. He had his graduation from Dhenkanal College, then MSc from Utkal University in 92. He did his PhD in IICT Hyderabad under the guidance of Dr. Gurjar, a good friend of mine. He had his postdoc, a couple of postdocs in USA. He was in Rutgers University, and I had visited that university, Rutgers in New Jersey. On his back, he initially joined Side Rocks at Hyderabad, and now he is in the MCure in Pune. He is in the rank of Vice President, Director of the Institute in Research and Development. Narendra Tripathi, I remember him very much because he was a brilliant student with very clear fundamentals in organic chemistry. I'm happy that some of our students are in the pharma industry, and I'm proud that Narendra is one of the speakers. So with this, I invite Narendra to deliver his lecture, and the time allowed is 25 minutes.
Okay.
So is Narendra Tripathi online?
Yes, sir, I'm here.
Okay.
Thank you
It's
so
my
much.
pleasure. Coincidentally, before you start, coincidentally, in this session, all the 3 students happen to be my direct students from Utkal University, and all had organic background. Narendra Tripathi, I remember him very much because just I will take 1 minute to give an introduction. when he appeared the interview in IICT Hyderabad, at that time Dr. Ramarao was there, and Dr. Gurjar and Dr. Jadhav, both were there, who we normally call them as left hand and right hand of Ramarao. And a particular question was asked, because this was told to me by Dr. Gurjar and Dr. Jadhav also. The question was, how do you convert to alpha phenyl ketone? Say alpha bromocyclohexanone, how do you convert it to alpha phenyl cyclohexanone? He explained the thing very satisfactorily, and from that day, Ramarao gave an instruction that anybody from Utkal University applies for research to IICT, take them for research. I still remember that sentence of Dr. Ramarao. And Narendra was the person who answered the question very clearly with all clarity. So with this, I invite Narendra to start his lecture.
Okay. Some screen.
Has he been made co-host? Devashish?
I'm trying to share my screen. Not coming.
Have you made him?
Co-host. I have made him.
Okay. Yes. We can see your
There.
Yes.
Are you able to see my screen here?
Yes. Go to full screen mode. And
Slide show mode को जान दो.
yes.
Is it okay now?
It's okay.
Okay. Thank you, sir, for the kind introduction. You have been such an inspiration throughout my career, starting from MSc. And good morning to all. Before going into my presentation, let me congratulate Dr. Sasdar Samal and the entire organizing committee to have taken such a nice initiative and conducting this OCS industry meet. I'm thankful to the team for giving me this opportunity to present what I've been working on for last few years. In the next 20-25 minutes, I'll try to give you a flavor of what's called drugs development, and particularly focused on API development. That's what I'm working on. I'm sorry, give me a second. Okay. Okay, so as Janice had told, I'm working in MCure in the API R&D department as the vice president. This is only the API part I'm taking care. Formulation is somebody else is looking at it. So there are two parts going into. Is this visible to everybody?
Yes.
Okay. I've been lucky enough to be associated with some great organic chemists in India and in abroad also. Technologist, Dr. A.V. Ramarao. Actually, he was my PhD supervisor, and Dr. Gujjar was my research intermediate when I joined for PhD. See what happened here after passing MSc in '92, I got a chance to join RRL Bhubaneswar. That time there was no organic chemistry department, and now it has changed anyway. There was forest and marine department, and I joined with Dr. Bapuji, who was one of the chief scientists that time. So in that year, what we did from RRL, we went to a symposium in the University of Andhra University, that's in Vizag. And well, organic synthesis industry kind of stuff. People from various academics and industry used to come and give lectures. So that was my first interaction with Dr. Ramarao, who happened to be one of the guest lecturers. And Professor Mehta was there. So I heard him and I was so impressed with the work they've been doing in IICT. I talked to him, sir, can I join you for PhD? Then what he said, you have to qualify CSR and then you can apply to me and then, you can join. So in 92 December, I think I applied for CSR and I got qualified. So Then I applied to him, and somehow I didn't get a response
Please
from Dr.
mute the
Amara
mic.
for joining PhD. So after 5-6 months, waiting for 6 months, I came to actually to IICT to meet him. Then what he said me, because there are so many applications coming to join PhD from all over India, because that was one of the prime institute for giving PhD in organic synthesis. So that's why we don't give anybody offer if they don't come personally. So now that you have come, you can join. So that's how I got into his group for doing my PhD. And incidentally, one of our seniors, Asmita Tripathi, she was one batch senior to me, and she was already there. So that was a good help. So, and as Jana sir told from the beginning, it was a little bit impressive from my part for giving answer those questions. So, so then on, I started working on PhD with him. He was the director of that institute anyway, so he was not having enough time to for the students for complete time. Administrative part also he was looking at it. So Dr. Gujjar happened to be the research intermediate who was looking day to day work, and final supervisor is Dr. Amaro anyway. So after completing PhD, I went to Rutgers. So there I spent about a year and a half. Then I got an opportunity to work with Professor Gunda Georg, who was that time was in the distinguished professor of medicinal chemistry in University of Kansas in Lawrence. She was one of the renowned medicinal chemistry, and now she's at as a chair of College of Pharmacy in University of Minnesota, and she's the chief editor of Journal of Medicinal Chemistry published by ACS. So I worked with her for almost 2.5 years. That's in total synthesis of anti-cancer agents. That's where I learned a little bit about medchem, and probably that is helping me a little bit here. And of course, Dr. Gujjar, as I said, he was my research intermediate during my PhD. He was in IICT in 98. By the time I finished my PhD, he moved to NCL. He was initially in NCL, so he was from Maharashtra. So he joined uh, this NCL, and almost 8-9 years. In 2007, he moved to MCure, and that's what I came in to from Sai. I moved to MCure, and I've been working in MCure for the last 13 years. So these are the people actually who taught me what I've learned so far in process chemistry and technology and everything. So this is our one of the building, and this is the lab actually in the right side. So the outlines of my presentation today will be a little bit, I'll talk about innovator drug, then a little bit about generic drug and Indian pharmaceuticals about, and API process development. This is where I have been involved for the last 10-12 years. And finally, if time permits, I'll give you a brief process development part of two molecules. One is RU486 and second one is fenceprad. Okay, so going into the now, here I'm giving a little bit of term of medicines. As we all know, what's a drug actually? What's a medicines? It's a combination of one part is therapeutics and other part is excipients. So it's a complete mixture of medicines. Whatever we are eating, it's a combination of therapeutics and one part is excipients. What is therapeutics? Is basically the active part. That's what is the active pharmaceutical ingredient in the drug is actually the therapeutics part. So there is a definition there, but basically we say the active part of the drug, which is responsible for the overall activity of the drug, is that's what the API. That's where the organic chemists come into picture in medicines. Otherwise, organic chemists will not be doing this. Now, the second part is the excipients. As the name says, it's a excipier, it's except, which is simply expressed other than. So everything other than the API in a molecule, in a drug, are the excipients. That could be many different things can be added to the API, which actually helps in taking the drug to the point where it's required. And that it helps in the mode of action of the drug. I'll talk about the API development a little later, but next slide. This is just a nice depiction of what is pharmaceutical manufacturing. And here you see that this is starting material or chemicals, common chemicals we take. We do in the plant, we convert those to the API. That's the final active pharmaceutical ingredient which goes into the drug or the medicines. And here, up to here, it's only organic chemist. And the second part, that is API and the excipients. This part is actually from the formulation part. The formulation team takes care. Those are the people who does this B farm, M farm, and all these things. They look at the DMPK and all sort of things of the finished product. And then finally it is packed in the primary package. We take off tear off that tablet and then eat it. That's the primary packaging. That also is a big study, what kind of packing is required for drugs to go in. And then the secondary package, the dabba, whatever we see in the stores and we buy it. So that's how pharmaceutical manufacturing goes. Now, I've just given a little brief on the excipient functions, what they do generally, because the API cannot be given to the human being directly. It doesn't go into the body properly. It doesn't reach there, doesn't absorb a lot of stuff. So these are the things which is components, fillers, binders, disintegrates, flow aids, lubricants, surfactants. These things help the drug to go to the point where it is required to be. It gets absorbed because of these things. So increase size and weight of the final dosage form, promote particle aggregations, all this. This is all the property of the drug. And examples are, this is microcrystalline cellulose, sucrose, pregnenated starch, hydroxypropyl methyl cellulose, sodium starch. These are kind of different kind of excipients which get into the drug actually. This is in combination with the API. And actually the solid, when we're talking of solid oral tablets or capsules or, that emulsions, suspensions, this is where the API is kind of a free base. So it's not exactly salt. So in the tablet formulation or the solid formulation, the excipients importance goes up. In the injectables, it's not that much, because the API is most of the times it's a salt of the API. So kind of hydrochloride salt or sulfate salt or organic salts like oxalate, succinate, all these things. So it's easier to get into the solution that because of the salt formation, and the injectables excipients are less. On the tablet part, you have used a lot of excipients to formulate the drug. So these are some of the examples. Okay, now coming to talk about a little bit innovator drug. What exactly an innovator is? The company which first launches a medicine into the market is the innovator of that drug. So the big pharmaceuticals like Pfizer, Merck, GSK, BMS, Novartis, and you name it, there are a lot of big companies which can actually afford to spend a lot of money on that, because it's usually taken 15 years from the discovery point to launch a drug into the market. So cost about 10 to 12 billion again dollars, based on the what kind of activity you are looking for, what kind of complex molecule you're looking, and the success rate is less than 5%. If you look at from the discovery point, a lot of molecules comes into the discovery phase, but then it fails on different stages. So the final outcome probably is hardly 3 to 4% success. But once you get into a market, the innovators make a lot of money. You see that Lipitor, which is called atorvastatin, it sells 10 to 12 billion a year for last 10, 12 years. Now also they're making a lot of money. So whatever money they spend, you can, really they get a lot more than that. The innovator company, what it does, this is just a sequence of process, how they go about. The first one is they go for some activity target identification, little bit on drug discovery. They get an API structure, tentative structure. Once you have a structure in place by this computer modeling, all these things they can go into. Then you do the SR studies, that's a structure activity relationship study, to finalize the drug molecule. Which one, what molecule? They always have a backup molecule in mind. One molecule they go, if something happens, they have another very similar with little bit chain. That's a backup molecule also goes into the pipeline. So what happens, initially they do medicinal chemistry route of synthesis. The medchem department actually synthesize few grams of the API, which the formulation team takes, and they make different dosage form. It could be oral, tablet, capsule, suspension, or injectables. Then they do the tox studies and DMPK, that is drug metabolism and pharmacokinetic studies, which actually gives a picture of bioavailability of the drug in the human body. So once they see any positive indication, then the process route of the manufacturing API starts development. The actual process development starts around that time, where they target around kg scale of API, which can be formulated and goes into the phase 1 clinical studies, phase 1, 1, 2, 3. Now they do 4 also. So what's this? Once this phase 3, phase 4 results are out, the positive results, they file with the FDA or the European agency. That's called a new drug application. That's NDA. When the innovator company files, it's called NDA. So this NDA is reviewed by the agencies, FDA, and then they go for queries, and then it's get approved. Then the actual commercial manufacturing of the API and drug product starts, and they launch the product in the market. So if you look at it, the innovator company actually does the a bunch of the main part of the drug development, which the generics doesn't have to do really. So now this is what the generic drugs means. This is where all Indian pharmaceuticals are involved. So generic drugs are basically copies of innovator, that is branded drugs approved by drug control boards like DCCI, FDA, EMA, Health Canada, and all sort of regulatory bodies. Meets the same high standard of the innovator drug. So basically it's an affordable alternative. So where a lot of people, common people can buy it. Innovator drugs are very expensive. So to qualify as a generic drug, these are the things we have to do, must do. This contain the same active ingredient as the innovator drug. This is very important. What happens here? The FDA and other people, they don't allow the API to be changed. So the name of the API has to be the same, exactly same. We cannot alter the API in a generic drug. The next thing comes in the same dosage form, that is oral injectable. If it is a tablet, your generic has to be a tablet, and the strength, whatever you are looking, if it is a 20 milligram tablet, the generic also has to make 20 milligram. So the strength also has to be same. It has to be bioequivalent. The generic product has to, we have to show that it is going to the same place, part of the body, and it acting on the same way. Meet the same standards of the identification, purity, the quality, and has to be manufactured with a high GMP norms. What is allowed for a generic drugs is may differ in some characteristics, such as the manufacturing process. So manufacturing process of the API can be altered or manipulated. You can change the manufacturing process of the API, but the final product has to be exactly same purity, with the exactly same thing. So allowed is manufacturing process of the API or even the formulation. Excipients can be reversed. The color, taste, packaging, this kind of stuff you can actually alter. So this is where a lot of companies comes into picture and do this, because they don't have to the initial study what the innovator had to do, where they have to spend a lot of money and a lot of time. So that's why the generic companies get into the market very quickly. Now you see generic drugs now represented more than 85% of drugs dispensed in the United States. So a lot of, in the US also, people are not buying the branded drugs because those are very expensive really. So now about the this one, Indian pharma industry, because of the cost, huge cost, basically time and risk involved, Indian companies which cannot afford, or maybe not ready to afford, maybe they're not willing to do it, or maybe not really they do the money and spend. Almost all the Indian pharmaceutical industry are into generic business. That's for domestic market as well as regulated and US. Because of that, what has happened, not a single new drug from India so far invented, a real drug. Not some biosimilars and all sorts of things coming, but not really a new drug coming out of India. But as a result, what has happened actually, India has become now pioneers in generics. We are one of the leading countries to be producing a lot of generics drugs. What are the reasons for that? We have great institute for basic science, strong foundation in chemistry, talented organic chemists coming out of IICT, NCL, ISM, CDRI, IITs, CSL labs, name a lot of very good institution where organic chemistry start as a PhD level. So then that's why people can think of new chemistry and comes into new routes, non-infringing routes. You can go into a lot of different, quickly get into API, and that can go formulation and this. So we have become very strong in API process development and production. So this is just a thing to prove that we have more than 50% of drug master files filed with the FDA are from India. More than 50%. You see that in 2019, India has, I mean, in all over the world, 616 filings are happened, and out of the 313 were from India, and we have 86 companies filed DMFs. This 313 from India, and China it is 113, and in the US itself is only 57 DMFs filed. Out of this, we get approval, few of them, and few of them goes into review and also. Now see a little bit more. India contributes more than 20% by value of total generic drug manufacture in the world. That's and almost 40% of US drugs by volume are manufactured in India. If you see in 1970, that was very early after independence, where Indian pharmaceuticals had a 5% share of the Indian market, and global companies had 95%. So you see 5% and 95%. But now if you look at 2020, around 50 years, it is almost reversed. It's 85 and 15. So Indian pharmaceuticals actually has grown out like exponential growth. From 5%, we have gone to 85% covering. So, and because of this, a lot of competition is between Indian pharmaceuticals, very high competition, leading to lowest prices of medicines. Indian pharmaceuticals is now 37 billion, growing as around 9 to 10%. Exports accounting for $18 billion. So basically what you see here is generics vs branded. You get the same, exactly same drug, basically the safety, the effectiveness, and the same active ingredients. But here, if you see, the money is here is dollar, here is a lot of dollars. It's basically around 60-70% difference in the majority of the drugs or medicines. You can see that. So just very little about MCure. This is established in 1983 by Satish Mehta in Pune. They initially involved in drugs manufacturing, working as a CRO for all MNCs like Pfizer, BMS. They were doing Benadryl kind of stuff. These are manufactured in MCure and marketed in India for domestic market. Started their own formulation in 1996. That's why they do ANDA. The generic company has to file ANDA. Then own API R&D was constructed, and manufacturing starts in 2001. So far, we have 120 US DMFs, 50 European and Canadian DMFs, 350 plus ANDAs. We have pioneers in iron products. That's one of the strengths. We do a lot of iron products, which goes for iron deficiency. Orofor XT is one of the brand which is going around 250 crores. And another branch, we have chiral drugs. This is where again we started this chirality in the drug. So you see a racemic drug, and we make the chiral version of that. So basically what happens, you get 20 milligram instead of 20, you actually have to take 10 milligram of the chiral drugs. The SM reducing, there are 15 molecules we have in the market now. So it's a 12,000 plus manpower, 6,000 plus crore turnover. We are number 11 in Indian domestic market, business in 50 countries, subsidiary and offices in US, Canada, UK, Germany, etc. So this is what I have been involved for the last 12 years after coming from SAI, API process development. This is how we go about. Once the drug is selected by the marketing or BD team, it's given to our API R&D team, where what we have to do is we have to look for literature, because generally our generics products are off-patent products. Very rarely we do patented products. You have to go for new licensing and power for filing and all this. So then we do the literature search. What are the processes available, and look for the IP analysis. We go for a route selection, and look for if there is old routes, we cannot do it, or we have to look for an NI route, and we can file a para 4. Then we do a feasibility study of that route, starting with 2-3 routes. We have to do very small scale new reactions and see which one is working better. So based on the feasibility study, we finalize the route, On that route, we do a process optimization or the conditions optimization. What kind of solvent, how much solvent, what kind of reagents, and how much of the reagents. That's optimization of the conditions. And this point, we give the sample to the formulation team for their development. So they develop their dosage form, whether it's tablet or injection or whatever. Then parallelly, we do analytical method development because purity and quality has to be maintained. So whatever we synthesize, those things has to go for characterization and analytical part. But the method development goes here. Then simultaneously, we work after optimization, we go for scale up. That's in the plant. And where in the plant, when it comes, so here there is some PSD. That's actually the particle size of the API what we generate. Particle size sometimes becomes very important for the oral tablets, because if you have bigger particles, the solubility dissolution sometimes becomes tougher. So it depends on the formulation team. They will ask us what kind of particle size they're looking for, and different polymorphs. If the APIs can exist in different crystal forms, we have to select which form polymorph is going into helping in formulation. Then we do the validation of the analytical method of whatever analytical method we developed. Validation means we have to do robustness, linearity, all these things. After this, we do process validation. This is what the mandatory requirement of FDA and all regulatory agencies. Process validation means manufacturing of 3 consecutive successful batches at plant. You have to have the same scale. You have to see that all 3 batches has gone well. That's why this material is used for exhibit DP batches, where they manufacture 3 drug batches, formulation team, and then we do the stability study of API and DP long-term and accelerated conditions, where 25°, if you have to 25°, we have to do accelerated 40°. See that the drug, the final product at the API is stable enough so that we can give 2 years expiry or 3-year expiry, whatever you see on the drug backside, we see expire manufacturer on this and expiry on this date. So then after with this stability study, we file ANDA and the DMF. The formulation, the generics has to file ANDA, where the innovator files NDA. That's new drug application. This is abbreviated new drug application with the agencies. Then the agency review our documents, and they have some query response has to go, and we get approval, and then the commercial manufacturing of the API and DP starts. And then in between, once we commercialize and send it to US or India, the auditor, it goes for audits. This people come from agencies to audit the facility. They look for GMP norms, whether you are following or not, what kind of quality system is maintained and all. So this is basically the way how the generics companies work. This is basically a formula. Almost everybody does the same things, and we do the same thing. This is where my expertise comes into picture, and the chemistry part. Okay. Now, let me tell you about a little bit about 2 molecules. This is process development of RU486. This is the molecule which I worked myself on the table in '93. Once I joined Dr. Amarao in PhD, that time the PhD students were never given basic research directly. They used to give 1 molecule. We have to work on process development. That's an industrial project for a year or so, and then we were given the basic part. So when I joined, this is the molecule they gave me to work on. The scheme was available, and the total conditions and optimization part was given to us. So this is a drug which is chemical name is mifepristone, which is an abortion drug. This was really a hot molecule, abortion pill. It was developed by Rausel Uclaf. That's a French company, and that's why the name is RU, and 486 is the number 82. The brand name is Mifeprex. Dose is it's a tablet form, 200 milligram. It is given in combination with misoprostol. That's 4 into 200 milligram tablet of misoprostol. And these are the basically excipients in that. You can see that in the tablet form, only 1 API, then there is several things added into the API to make the drug tablet. And I'll tell about a little bit about this second molecule, which we have done recently here in MCure. So this is the molecule I worked 25, 26 years back. That's RU486. Let's see how it went about it. So this is the molecule that's Ru-486, and this is the starting material. That's called estrone. And when I joined in '93, this after a couple of weeks, I met this gentleman. He's a Nobel
Narendra,
laureate.
you can wind up another 2 minutes.
I have 2 minutes, sir. Okay, I'll just run through quickly. Run through this only one I'll show. This is this was the starting material was supplied by Professor Sune Bergström. He was from Sweden, and you see that how we go about. We did the phenolic OH as a protection. Then we did sodium borohydride of the ketone reduction. Then we did a birch reduction. This is what the lithium liquid. This is where I remember always Janasha. He used to teach us this birch reduction, how this works. We have a methoxy here, we get parallel double bonds, and we put a little bit acid, this methoxy chops up, and we get the ketone, this isomerase to ketone. So once we have this, this is the fragment. The critical part is here, if I have more acid, this double bond will migrate to give this alpha beta unsaturated ketone, which is basically not useful for us. So the next step, the double bond, here we have what we have to create the other two double bonds that we do by iodine addition and then elimination by putting base at -40. We get a 40% yield of this diene. This one we protected. This one actually it is very critical here to mention that if the elimination goes this way, we go back to estrone kind of molecule. That again is not really helpful for us. We do oxidation here, protection of the ketone here as ethylene glycol protection. Then we do a strong oxidation of the alcohol to ketone. Then here we do N-butyllithium and propine addition to this. Because of the angular methyl with beta, this propine goes into the alpha side, alpha attack, and we get very good selectivity or 98 plus selectivity of the propine addition. Then in the next step, we do epoxidation here, and selectively we get the alpha here. We get a mixture, so we do purification because we need the alpha so that this dimethyl grignard, dimethyl aniline grignard can attack on the beta phase. And next step, you see that here we take this bromo and we generate the bromo grignard reagent, which attacks on this beta ketone, and we have the double bond migration and the epoxide opening. We get this, and finally, what we have to deprotect this ethylene glycol, which we do by acetic acid. We get the ketone back, and once the ketone forms, this proton becomes acidic. So this hydroxy eliminates out to give us the final molecule. So this is how the total scheme looks like. The 11 step synthesis, we got 15 gram of API with 98% purity, 4 column chromatography, all the reactions, birch reduction, Schoen oxidation, epoxidation, grignard. It took 15 months for 2 years. So should I stop or I can continue for another 3 minutes?
I think you have to stop.
Okay, I'll stop here. So I hope I have given you a flavor of what we do and what's basically drug development involves. Thank you so much. Thank you for your kind attention.
Thank you, Narendra, for an excellent lecture on the generic drug development and your association with the MCure. We can take one question if there is any from any person.
Yeah, I have one question.
Sir.
Okay.
May I ask?
Please.
Yes.
Yeah, Dr. Narendra.
Sir.
See, we are hearing, you said that it takes 15 years to get, one drug finally to be useful for the requirement. But today we have seen one vaccine has come for this in less than a year. And I think the data analytics and the technology is helping so rapid. This is one is called the disruptive innovation. And also we are hearing in generic that the DNA tests have come and they are going to tell us if anybody is going to have a heart attack after 10 years. Today, the DNA test tells that when you are going to have it. And also I understand that some of the medications are given, now developed specific to the particular patient. The drugs are being manufactured by the pharmaceutical companies specific to one person. I think we must, I understand it is because of the data analytics, which is the artificial intelligence. It is helping tremendously to help it. I would like to have here something from you. It is generic again. You are the master in this. Can you throw some light on this to us?
Yeah, I just say that, sir, for the innovators to get into market, we have to do this phase 1, phase 2, phase 3. Now, what have happened because of the pandemic? It's spreading so fast in everywhere. The FDA and agencies, they are kind of waving off. Phase 2 and 3 is going on probably. In the due course, we'll get it. To put a vaccine actually into market in 1 year is kind of almost impossible. But then, because seeing the situation, and as you said, new things they have given, that the companies has given them, the FDA, the agency, they're approving it to be used it. So we don't know really what are the negative part, the talk study, how much they have really done it. We don't know, but people are ready to take a chance and to go for the vaccines. Let's hope it works, and we get very low negative part on that. Okay. Thank you.
Professor Jana, are you there, Professor Jana?
Next speaker.
Jana, you can hear
He's
us?
on mute. Sir is on mute.
Mute. Devasis, can you?
Am I audible now?
Yes, yes. Go ahead, please.
Okay. So let me thank Narendra for this excellent lecture.
Thank you, sir.
And if anybody has any doubt, they can directly contact Dr. Tripathi of MQ. So thank you, Narendra, once again for this excellent lecture. Now we can go to the second lecture. The second lecture will be by Dr. Subhra Mahanti. Dr. Subhra Mahanti also happens to be our student. I think same batch, 1992 batch. And Dr. Samal, you see, there is a coincidence. 3 persons of the same batch. Yesterday, Bankim Tripathi, and today Narendra Tripathi and Subhra Mahanti. All of them belong to 1992 batch. An excellent batch. All the top 5, 1 to 5, they all qualified CSR net, and I still remember that batch. So Subhra Mahanti, she graduated from OUAT and then MSc from Utkal, 92, and she had her PhD from IIT Kharagpur. Then after a couple of postdocs, she joined she joined the company, the polymer company, and she is well trained. Her area of research is polymer chemistry, polymer synthesis, polymer blending, polymer, all sorts of polymer things. And she is now working as a group leader in the polymer sector, polymer industry. And she has also taken some of our students to her group. So with this, I frequently also meet Subhra whenever she comes to Bhubaneswar. So with these kind words, I now invite Subhra Mahanti to deliver her lecture. Subhra.
Yeah. Can you see my screen?
Yeah, please put it in full screen.
Yes.
Yes. Okay. Let me put it.
So Subhra, you are given 25 minutes time. 25 minutes time.
Definitely, sir. I will manage to the time. I'll stick to the time. So, thank you, sir. I think it is my privilege, to be introduced by you. And it is really a nostalgic feeling that my friends are there, Bankim is there, and Narendra is there, and all my seniors are there. Even I see my professors, even Professor Das is there. So it is really an honor for me, and I feel good that we are here together again to do this thing and taking a bigger challenge for us to take the Odisha chemistry students ahead. That is again a challenge for us, what we have got. So with this background, again, I would like to thank Dr. Samar to take this initiative to bring all of us together and thinking about the future students. So that is another great thing. And this OCS Industries definitely, we are thinking that it is going to achieve the goal, what we are expecting. we have a panel discussion. Definitely we'll discuss on those parts again. So with this thing, I will go ahead with the my lecture, which is, it's not any scientific lecture. I want to give an overview of the polymer industry. As you have seen, like I think from yesterday we have chemistry, we have minerals, chemicals, and followed by the today a beautiful lecture on pharma. So I will take you the another beautiful material, which is the modern day material. I would say it is the polymer, where we are going to look into this. What is the market? What are the opportunities? How our students can really enter to this area. So this is one of the small disclosure which I will put it as. Actually this whatever I am presenting, this belongs to me, not belong to any Reliance, because I work for Reliance. So this is nothing to do with Reliance Industries. This is my own thought process and presentation that is mandatory from Reliance Industries. So on this topic, after that, I will take you through what I am going to present you in this. I would like you to take through some of the growth in industry, how it has grown. Then the polymer industry, polymer being a material. Then what are the requirements in industry and opportunities and scope. These are the few things I would like to cover during my lecture. Next 20 minutes, 25 minutes. So let's see that what is we are thinking of. I will take you through the industrial revolution. You know, industrial revolution, it has happened. We are today talking about the industry and academic collaboration. So thing is that what is this industrial revolution? How it is evolved around to till date? So if you think about this, why the industrial revolution happens, this is because of a better social life. Everybody wants a better social life. Even today, during this pandemic area, we also still need a better social life. And that's what the industrial revolution or the changes comes in industry. So I will just take you a few minutes through this industrial revolution, where you will, understand how the revolution happens and how the world changed during this period. The first industrial revolution happened in 1765 to 70. During that period, when the steam, coal, all this power generation was there, or then the mechanization was there. So it is fully depends on the energy. All the industrial revolution mostly depends on the energy. Still now what you are seeing it, or making our life better. So the first industrial revolution came in 1765 to 70, when the mechanization came. Then the next one came, which I would say the next one is second industrial revolution, which is after around 100 years, where the electricity was developed, where the mass production was communicated. So that is called as the second industrial revolution globally, where a lot of production, electricity was the new thing that time when it was discovered. So the energy source for the industry was electricity. That's what we call it as the second industrial revolution. Third industrial revolution came when, this is again after 100 years. If you see the chronology, it is after 100 years, where the electronics or the nuclear power came, where the automation came. If you see the time period, this is 1969, afterwards is 1980. And this is exactly after the Second World War, when world has gone through a deep crisis. Across the globe, everybody was involved in the Second World War. So we have destroyed many things. And during that period, after this development or this revolution came, that is what we are seeing it here as a third industrial revolution, where all the electronics equipments came, the nuclear energy came. Whatever we are seeing today, that also make our life much more comfortable, better social lives. That's what is the third industrial revolution. And you see, when there is a challenge, then when the revolution come, then the opportunities come, then we get a better opportunity, or the better things came out of these challenges. And just after 30 years, from 1969 to 80, and then you see the 30 years, we see another revolution. We call it as internet or the renewable energy. As I told you, these are basically based on the energy sources for the factory or the industries. So this is the fourth industrial revolution came, where everybody thought that, okay, this is now high time we are using our resources. Now let's see how we can use the renewable energy. So the solar energy or wind energy, everything came here during this 2000 period of 2000. And the other important thing which has come up, and which we are enjoying our life today, that is of internet. all of us now going through the Internet of Things, Internet, data management, everything. That is a part of this fourth industrial revolution, which has changed all our lives. Today, I'm talking to all of you sitting around almost 2,000 kilometers away. So that means there is a difference. There is a it made our life so easier that we don't have to travel, but still we can communicate. And that's what is the fourth industrial revolution. But this is little bigger fourth industrial revolution, where I will take you through that little bit in a bigger way. So what is this fourth industrial revolution where we are going through? If you see this fourth industrial revolution, actually, I want to put it to the students who are thinking or who are getting this opportunity to listen. In our times, we never get. So if they have a bigger view, then they can enter to the right area in the right time. And of course, they can modify themselves or prepare themselves for the next age of chemistry. This fourth industrial revolution, if I say, there are three distinct parts of it. One is the physical. Physical means it is material. Digital. Digital means all of you are now, we are digitally connected. So that is digital. And the biological. Just now, my friend Dr. Narendra, he said about the biological chemistry. And this COVID-19 or this pandemic also showed us how the biology or the safety of the human beings is important. So today, this 20 onwards, 2000 onwards, we see this is really very much valid, this physical, digital, and biological. So these things are now, there is no distinct division between these areas. If I tell you today, it is the amalgamation of physical, digital, and the biological chemistry together. So in this case, if you see, very small example I would tell you today, if you see this oximeter, all of us, we are using oximeter. What is it? It is made up of a physical material. Some digitization is there. So there are some figures which is coming. It is biological because it is measuring our oxygen content. It is giving our heartbeat. So the future generation or the future development or the future manufacturing, everything will depend on this amalgamation of all the three areas. If you think about any of this thing, what I have put it here, if you see in the physical 3D printing, today nobody is ready to go for any of this mixing molding things or for the any article, for the any manufacturing. So people are trying to print it like a paper you are printing it. Everybody wants to print the article. Nanotechnology. Nanotechnology, another important area, which is again very important, being a physical material, very small nano size material, which is very helpful for the biology or the health segment because it is carrying drug and other things. So you know this is a drug carrier also, as well as it is digital. You see that today's nanomaterial, graphene is a miracle material where all the digital, you put a thin layer, it conducts electricity. So it gives a good, very thin material, or you see your computers, your laptops, very small things, but it can do miracles. So that is what the nanotechnology. Medical sensors, I told you oximeter, that is a very good example. All other things today, if you see it, even the wearable, you wear a watch today. That watch is also a smart watch. So that is again made up of all the three things today. So we need to think, or our future generation needs to think about this amalgamation, how to do the different innovative things. We have material, we have polymers, we have digitization, we have everything, but how to combine together and get a new innovative material. That's what we are looking for the next generation. They should be, make them ready for this type of futuristic research, I would say. Now, today, all of you know that we have gone through this COVID pandemic era, which is 2020. None of us we worked. Even globally, almost I would say 2 to 3 crores people are affected. So, does it mean that we are moving to Industry Revolution 5.0? So, let's see what was the thought process before this thing. Here, I thought to connect with the people. I thought that I'll put 2 pictures here. So in this case, if you remember, I don't know, many of my age people, maybe they will remember this thing. There are 2 things here. This was one serial that time it was running in, I think, late 80s. This is, Star Trek. This serial was Star Trek. The other one is, this was something, if I remember, it was called a giant robot. I always remember these 2 things. That time, there was not even a mobile phone. That there was not even a landline also in many places other than few areas. We had a big phone. But this time, people have imagined these 2 things. And we are here. This is only 40 years. If I say late 80s and today is 2020, this is only 40 years. In this Star Trek, if you see, if any of you can go back and see those serials, sometimes they used to appear, sometimes they used to disappear. That was the thought process. Today, Google has developed those packages. You can virtually present everywhere. You can virtually do many things. So that is what the virtual augmentation or the virtual reality, I would say. Now, in this case also, if you see this giant robot, this is a small boy who was called, I think, Johnny Soko, and there is a big robot. So he was controlling, the small boy was controlling the big robot. That was the thought process then. And he was talking through a small watch. That's what I remember still today. And that was the thought process of some scientists, some fictionists that day. And today it is the reality. You have the, pilotless aircrafts, you have the drones, you have many things. So which is possible, that's what we are seeing it today. So you dream it today, tomorrow that is possible. That's what we are thinking of. So when we talk about this moving to the Industry 5.0, Industry 5.0 is what? Like, let's see what is this Industry 5.0. If you see this Industry 5.0 would be, most of the times, it is man and machine. So man will create some machines which will talk to the, generate some data for you. And based on the artificial intelligence, I think someone was asking about the artificial intelligence, big data, data analytics, all these things are coming up. Today, even in Reliance, we are working for something, some data, like we don't want to do experiments. We have some statistical data, we will extrapolate it and get the product, get the properties, what we want. So that there is no more, a lot of experiment, there is no expenditure, but we can get the data. We can, on the basis of that, we can make some prototypes. So those are the thought process. That's what industry is moving. So based on this industry background, if I tell you, or let's go to the particular thing. If you see here, again, there are three things: physical, digital, and biological. So physical means it is material. So the next generation, when we are going for the next era, it will be physical advanced material and the advanced energy. Advanced energy, many of you must be working like now the hydrogen energy and all different type of energies are coming up. Advanced material. So in this segment where the polymer is coming up. Polymer, it's not a very old story. Earlier, when you go to the, somewhere we don't have those polymeric metals. Today, that's why I put it. How you will see polymers? You don't imagine, you don't see it. Like this is a development done by somebody, and that is a plenty of opportunities. What we are seeing it today. If you say you get up in the morning, you have a brush that is made up of plastics. You have all those utensils that is made up of plastics. Today you go to the agriculture, that mulch films are there, which will absorb water and keep it, retain it for a longer time. We don't need a lot of water. So that is what it is coming up. Then after that, you see, when you go to the office, you have different type of, equipments, mobiles and other things. See, in every part of the life, every span of life, you have polymers. During even to the bed, when you sleep on the bed, then also you are using some synthetic materials based on the synthetic foam or fibers and other things. Your LED, LCD, all those TVs are there. Those are again polymers. So this is polymer is a revolutionary material. You have to take it ahead. So I will just take you a few of those things where this plastic market or the plastic growth is there. Where if you see, like I wanted to show the opportunity to my young friends who can see here today, in 2016, it was around 72 US billion dollar USD in the billion dollar market. So after that, around 10 years, it will reach around almost 1.5 times. So that is the growth pattern in different polymers. You can see it. For this, we need manpower. We need the trained manpower who can do it, who can innovate the things. That's what we are looking for. So people should make themselves ready for these things when there is a growth. And India, as I told you, globally, I think if you see the statistics, around 10 to 11 kg per person uses it, whereas in India, it is around 1 to 2 kg. So that growth would be phenomenal in future by 2026. So our people should be ready to handle these things. And definitely Odisha should not lose the bus during this period. We should also be a part of this developmental phenomena. And these are the different big companies who are there. And if you work in these areas, maybe these are the opportunity. who are the global leaders in chemistry or the petrochemical industries. You talk about Dow, you talk about BASF, Exxon, everybody. And Reliance, as you know, Reliance is the biggest or the largest manufacturer of polymers in India. Around, I think, 8 million metric ton of polymers we make per annum. 8 million metric ton. It's a big number. So we need the manpower. We need that much of talented manpower to handle this size of market growth. If I talk about the global packaging, again, you see this packaging today, everything we get packed food. This 45%, which was there earlier in 2018, it is going to be 55%. It is more than the half of the pie, if you see. It is going, this polymer plastic materials is growing tremendously. So that is what we are looking for to handle this growth, this innovation. We should be, our students should be part of this innovation and take it ahead. I will just put some other thermosets. Those are the thermoplastics. If I talk about the thermosets again, there is a phenomenal growth. If you see, it is growing from the 2000. If I put 2017, it is almost I would say double. And henceforth also, I got this much of data. This also, it is going to grow around 4 to 5%. So that growth is also a phenomenon. In this area, we need people, we need trained manpower, we need the knowledgeous people or the hardworking people where we are doing. So these are the big companies who are in this area. These are the global leaders other than the Reliance in India. So in this case, any of you who are working in this area, these are the opportunities. That's what I wanted to share you, which at least in my time, we never knew what are the opportunities. Today we know these are the opportunities. You can go and you can work in these areas. So these are the different areas what is coming up. If you see polymers in the digital, biological, or the physical platform, as I told you, you know, all of you know the smart cities. Bhubaneswar is a smart city. So many things will come there. So all of you need to work, and those are all materials. And out of those material chunk, definitely polymer is one of the bigger area. And then automobiles, you know, today Tesla is having a car which is fully automatic, battery-driven car. And this Japan Tokyo Olympic also going to have different, you know, pilotless cars or driverless cars. So those are the physical platform materials are there, which are advanced materials. Those are not the conventional materials. And in the medical segment, again, you see here, these are like everybody go for the knee surgery. Those are, you know, ultra high molecular rate polyethylene materials. So those materials are also you can develop. Reliance is working in this area. This is one of our big projects where ultra high molecular weight polyethylene is there, and they are using it for the even the bulletproof jackets and other things. And this is one of this, you know, Professor now Robert Groves, who is a Nobel laureate. He has developed some polymers. You inject these monomers to your eyes, and the polymer is formed, and your vision is rectified. That's what he has developed. So these are the development which is happening here. And of course, all of you know you are using the mobiles. I think few years back, mobile battery was 1 hour, 2 hours. Today it is maybe 2 days or 3 days with the video on. So that is a battery, that is a lithium polymer battery. Then the flexible electronics, which is coming around, which is another big area. Everybody is working on this thing. This is again robotics and other things where people are working in these areas. I think our time is little short, so I will not go through a lot. But sustainability or the recycling is another area where everybody is working, even including Reliance. We have polyester synthesis material. Currently, we are also working in this tire recycling, which is a thermoset. We want to recycle and take it back to the monomers or some long molecular weight, at least some oligomers type of material so that you can use it. Then from a waste polyethylene, you are making your diesel and this diesel and petrol type of material fuels. So that is another area which is a lot of research is under progress currently because we are polluting a lot. So other than that, what our area of research in Reliance, what I am doing or in our polymer industry, we are doing it. The different type of catalyst we are doing it. You talk about metallocene, ziegler-natta, metal organic, and then periodical. These are the different type of catalyst we are working in our research also. Other than that, in the synthesis and process, when we have 8 million metric ton of polymer Reliance is manufacturing. So we have a bigger synthesis or optimization parameter. Then you have to improve it every day. You have to improve the productivity. You have to improve the quality so that you can compete. You can be competitive in the market. So that is what the polymer synthesis we are working with. These are the different areas of polymer where Reliance is involved, and these are my areas of research where we are doing some styrene butadiene or different type of elastomers. And then new materials, what we are working on. There are two things, just I put it where we are currently working. This is a self-healing elastomers, where your tire, today it is getting punctured, and tomorrow we are thinking of a tire which should not be punctured, or different type of self-healing, like if there is a puncture, then automatically it will heal, and that it should be puncture free. And then when we're coming for this, automobile, this is another area where we are currently involved. This is where this next generation cars, where you want your carbon pollution or the carbon footprint is minimized. In those cases, we develop some material where your fuel consumption will be low, or your carbon pollution will be less. It will be like different color tires or white tires, or different things. We are not going to use any carbon molecules. So we have developed a new polymer for this thing. So this is another area. These are our research area. Other than that, this product applications, there are a lot of opportunity for the young scientists. This same product, you can take it to the different developmental by preparing composites, blends, alloys. Then different applications you can use it. Yesterday, suppose one great example of this thing. We had polypropylene. All of us, we know that. Today, all of you wear the mask. Those are made up of polypropylene, and of course, that is how you thought of, okay, these are the masks, these are the polypropylene, you can make a nanowoven fabric, you can use it as a mask. So that is a new area of innovation. You have a material, but you need to innovate the new applications. You don't need to invent a new molecule every time. You have to innovate it. That's what we are looking for. Then the product manufacturing, this is another area where again our scientists can go. Being a chemistry background, we all can go to this area where a lot of opportunities are also there. But then opportunities always come with the challenges. So these are always reversible reaction. All of us, we know reversible reaction, being a chemistry student. So opportunities and challenges, they are reversible. In this COVID era, or the pandemic era, all of you know this digitization has come up multifold. Even a chartwala or the roadside thelawala, he is also using Paytm. That is the opportunity. That is the challenge. So that's what we have come up with this thing. So now a lot of opportunities we have coming out of this COVID era or the pandemic era. So all of you need to think about that, to how convert these challenges into an opportunity. If there is no challenge, there is no opportunity. So this is what I want all young scientists, all young people to focus on this thing, how to convert the challenges into opportunities. So these are, again, I put it here so that these are the different areas where you can work. This is open for all of you, and a lot of innovations needs to be done. Nothing is saturated. We always think that this is saturated. Nothing is saturated. So we can do a lot of work in these areas. But this is one of the most important slide. Again, I would like to say, this is what the product development cycle goes for any material, for any polymers, because these are all materials. So conceptualization, then you are designing and development, then deployment, that is manufacturing, and then maintenance or support. This is what the cycle, product development cycle is. And to cater this cycle, what caliber or what competency should be a young scientist should have? I just put few things after listening from yesterday. They should have a basic chemistry knowledge. Without that, nothing is possible. You need to have your basic chemistry knowledge. If we say that you don't understand the periodic table, there is no scope for you. So even if you have the opportunity, you cannot be successful. You need to have the basic knowledge. You need to have the scientific inquisitive. You need to develop, whenever you see a small thing, analyze that, understand that, you have the opportunities there. Then you should always update your knowledge, whoever, maybe all of us, everybody need to update our knowledge. And the most important is attitude. That is very important in industry. You have to have a can-do attitude. If you say, no, this is not possible, then you are gone. So you are not required at industry. Then adaptability. There are very difficult situations many times. I think all my industry friends, they understand this thing. It is very difficult. You need to adapt with the situation. Today, again, this pandemic era taught us adaptability. Everyday human being, they are prone to go out and enjoy outside. But today, from last 1 year, we are locked inside. The globally, everybody is locked. That is what the adaptability is. Dedication. Without dedication, you cannot achieve anything. And speed of innovation. I think sometimes back, we see this COVID pandemic had told us, even 1 year we can develop a vaccine. Or maybe whatever with class count, 60%, 40%, but speed of innovation is much more important in today's world. Perseverance, you should not leave certain things. Okay, it is done, I cannot do it. So perseverance is another area where you feel that it can be done and we will do it, we will not leave it here. And the communication at the end, where we need to develop our communication skill in all formats, in written format, in spoken, and all other things. These are the requirement for the young people whom I'm talking right now. They should develop these things to go ahead and take up the floor. That is what my message to all the young scientists. And this is from my side. I guess I maintained the time a little bit. Yes, that's all. Thank you.
Yes. Professor Jana. Devasis. Devasis, are you there? Professor Jana, are you there? If they're not able to listen to me, we now invite questions. Dr. Mahanti is here to answer a couple of questions.
I'm Dr. Vishwal speaking.
Yes, please.
Just I want to curiosity, are you using graphene in your polymer or rubber products?
Yes, we are using in some developmental work, because we are basically in a Reliance capacity. We are a commodity chemical or polymer manufacturer, but for applications, yes, we have started using graphene in many applications. Even some of our products, we are using graphene right now.
Okay.
Good morning, ma'am.
Good morning.
Is it Trupti Das?
Actually, I want to ask you that whether the Reliance Industry is working on the development of biodegradable polymers to a large extent. It is not that to a small extent, large extent, because right now we are generating a huge amount of e-waste as well as the non-biodegradable substances which need to be captured. As you said that India is using only 1 to 5 kg of polymers. So we may increase the input, but at the same time, we should be thinking about the sustainability, the biodegradable part. So this is my question, whether Reliance is using anything like that or not.
Reliance is working on the biodegradable polymers. We have developed this material in our polymer. I am also part of that. We have developed that material. So this is a polyester based material. So we will be doing this commercialization sort. Maybe very certain another few years, 2 to 3 years down the line, we are planning to do the commercialization of that. But saying so, biodegradable is not the future. If the material degrades within 6 months, what you will do in some applications. So this is another area where, that's what I put this slide, sustainability and circular economy. Whatever we have, can we reuse that? Can we repurpose that? That is what our thought process, and going ahead, we want to put. We have put a lot of focus on this area also. So this is also in the commercial scale. This is available in the market, real lamp. So from the polyester, we are again the largest manufacturer of polyester in India. And so we are now converting these bottles into these fabrics, and that's what we are doing it. And of course, from the waste polyethylene, polypropylene, we are trying to get some of these wax and quills and other things.
Okay,
ma'am, yes.
But unfortunately, I cannot give time for that. We are out of time.
Okay.
And let us thank Dr. Mahanti. Dr. Mahanti will be there in the panel discussion. If anyone has some specific questions, maybe there will be some time to interact with her. So let us thank her for the excellent presentation. And let us move on to the next speaker, Dr. Mona Lisa Swine. Mona Lisa, are you there?
Yes, sir.
Yeah, please put up your please make her co-host.
To share my screen.
While she is sharing her slide, let me introduce Mona Lisa. Indian Institute of Science Bangalore in and she has 12 publications.
Sorry, sir, there is a lot of noise. Sorry, there is a lot of noise here.
Those
We
who
are
are,
not able to hear anything.
yeah, except the speaker and myself, please everyone mute. Yes, thank you. Her research interests are small molecule targeting for oncogenic and viral protein and nucleic acids. She obtained PhD from Department of Solid State and Structural Chemistry and NMR's Research Center under the supervision of late Professor H.S. Atria. At the end of 2011, she moved to the Department of Chemistry, Yale University, USA, for postdoctoral research. In 2014, she moved to National Institute of Health for another postdoctoral research at the Department of Structural Biophysics Laboratory. She is now employed in NIH as a scientist. The title of her lecture is Small Molecule Targeting for the Oncogenic and Viral Protein and Nucleic Acids. So she will be speaking on this. Just before you start, Mona Lisa, may I ask you what is the local time there in Maryland?
It's going to be 1 o'clock in the middle of the night. Yeah, it's like 12:45 a.m.
Oh, sorry.
So
Please,
it's
yeah,
probably
please go ahead.
27.
Yes,
That's fine. I just have to, I moved to a different place this week and I don't have internet in that place. So I have to move back to my old house and then use the internet. Then I have to travel now 15 minutes.
Okay.
So
Very
it's
sorry.
okay. That's fine.
Yeah.
I need permission to share my screen.
Devasis, please make her co-host. Devasis, are you there?
Yes, yes.
Please
Madam, what is your in which name you are joined?
Setsuen M2.
Sign M2. Right here, we can see her.
And now you can share your screen.
Yes.
Hi, perfect. Thank you. It's just letting me to do some security. Okay.
Yeah.
So
Put the PowerPoint on, yes, in full
yeah.
screen. So your time starts now. You have 25 minutes, please.
Hi, thank you so much, sir, for the kind introduction and making this such a wonderful initiative and giving me the opportunity to participate in this nice industry and academia collaboration symposium. So the title of my today's presentation is a little different. So I'm going to talk about, so it's called Conformational Dynamic Study in RNA and Proteins, studied by solution NMR spectroscopy. So this is just a small part of what I'm doing, what I did previously in Yale and at NIH. So these are just a few publications which I had in my PhD. So I did PhD in IISc Bangalore, and it's a joint department between NMR Research Center and SSU. And from there, I just want to give the outlook of my today's talk. So it's giving an introduction to NMR relaxation experiment, and then I'll give what is the conformation dynamics in the protein, which is specific protein. I'll be talking about DNA polymerase beta, and it's a oncogenic protein. And the conformational dynamics in riboswitch RNA, which can also be involved in different growth factors. So first of all, why do you study dynamics? It's a little different than the previous talks we saw. So first of all, what is dynamics? As you know, in our body, we have different biomolecules like proteins, DNAs, all nucleic acids, RNA. And our bodies have a lot of water. So in solution state, they're like fluctuating at different time scales, which varies from picosecond to nanosecond time scales, and in the slowest of the few seconds. And these conformational motions are always coupled with their function, which is like enzyme catalysis or protein folding or RNA functions. And these domain motions can be studied by different techniques. And one of them is nuclear magnetic resonance technique. And this is the only technique which can study at an atomic resolution level, like each and every residue of the protein or RNA we can study by NMR. And why do you study this conformational dynamics? Like what's the importance it has? As you see in the previous topics, we have drug discovery. So by studying the conformational dynamics, it can help in rational design of the drug candidate. Like if you know which residues are highly dynamics, and we can design a drug candidate which can specifically target those residues or in that binding pocket. So this is just a overview of what is the different conformational motions exist in our body, which is like varies from femtoseconds to few seconds. And as you can see here, NMR can study very fast time second motions to few like slower the few second motions. Oh, sorry. So what I'm going to talk today is about a small experiment of NMR, which we call as a CPMG NMR experiment. And what you see here, like curves like this. Let me move to the pointer mode. Let's point. So it shows nothing like as a exponential decay plot. So what I study here, like in each and every experiment, these are like pulse, which are nothing but the electric pulse, and we have the total delay. So the product of the tau, which is called the pulse, and the number of the pulse, which is known as by n, is constant throughout the different experiments. So each of these dots are each a single individual experiments. And the way we study in NMR is we saw a single peak for a single residues. And if there is a change in intensity of the residues, then there is a conformational exchange going on. If there is no change in the intensity, then there is no conformational exchange. And these are like basically 2 states or 3 states. So this is an example showing that if you don't have any dispersion, it will be straight line. But if you have a dispersion, it will be like exponential decay curves. So the specific system what I studied when I was at Yale University is called DNA polymerase beta. And this is the kinetic scheme of the DNA polymerase. So this is the enzyme or protein which then binds to the gapped DNA. As you see, there's a gap. And then it, so this is how the resemblance X-ray structure of the protein, which is open conformation. And then when it binds the DNA, this finger domain of the protein folds like 20 angstrom to tightly bind the DNA, and it has a different conformation compared to the apostate. And then it binds the correct nucleotide, which in this case we say by DNTP. And then this domain, the protein which moves another 7 angstrom to form another very tight rigid complex to carry the catalytic function. And in this catalytic scheme, there is also one step where it happens a lot of conformational rearrangement upon binding the nucleotide. So what I have did is like study the dynamics in each of these individual states of the protein. So this is how the dispersion plots look like, and I'm showing here just for the apoprotein. As you see here, we did the experiment on both 800 megahertz magnet and 600 megahertz magnet. And these are the individual KX of these residues. So total there are 18 residues which give a conformational dispersion plots like this. And all of these 18 can be globally fit with a global KX parameter of 1400 second inverse. So this indicates that these 18 residues are fluctuating at a single time scale, which is called as a global time scale of 1400 second inverse. And there are few residues which are very fast, but I'm not going to talk about them because this is beyond the detection of the technique I'm using. This is how the NMR spectra look like. And as you see here, I'm showing in the red, this is the apoprotein. And then what we did, like we add the gapped DNA, as you see here, there's a gap here. So this double-stranded DNA, there's a gap opposite to G. So you add this gapped DNA and then collect the spectra. And as you see here, each and every spectra, residues of this protein has been shifted, which indicating there is a change in structure and a conformation of the protein. I also studied in the binary complex, but interestingly, in the binary complex, there is none of the residues chosen in dispersion in the CPMG timescale, which means these bindings, which had been showing previously, the conformal exchange has been quenched upon binding of the double-stranded gap DNA. As a resemblance I have shown here in the apostate, E21 was showing 1200 second inverse dispersion, whereas in the binary, it has been much reduced, like 400 second inverse. So the DNA binding quenched the millisecond motions in the polymerase beta. This is how we mapped all the residues which shows dispersion in the apostate to the crystal structure, and most of them are over this domain. And if you remember, this domain folds 20 angstrom upon binding of the DNA. And further, what it did, like this is a different mathematical, using different mathematical equation of this. We try to plot what is the REX, we call as exchange, versus the chemical shift difference. And all of these are overlapped in this domain, sorry, in this plot. So from this slope, we can get the population. And what we get is like the PA we call the major state has a 95% population, whereas the minor state has a 5% population. And the minor state is nothing but the DNA bound state. So what it means? It means like even in the apostate, in the absence of the double-stranded gap DNA, the protein is in conformational exchange with its bound state. So it knows where it has to go when it binds the DNA. And that minor state is in 5% population. And what we looked at is like we plotted those dispersions. This is not just the dispersion we call the R2s, which are residues showing much higher flexibility. And this is in the apostate, and this is in the binary state. And all of these residues which are being circled, they are the oncogenic residues. Means the mutations of these residues causes cancer in this protein, and even for the binary state. So as you see here, those residues are like circle, they're present in the hinged regions. So this indicates that the residues which are oncogenic, there needs to be highly flexible. So thus, upon targeting these residues, we can achieve some drug candidate, and targeting in different domains, it will be helpful to can achieve a drug candidate for this protein. And then I further studied that ternary complex, where I call this is done by ITC. So what I'm doing is I'm adding DNTP, which is this is correct nucleotide GC, as is a G, the C is the gapped DNTP, and it gives a KD of nanomolar binding, whereas the when it GA, which is a mismatch complex, it gives a KD of millimolar binding. That means very weak binding. So the system itself knows which one is the correct nucleotide and binds tightly. But I'm also interested in studying this by NMR. So as you saw here, this is the NMR spectra, where the red one is binary and the blue one is matched complex. Similarly here, the red one is binary and the blue one is mismatched complex. As you see here, there is huge difference between the match and the mismatch complexes, as you see compared to the binary state. So that means there is a huge difference in the structure of the match and the mismatch complex. But from X-ray, which actually crystallizes a single conformation, doesn't have that much difference. So the NMR is the only technique to tell, even the structure is very similar, is there any conformational differences or dynamics difference. And then what we did here is we plot the chemical state perturbations. That is the chemical state difference between the binary and the match for this plot A, and then the binary between the mismatch for this plot B. As you see here, all these residues which are shown higher dispersion compared to the red line, they're all mapped in this domain. So if you remember, this domain is when it binds to the correct nucleotide, it folds another 7 angstrom. Whereas in the mismatch domain, there's the difference in the chemical state is very little, indicating that not much change in the structure. That means the protein remains in the open conformation as equivalent to the binary state. So this is just a single residues I'm showing here for T292. The incorrect residues shows in the green, whereas in the correct residues shows a huge shift. We did the CPMG plots, and we see 3 different time zones, 3 different KX. So we thought of doing a different way of observing them, like looking at the line width. So in NMR, not only just the shift matters, but also how the broad or soft the peaks are. So by measuring them, what we found that the K off is 5.4 second, which is almost half of what is the K pol is. Like we know what is the polymerase, it's about 10 to 15 seconds. So what we see is we wanted to first in the beginning, we saw like which one is the slowest step, that is which is the rate limiting step of this kinetics. And we found that when the enzyme opens off, like after insertion of the correct nucleotide, when the enzyme opens off, that is the rate limiting step. As we see, this is slower than the K pol of the enzyme. This is just a brief summary slide for the part 1. As you see here in the open conformation, this protein exists in 95% of the population, and in the binary conformation, it exists. Whereas in the open conformation, there is a minor state, which is like 5% of the population. So there in the apostate, there is conformational sampling, which dominates the function of this protein. Whereas when it binds the nucleotide, whether the correct or the incorrect nucleotide, is the induced fit mechanism that dominates the function of this protein. And then coming to the second part of my talk, that is conformation dynamics in the riboswitch RNA. So riboswitches are these are gene regulatory systems, and they're mostly present in the 5' terminus. So this is the green run is the coding strand, which codes for the protein, and then the 5' and the 3', these are the non-coding strand. There are few riboswitches which are present in the 3', but most of them are in the 5' UTR region. There are specific riboswitch I'm going to talk is called as adenine riboswitch, or we see ART riboswitch. This is how the schematic representation of this RNA looks like, and it has a very dedicated ligand binding pocket, as you see here. So this is called M-, means it's an apostate. But when the ligand is bind, it binds in very high affinity, like nanomolar binding. There is a huge conformational change happens, which triggers its function of the RNA. So this is how the secondary sequence, secondary structure, we call secondary structure of the RNA looks like. As you see, there is an open pocket, and these are just loops. When you see a loop like over here, that means they're not hydrogen bonded to anything. And when you see a stem, which are given by the dot lines, that means they're hydrogen bonded to each other. So over here, when it binds to the ligand, it makes changes in the conformation. As you see here, it establishes a new stem loop, and there is a much rigid structure upon binding of the ligand. And why this is called as a switch? Because it exactly works like as on and off switch. So this is the off state, it has no function. And when it binds the ligand, it becomes the on state, and it carries out the function of the RNA. This is just a schematic representation of how we purify the RNA. I don't want to go into too much detail, but it's very different. RNA is completely different compared to the proteins or DNA. So we use solid phase synthesis that has a streptavidin biotin tag on it, and then we add the DNA. So each time the DNA can be recyclable, like means once you make the RNA, you can go back, clean the DNA, and the DNA will be still intact to the bits. And you supply the enzymes, and we can make the RNA. And then we purify by the urea gel or by gel filtration column. And finally, we see a pure RNA. So this is how the timescales, as in like proteins, RNA also exist in a similar timescale motions, and it has different, these are the reporter atoms of the RNA for the NMR. So this is how the secondary structure of the adenine riboswitch RNA looks like. The ones I'm showing in green, they're like hydrogen bonded over here. And this is the opposite NMR spectra of the adenine riboswitch. So in the upper spectra, since this part is a big loop, we will not see any of these residues, neither from this loop. But when you add the ligands, there are a lot of residues. So as you see here in the orange, those are the residues primarily indicative of in this binding pocket. So it clearly indicates that which are the residues binds upon adding the ligand called adenine. So now we see this is well studied by X-ray structure. So this is how the X-ray structure look like. And what I'm showing here is the delta chemical shifts. That is this is the what's it called amide. This is proton N15, and these are called as the N1, N3, N7, and N9. These are long-range nitrogen shifts. So as you see here, the N1, N3. If we can observe these residues even in the loop regions, but not the immido residues. So this is what the chemical shift difference or CSP plot we see between the ligand bound and unbound. And then when you map them to the structure, as you see here, the blue ones are in the stem regions and the red ones are in the loop region. So using this technique, which is N1 and N7 technique, we can observe the residues in the loop. And I further developed the method making this N1 and N7 to CPNG experiment, like measuring the relaxation of this RNA using N1 and N7 probe. So this is what the KX of the RNA look like. As you see here, all of them residues show a single global KX of 1200 second inverse. And this is mapped to the structure, which is shown in the purple. They all are in the stem region. Then we further did the N1 and N3 residues, and we can see the loops here. As you see here, when it binds the ligand, which is adenine shown here, the P1 and P3 stem, we know that it stacks from the biological experiments. But interestingly, the P1 P3 stem residues, which shows dispersion, they have a global Kx of 600 second inverse. And the loops has a global Kx of 1300 second inverse. Now, if you compare this to the apo Kx, the loops, which has the same Kx compared to the apo, whereas the stems, they get more rigidified compared to the apo upon binding of the ligand. And we also studied the X-ray structures. This is studied by my co-authors. So from X-ray, we know there are 4 states exist. There's 2, this is the primary apostate, then there is a minor apostate, and there is an inactive state, which is ligand-bound inactive state, and there is a, this is the ligand-bound state. So we are able to establish the relation between the X-ray structures and the dynamics in NMR, studied by NMR. With this, I'd like to acknowledge my mentors and their lab. So Professor Patrick Loria, this is from the Yale University. In his lab, I did postdoc for a few years. Then moved to Yun Sing Wang lab at NIH. And this is my present lab, Stuart Legris lab, again at NIH. And my sincere thanks to all my teachers, including all the MSc teachers which are present now in the audience. And with this, I'd like to take any questions. And before this, I take any question, I just have 1 minute to contribute like any suggestions about how to improve the efficiency of the students. Like I see here, I have students coming from the high school and even college grade come and do some experiments, like doing a summer training between few months or doing as a coursework, like taking few credits for their course and coming to the lab and doing few hours a day, few days a week, and getting some credits for the course, getting opportunities to research environment and different other industry environment. And also we have like industry and NIH collaboration. My current PI, he used to coordinate a symposium. We meet quarterly. And so this kind of like collaborations meetings between the industry and academia will also helps. But we create a small funding and give one student small funding to go to the other lab, like a month or like few days, just do some experiment and get some exposure. So yeah, with this, I'd like to thank and happy to take questions. Thank you.
Yeah, myself Tapan Raut. So I have not questions, but rather I would like to know more. So you have done very good basic research, but if you think the objective and your research is heading towards to achieve something, some goals, am I right? So can you explain those, how this can be applied to get some kind of application information with your research? Yeah.
Yeah, so if you see here, like these days, NIH is prioritizing more research in basic science. Like if you know more strong of the basic science, like how does this virus interact? rather than like understanding the most basic, like how this protein, so the DNA polymerase beta, it is known as most of the 50% cancer patients that there is a mismatch in the DNA repairing. So it fails to repair the gap in the DNA, and that leads to a lot of cancers. So understanding this, like the structure, if you don't know the structure, you cannot design any drugs candidates. If you know the structure by NMR or X-ray by cryo-EM, so these days everywhere there is a lot of input going on about the cryo-EM. So even the X-ray cryo-EM structures of the COVID vaccine, it came like in very quick. It came like in months of time. And when you get the structure, you can design the drug candidates. So this is forming a basic background of primarily forming a basic background of, okay, how do you know the primary step, like how do you know the structure? If you can design the drug only when you know the structure. There is a lot of computational ways people can get the structure. From like RNA, it's very hard to get the structure. So there is a lot of computational inputs, but again, it is a computational way. If you have more experimental evidence, it can always help the computational way of predicting the correct structure. So it's basically my research is like structure deriving function. So structure function relationship and then drug targeting. I did not talk about the most recent work I'm doing in my Stuart Legris lab, where we have screened a huge library of the small molecules. We have like few million small molecules we screen for the RNA and for the proteins, and now we're doing for the RAS protein. So if you know, RAS is like 70% cancers, pancreatic cancer, like every cancer has the RAS protein. So there is a huge effort going on, RAS initiative to screen the compounds, like all industry collaboration with China, India, and we screen those compounds. So we form the primary basic, like the chemist synthesize the compounds, then send it to us. We screen those.
Thank you.
Thank you
Very much.
Your point is, your answer is well taken. So no time for any more questions, but I'll simply put one question. Your experiments are basically in vitro experiments. You are putting the materials in NMR tube, and as you know, the NMR tube is rotated at a very high speed. And then you are measuring these vibrations, which is very high, about 1400 times per second. So how is it this thing very much close to what is happening in vivo?
Right, so this is what I'm doing is solution state. We don't rotate here. So solid state NMR, they put the sample and rotate it. But what we are matching is the time scales. I talk about those few seconds or is few milliseconds. Usually in our body, there are broad range of time scale motions. Few of them are like few seconds, and some of them are like cannot be detected by any other techniques, which are like very fast fluctuations, like bond rotations. In chemistry, we see the vibrations, like carbon-hydrogen bond rotation vibrations. Like when you have these bond rotations can affect in the oncogenic state. That's what we are studying. Like in the normal patient, is this kind of kinetics is the same, the conformational exchange is the same state, or is a different time scale in the oncogenic mutants, which are like cancer patients we found. So we have, there is in cell NMR studies, people study where they directly take the samples from the cancer patients or from mice, and without doing anything, just look at the NMR, put it in the magnet and just look at which signals we are seeing. There are protons, we look at them instead of looking carbon and nitrogen. There's no labeling required, no in vitro sample preparation. So yeah.
Okay, Dr. Mona Lisa, great work. You are doing wonderful work, and your suggestion about having some more trainees in with some funding is well taken, and we shall discuss about it. We know this is the kind of way elsewhere in the world, but we have to adopt those settings. Means it involves so many parameters, and we shall definitely take that point into
Like
consideration.
introducing
Thank you
very
very
early
much.
stage, like at the level of the college. I have one high school students to come in and do some experiment.
Sure. So let us join hands and thank Dr. Mona Lisa Swami for our wonderful lecture. And thank you, Mona Lisa. And we are again sorry for the time. Please, good night. Good night to you. Thank you.
Thank you, sir. Thank you.
So we
Bye.
shall move to the next session. Over to Dr. Devasis Mahanti. He will be anchoring this session. Yes. Dr. Mahanti, please.
Thank you, sir. Thank you providing the opportunity me to anchor this session. This session will be chaired by Dr. Saurabh Chandra Das. Now I'm going to introduce Dr. Saurabh Chandra Das. Professor Dr. Saurabh Chandra Das did his MSc in chemistry in 1976, PhD in 1994 from Utkal University. After working as lecturer, reader in chemistry in various colleges of state, he retired from Salipur Autonomous College, Salipur. 10 scholars have been awarded PhD degree under his guidance from Utkal University. He has published 52 numbers of research papers. In connection with research presentation, he had extensively traveled to US and UK. He was the secretary cum treasurer of Odisha Chemical Society and the president of Odisha Chemical Society in the year 2017. So now I request Professor Das to take over the meeting.
Or hey.
Yeah.
So thank you, Mr. Organizer. And
video टा on करो समझ देखो तो
देखिए कौन ठीक है ठीक चला जी thank you all organizers for giving me this opportunity to share some of my feelings with the speakers and also audience. So this session was supposed to start by 11:15 as the reschedule is going to me, but we are running late by 20 minutes, I think. So that we shall have to cover within this time span of 100 minutes, we shall have to cover 4 speakers. Dr. Subhash Malik, Dr. Nilkamal Mahapatra, Professor Suthiranjan Parida, and Dr. Pranav Kumar Patra. 4 speakers, I think they must be available. And let me invite first Dr. Subhash Malik, who happens to be the director, LN Intech Services Private Limited of Bhubaneswar. So you would like to apprise us on a very recent topic, that is innovation in wastewater treatment technology, which is of course really that is the need of the hour. Dr. Malik, please start your speech, and the time schedule is 25 minutes, and you can highlight all your achievements within the same stipulated time. Dr. Malik, please. Yes, please.
Good morning, all of you. Respected chairman of the session, the respected president of OCS, Professor Samuel, respected president of 4MSI, Dr. Biswal, and eminent scholars, professors, scientists, and students, and my dear friends. I am myself Dr. Subhash Chandra Malik from LN Intech Services Private Limited. Actually, this LN Intech Private Limited, a R&D and technology-based company founded by a group of scientists and engineers. We have left some high-profile jobs and started this company. And the main idea of this company is to disseminate technology to the ground for implementation in various sectors. So our main area of basically in Odisha, and we work in pan-India basis, also in abroad. So we have expertise in many other areas, but while doing that, we thought water treatment will be another area where we can do much more. So with that, we have started our Elcogen technology, one of the versatile technology which add to the value to the conventional treatment process, which failed in many ways. So justice to provide the new generation to the next generation people for the clean water and sufficient water to survive. So before I going to discuss, I will give my topic will be I will give a overview of water wastewater treatment issues, present treatment process,
and associated issues, need to move from conventional high operating and synthetic chemical based technology to green and fast treatment approaches. And I will give a bureau of our Elcogen treatment technology. I've been knowing that the present scenario that water is rapidly contaminated due to urbanization, improper agricultural practices, use of high large scale pesticides, social and religious practices, and a recent pandemic, and given another challenge for us, where a large number of water pollution is taking place due to large use of surfactant, soap, detergent, salt, alcohol, quaternary ammonium salts. So this is another challenge. So we need to do something to provide a clean water and a sufficient water for our society. So again, that industrial pollution, as you know, that a large number of water waters generated from industries, it is treated or untreated, it is going to the our river body, our water resources, contaminated entire water bodies in downstream, and create lot of problems in our water systems. So we have to do something so that we can get a good quality of water for our all applications, including agriculture, drinking water, industrial application, and actually most of the water now unfit for human consumption as well. So as you know that water is a fastly a dwindling resource because of water pollution and climatic changes and the environmental pollution. So most of the part of the world is going to face a severe crisis of water shortage by 2025 or in the near future. So we have to move from the conventional water treatment technology to the some new kind of approach, where new and a fast kind of approach, so that we can provide clean water for our society. So city, then in recent time there are a lot of news are coming. So most of the cities in the world are facing a severe crisis of water. So the cities are, you may see that Cape Town, Mexico, Cairo, in India also, Chennai, Bangalore, and some other parts, smaller cities also are going to face a severe crisis of water shortage during summer. Even in other season also, they are not getting good quality of water for various applications. So challenging pollutants, our water bodies is no more that conventional pollutants like COD, BOD, and some other inorganic pollutants. But nowadays this water is being contaminated with very toxic materials like arsenic, mercury, hexavalent chromium. Then you will move on cyanide, fluoride. Then conventional, you know that BOD, COD, pathogen, virus, bacteria, pesticides, herbicides, germicides, synthetic colors, and is extensively used in our agricultural field. Again, that our industry is coming polyaromatic hydrocarbons, polychlorinated biphenyls, phenols, heavy metals are very challenging pollutants and posing a very serious challenges to our conventional water treatment system. So we need to move from this conventional water treatment system to a very modern approach, and where less chemical intensive water treatment systems. So wastewater, as I told that we have that our water system is designed for removal of TSS, TDS, this pathogen, bacteria, virus. We have this primary treatment, secondary treatment, and tertiary treatment, which is involved in the same conventional chlorination, some oxidation systems. But again, after little modern approach, if you go to some advanced process like RO, microfiltration, ultrafiltration, nanofiltration, some other filtration system. But all these techniques also generate a huge quantity of backwater. So that has to be again treated, or it has been thrown to somewhere. So then we have to need to something so that we can justify a water system where that we will generate less that reject water, as well as less chemical intensive water treatment. So in this connection, that you can see that all over the world that a massive investment is being done to put the water treatment plants. But do the water treatment plants are giving justice? If we see the most of the modern standard, actual standard, the most of the discharge water from this water treatment plant is unfit to discharge to the water bodies, the river, lakes, and even seas. And we know that if you pay something, you can discharge any kind of water to our water bodies. So are we doing the justice for this? I certainly it is no. So then what to do? Then we have to move from this conventional treatment system to non-conventional or to modern approach, wherever it is possible, and less cost with less capital intensive, less land requirement. So in this direction, our team, that LN Intech Services Private Limited, is develop some kind of new approach, new technique called Elcogen technology. So this Elcogen technology is nothing but a simple technology. It is a composite treatment process. It is mainly based on the electrochemical as well as some advanced oxidation, that is called hydrodynamic cavitation, and that micro nanobubble system, and some another electrochemical system, that corona discharge based non-thermal plasma based. Then the high performance conventional chemicals like alum would converted to another high performance chemical, that your PSC will be converted to another high performance that multifunctional chemicals. So with this approach, this technology designed, and we are already tested this technology in various parts of our country, including metallurgical industries, in domestic household wastewater, electronic industries, then your petrochemicals, various applications, and a fantastic result we are getting. So I think this technology, if we adopt in the near future, we may give a better result, and we can justify that better water purification systems. So this is our laboratory demo model. This is a Elcogen model. Then this Elcogen system already a trademark system. We have taken trademark for this system. So we are moving with the large implementation of this technology in industries as well as in municipalities and many other areas. So what is this? Is the component of our Elcogen technology. What is the main component of our Elcogen technology with the advanced aeration using micro nano bubbles? So you know that our conventional aeration system is just water blowing and blowing, so it is little effect. But we have moved from that to go to advanced aeration using micro and nano bubbles to various advanced techniques. Then we have moved to the aeration oxidation using hydrodynamic cavitation. So hydrodynamic cavitation is another interesting subject. In our country, there are a lot of expertise in a laboratory scale, but with that concept, we have taken to the very large scale applications. Nanomaterial assisted nano bubbles based aeration. When we are taking this micro nano bubbles, we also take the advantage of the nanomaterials. So if we put little nanomaterials, by that time this nano bubble and the synergetic effect of this nanomaterials give a wonderful results in many areas. So electroflotation, electrocoagulation, and electrooxidation. Being a electrochemical scientist, we understand the nitty-gritty of the and the pulse of what electrochemical system, how the electrochemical system works, what kind of mechanism is happening during electrolysis. With that, we have taken the that understanding and design the total electrochemical system consisting of electroflotation, electrocoagulation using various soluble anodes and composite anodes, as well as that alloy-based anodes, and with the combination with electrochemical oxidation using very catalytic active anodes, including BDD, barium doped diamond anodes, and conventionally this platinum alloy, that mixed metal oxide anodes, manganese dioxide, lead oxide. So wherever depending on application, we take the advantage of the that electrodes. We apply this for the oxidation, electro oxidation. Then we can micro dosing of electro-generated powerful oxidizing agent, reducing agents, and benign chemicals. While doing, we don't we add a lot of chemicals from outside. So what do? We put some that electrochemical system. We can generate powerful oxidizing agents like hydroxyl radicals, hydrogen peroxide, peroxy compounds. So like a lot of oxidizing agents generated in situ, and oxidize all these powerful that refractory organic compounds, and it will destroy completely. And also we take the advantage of the non-thermal plasma-based treatment, which can generate that oxidant and kill all these bacteria and all these organic pollutants present in the wastewater systems. So if we'll go micro, this is the some details of the micro nanobubble of the wastewater treatment. Very small gas bubbles, high rise velocity. In conventional water that aeration, it is just bubbles come generate and it goes out. In this approach, it will remain in the water for hours together, and it gives the oxygen level in the water. If we suppose a 3 to 4 ppm of the oxygen oxygen in the water, it is there. So it can go to up to 15 ppm, 20 ppm, like that, DO level. So that it effectively multiply, and it create a real oxidizing atmosphere in the water. And also that it is that pollutants, that mainly TSS, it will come to the surface, and it is very high surface area. Definitely, that is tiny bubbles make a wonderful effect inside the water treatment system. Micro nanobubble technology will be the next water treatment technology, and it is a new concept, and slowly devices are coming. So it will take away the from the that conventional the aeration system. Micro nanobubble treatment will replace costly toxic, costly like ozone, and toxic like chlorine, and with zero chemical applications in the water treatment system. You will see that this is the nanobubbles in the range of, this is the micro nanobubbles. If we have less than 50 micron, you can say micro nanobubbles, it make a wonderful effect into the water. And this is a bubbles of 1 to size. This is a micro nanobubble of the size of less than 50 microns. This is the how the surface area of the bubbles you can see, and they are because of high surface area, so it is make a definite changes in the water system. And these are the how micro nanobubbles are created. There are a lot of devices are there. We are taking the we have developed a different approach. We have some porous membrane system with the application of with some synergic effect of that some electrochemical device we introduce inside, so that it will create a very effective nanobubbles inside the water system, so that it will effectively be multiplied, and it will give a very good effect in the water treatment system. Then will come hydrodynamic cavitation in water system. This hydrodynamic, we know that sonochemistry, that when that application of energy, that bubbles burst, the tiny bubbles create formation of tiny bubbles and the collapse of the tiny bubbles. When that tiny bubbles collapse, it create enormous energy inside the surrounding areas, and even that impact that while collapse, that it create a sphere where that energy is equal to 3000 degree temperature effect. So that it will by this way what will happens, that create lot of radicals inside the water system, and create a very effect into the water system to kill all these bacteria, virus, organic pollutants, and also take away all these soluble that TSS to the surface. So it's a very interesting subject and apply to many areas. And in our country, that ICT Mumbai has done excellent job in these areas. So with that concept, we have developed that a similar kind of setup, and by generating the hydrodynamic cavitation by throttling, such as by venturi orifice, and by hydrodynamic cavitation, we can able to treat any kind of wastewater with the complex or to complex nature. So these are the devices. You see that when water enter through a narrow tube, and when it is expanded, so the bubbles, that tiny bubbles created forms here and collapse in the expanded area, and create a huge impact where you can get energy. You can that mainly that mechanical energy will be created into heat energy, and the heat energy that create a lot of free radicals like hydroxyl radicals, oxygen radicals, as well as generate hydrogen peroxide and many other radicals forms, and create a very powerful oxidizing atmosphere. So kill all these pollutants from the wastewater. So these are very interesting subject, is part of our ecogen system. And also it is defibrillate the pollutants like biomass, some pathogens, bacteria. It will like if you will squeeze something and relax something, so it will be the water that water will goes inside, and it will just break inside like that biomass. Lignin is a very stable compound. It generally don't destroy. We have to burn it. So if we put under this cellulose, hemicellulose is surrounded by this lignin. When you put this kind of tiny powerful bubbles inside this biomass, and when you relax it, so it will defibrillate the entire biomass, and it will come out from the water system and fragmented, and it is liable to decompose further by the action of the various radicals. So this is a powerful tool for the modern this is one kind of modern technique for water treatment systems. So this is also a part of our ecogen system. So then, as I told, that electrochemical coagulation, electrochemical oxidation, and electrochemical flotation. So what is electrochemical coagulation? When we add this, we know the conventional coagulation as like we add alum or something organic coagulant, flocculant. So it is coagulate the suspended particles. So come out and take out from the system. But it is very large chemical consuming, and the synthetic flocculant you are using a large number. We don't know what is the life cycle of this. What is the that impact of in the next 5 years, next 10 years, next 15 years. So we need to understand this, and we move to some kind of a system where we know how it works, and there is a less chemical intensive and act in a micro and a nano scale. So in this case, that instead of adding the alum, instead of adding this ferrous sulfate, what we do, we use the soluble anodes of aluminum, titanium, even alloys of aluminum, alloys of iron, or some other metal alloys, so that depending upon the pollutant present in the water system, so what it will act is a micro scale, a nano scale, and a nano dosing takes place. It neutralize all these particles as positive ions, negative ions, neutralize, and make a huge impact in a very quick quick succession, and give a complete removal of the coagulation and a complete removal of the suspended particles from the systems, and as well as make a flotation, so that it will remove from the system. Similarly, electrochemical oxidation is general power. It is as such it oxidize the compounds, reduce the compounds, and also generate the powerful oxidizing agents like peroxide compounds, hydrogen peroxide, that ozone, as well as other free radicals, and also hydroxy radicals. So destroy all these compounds like polyaromatic hydrocarbons, that polychloride biphenyls, and many recalcitrant and stable organic compounds. So non-thermal plasma is another interesting area where that is also part of our system. So we apply this non-thermal plasma so to generate these powerful radicals. So wherever all these fails, we apply this non-thermal plasma treatment system to treat the water. So when there are about COD contain around 60,000, 1 lakh level, we can use this, and a lot of heat generated. That heat we can utilize for various other purposes.
Dr. Malik, 2
So
minutes. Dr. Malik,
I'm completing. Yeah, yes, sir. That sir, I'm completing, sir. Multifunctional benign chemicals, we will move. We are giving a platform to moving from the alum, conventional alum, to aluminum persulfate, which not only coagulate but also oxidize. It is a very synergistic effect. Similarly, polyaluminum chloride to modified aluminum hypochlorite system. Similarly, material based reducing agent reduction of hexavalent chromiums and all. So these are you see can all these that very toxic industrial wastewater, how it can be cleaned and give a very very pure water for recycle. So advantage already I told that you give a very cleanest water and less chemical intensive, less that it give a very that high, but in a short time it will give a high output of the treated water. We can apply to many industries and also that municipalities. So our system is applicable to almost all sector, all industrial sector, domestic, in that household, everywhere you can even you treat the that in fluoride, cyanide, arsenic in a single system, and I think this system is working a wonderful way for giving a better quality of treated water for our society. So thanks to the organizers for giving me an opportunity to give an invited talk in this forum. Thank you all.
Thank you, Dr. Malik. So from my side, one question, not exactly one question, just a suggestion from you. What should be your message for the young students to be attracted for your scheme? What should be your message?
Sir, actually, I have not gone in details. We work in a reverse way. Whatever there are a lot of, if you see almost all here and present in this forum, almost all people might have worked in the water treatment system. So how their water treatment system ideas or research can implemented, that is my message. If you know something, so discuss with those who are applicants, so their ideas can be implemented successfully for the water treatment system. So idea should not remain as an idea; it should be implemented somewhere.
Thank you, Dr. Malik, and you have really given an excellent idea about this water treatment, the plan you have done in your organization, and I hope our generation, they will be extremely benefited from this scheme and they will be attracted for your scheme also. Thank you very much.
Thank you, sir.
Next, I may request Mr. Nilkamal Mahapatra, who happens to be the Vice President, R&D, NCPU Inks and Coatings Private Limited. And his topic is flexible packaging and allied industries, a lucrative career option for ODIA students. It's a very good thing because packaging really that attracts everybody whether inside the packet there may be something or nothing, but the good size, good attractive packet must attract everybody. So we hope your lecture will be more interesting and it will be giving a very good signal to our young students. Yes, Mr. Mahapatra, flexible packaging and allied industry. Please, the timing is 25 minutes. So please stick to the time. Thank you, Mr. Mahapatra. Please go ahead.
Thanks a lot, Dr. Das, for your kind introduction in the beginning.
Yes.
And very good afternoon to all of you, and good morning to our friends in US. My company, Ansefo, and I are grateful to Professor Samuel, President OCS, and the organizing committee for providing me this opportunity to deliver this presentation in such an important concept. To respect the stringent timeline, let me jump into the topic immediately. Give me some time just to share my screen. Yeah, as told by Dr. Das, I'll be describing or delivering the presentation on the flexible packaging and allied industries, a lucrative career option, and of course for our ODA students. This presentation demonstrates the wide spectrum of career opportunities available in flexible packaging industry for students or newcomers who wants to make potential future in rapidly growing industries. So the industries including printing inks and coatings, packaging films, lamination adhesives, and packaging are expected to witness significant growth in the future due to its increasing demand in end-use industries such as food, beverage, cosmetics, and personal care, and of course, pharmaceutical. As these industries emerge, the number of career options will potentially and proportionally increase. A brief description about all these industries related to flexible packaging will be provided in these slides. Let's look at the, now let's have a quick look on the contents we are discussing in this presentation. Overall, I shall focus on the why factor. That is why flexible packaging would be a suitable option for the newcomers. Flexible packaging is a means of packaging products through the use of non-rigid materials, which allow for more economical and customizable options. Flexible packaging has grown popular due to its high efficiency and cost-effective nature. This packaging method uses a variety of flexible materials, including foil, that is aluminum foil, plastic, and paper, to create pouches, bags, and other fillable product containers. Flexible packaging are particularly useful in industries that require versatile packaging, such as food, beverage, personal care, and pharmaceutical industries. Let's have a quick look on the examples of flexible packaging. This shows that almost all the products all of us use in our daily lives, including groceries, shopping bags, and medicines, to all the delicacies. In short, almost everything comes packed is with flexible packaging. This graph expresses the consumption of flexible packaging globally. Asia Pacific is clearly the region which consumes almost half, that is 47% of the total flexible packaging consumption worldwide, followed by North America and Western Europe by 19% and 17% out of the total global consumption respectively. Eastern Europe, Middle East, and Africa consumes 7% each, by South and Central America, I think by 4%. The global flexible plastic packaging market size is projected to grow from 160.8 billion USD in 2020 to 200.5 billion USD by 2025 at a CAGR of 4.5% from 2020 to 2025. The figure shows in this graph represented in Packaging Outlook report clearly shows that how rapidly the market size of flexible packaging has been consistently expanding with every passing year. As per the Smithers-Pira and PTIS 2017 reports, the market size of the global packaging, including flexible packaging, has largely grown in the past decade, and it is expected to grow tremendously in the next few years. This slide expresses global flexible packaging consumption by packaging materials, and clearly flexible packaging occupies the largest share, that is almost 39% out of the total packaging materials consumed, followed by rigid plastics with 8%, and then metals, glass, paper-based containers, and liquid cartons with 11%, 9%, 7%, and 6% share respectively. If you see this graph, it shows the total packaging materials consumption in Indian packaging industry. Flexible packaging materials share with 22% of the total packaging market, which is quite a large number, followed by the rigid packaging material with 18% share, printed cartons with 17% share of the total packaging market. Packaged food, the main reason that is contributing in the growth of Indian flexible market. This graph clearly portrays the growth of organized packaged food growth in India, which is proportional to the growth of Indian flexible packaging market. According to the reports, the flexible packaging market size in India has the potential to grow by 9.76 billion US dollar during 2020 to 2024. This pictorial representation shows the complex web of packaging value chain formed by raw materials and packaging material manufacturers, suppliers, retailers, brand owners, consumers, and other players related to these mentioned members of the supply chain. This web obviously seems complex, but along with the complexities, it creates ample number of work opportunities. As you can see, more than 20,000, it's a quite huge number, packaging professionals are currently working. However, the requirement is even more, that is 35,000. Isn't it great? To explore these opportunities, I must say the requirement of these potentials, the requirements of packaging professionals are visualized worldwide. You know, just a couple of days before, I conducted a patent search in a professional patent database, and the results are very, very encouraging. Flexible packaging is not only business-driven industry, but also an innovation-driven industry. Shows the major patent assignee related to this industry in several countries of the world. The geographical spread is huge. This graph represents the major number of companies work to develop and innovate products based on newer chemistries and engineering, of course, engineering. So they work side by side. Printing inks, varnishes, printing and related services are among those industries in which major number of innovation work has been done in the past few years. The scope of innovation and development in the packaging industry has been growing with every passing year. Now, coming to the chemistry side, we briefly discuss about the few major industries related to the flexible packaging sector. These industries include printing inks, films, substrates, coatings, converters, and lamination industries, lamination adhesives. If you consider the printing inks, let's start with the printing inks are majorly comprises with polymeric resins, ink binders, varnishes, pigments, solvents, and additives. Polymeric resins or ink binder constitutes from 15 to 50% of the consumption of the ink. It is designed to wet the pigment, which facilitates the dispersion of its particles. They provide adherence characteristics to the printing ink to bind with the substrates. These substances additionally provide adequate gloss and wear resistance. Usually, various types of resins are used as binders. Nitrocellulose or cellulosic materials, polyurethanes, acrylic, ketonic, polyamide, polyvinyl butyral, PP, cellulose acetate propionate, cellulose acetate butyrate are some examples of polymeric resins or called ink binders used to design different type of printing inks. Varnishes are a blend of resin, drying oil, dryer, volatile solvent, and additives used to formulate printing inks. In its native state, it has little or no color, but may be pigmented or as desired by the end user. Pigments basically imparts or adds color in the printing inks. Inorganic, organic, and metallic pigments and dyes are the kinds of colorants used in the printing inks depends on the end user application. However, organic pigments are majorly used in most of the inks, particularly for food packaging. They constitute from 5 to 30% of the ink. Particularly the inorganic pigments has higher concentration than of the organic pigments. They are usually pigments, dyes, or lakes. There are now coming to the solvents. These are the substances that are designed to dissolve the binder and mix with the other components of the ink. The diluents is typically 15 to 65% of the ink consumption. Ethyl acetate, ethanol, isopropanol, normal propyl acetate. These are some examples of common solvents used in the printing inks. Additives are substances which are used to provide the special characteristics to the inks and sometimes to enhance the existing ones, such as adhesion, gloss, defoaming, tackiness reduction, sleep, wetting, and waxes. As you say, the waxes, chelating agents, plasticizers, defoamer additives, adhesion promoters, anti-slip additives are few examples of additives used in the printing inks. Whenever a layman listens about the types of inks, he gets confused. To simplify the understanding, we'll discuss about different criteria based on which the printing inks are classified. On the basis of drying system, inks are classified as oil-based inks, UV or radiation curing inks, solvent-based inks, and water-based inks. On the basis of printing processes, these are known as offset inks, gravure inks, or flexographic inks. On the application, there are two kinds of inks, surface or reverse. The reverse inks are usually laminated. This slide is all about the coatings. Coatings are the substances used with printing inks to impart special characteristics to the printing articles to provide the aesthetic appearance and protection to the final packaging. Classification of coatings can be done on the basis of different chemistries. Coatings are basically customized on the requirements of the end user application. Solvent-based coatings contain solvents as diluent medium. Water-based inks comprises water as diluent. Again, on the coating side, the UV or LED coating or radiation curing coatings, they don't have any solvents or water as diluent, as they are cured immediately in the presence of radiation. Particularly, it is UV radiation or LED radiation. The coatings can be customized on the basis of their application. Primers are used to enhance the surface property of the inks. These coatings are applied on the substrate prior to the application of the printing inks. Top coats, overcoat varnishes are also used to enhance the surface properties such as gloss, sleep, and so on to protect the printing ink layer. These are applied post the application of inks on the substrate. Heat seal coatings are used to seal two layers of substrate such as polyester and PVC to paper, just for an example. Nowadays, barrier coatings are gaining importance. Barrier coatings are used to provide resistance to various factors such as moisture, oxygen, UV rays, migrants, etc. in order to protect the final product. Functional and specialized coatings are used to create different effects such as matte and gloss coating, embossing coating, soft touch coating, etc. In the lamination adhesives, these are the substances are used to join two layers of the substrates to make a laminate composite such as film to film, film to metallized film, film to paper, and aluminum foil. The lamination adhesives are classified into six categories based on their application. These can be solvent-based adhesive, water-based adhesive, solvent-less adhesive, hot melt adhesive, radiation curing adhesives. Lamination adhesives are further classified as general purpose, medium performance or medium purpose, and high performance adhesives on the basis of their lamination types. The general purpose laminates comprises film to film, film to paper composites, and are typically used to pack dry food, which has to be stored in the room temperature. Medium performance laminates are used in fatty or acid kind of food packaging, where the temperature treatment can be up to the pasteurization temperature, and foil laminates. High performance laminates are used used for the boil-in bag type of application, hot filling applications, sterilization processes, maybe up to 134 or 35, particularly in the pharmaceutical applications. Classification of adhesives based on their lamination processes are of two types in general. One is dry lamination, where a liquid adhesive is coated on a substrate, dried with heat and airflow, and then laminated to a second substrate via a heated compression nip, so that to join two substrates. Then wet lamination it can be described as a liquid adhesive which is coated to a substrate and then immediately laminated to a second substrate via a nip, followed by drying with heat and airflow. Here, the important point is that one substrate must be porous, particularly paper kind of substrates are used to allow evaporation of water or solvent. Packaging films, I think this morning we had listened to Dr. Subra. The packaging films are the materials used to make packets, bags, and pouches to pack any product. Resin chips or virgin chips, or sometimes referred to as master batches, are the major raw material used for making the packaging films. LLDP, that is the polyethylene. There are different kind of polyethylene grades, cast polypropylene grades, polyesters, PVC, EVOH. There are different kind of flexible packaging films are available, and of course, Reliance is one of the major producer of such kind of materials, raw materials for producing flexible packaging films. Packaging films are produced using either blown film or cast film process. Last but not the least, we'll discuss about the printers and converters. They are our customers. Converting companies are the companies that specialize in modifying or combining raw materials such as polyesters, adhesives, adhesive tapes, foams, plastics, rubbers, liners, and metals, etc., to create new products. In these industries, converting process including printing, coating, and lamination are executed. On completion of the converting process, the rolls may be cut into smaller rolls or a slitting machine or a seater. These rolls or sheets are then converted into a convenient size for handling on packaging and other machines. After slitting or sheeting, these sheets are further processed for bags or pouches formation to make the final packaging material. That's all for the packaging, flexible packaging domain. But however, it's all about our next generation or the newcomers to opt for the career in this flexible packaging. And the one message I can give it to the students or the newcomers, that there are three words which are very necessary to grow in these industries. One is network, network, and network. The more you network, the more you find different ways, different means to enter into the industries. So that's all from my side. Once again.
Thank you. Thank you. Thank you, Mr. Mahapatra.
Yeah, I thank OCS organization.
Really, I'm happy that you are dots on time. You have really completed your lecture within this stipulated time, which is given by the organizer. So
Yeah, thank you, because that was the message I have received 2 days back,
yes.
Dr. Samal, that you have to stick to the time, and I am an admirer of that. I'm just taking some time to thank OCS organizers for coining such an appreciable theme, this webinar, and providing me such a wonderful platform to share my views and interact with all of you. If you require any further clarification on my presentation, you can contact me at any point of time, or now it's up to you, Dr. Das.
We can invite at least one question from the audience, those who are listening to your speech. At least one question for 1 minute, like that. Any questions, please?
Sure.
I have a question, Nilkamal.
Yes.
Please.
Thank you for sticking to time and brilliant lecture. Now, the packaging industries, they are needed for us, but as you know, they once the package is opened and we use of the stuff, whether it is medicine or a food, we throw it out. In our country and in many countries, the same story. Will it be the responsibility of the packaging industries to recycle the material, or do something, do some contribution as regards to preventing this used stuff getting into our to our biodiverse system. Plastic pollution is major cause of plastic pollution is packaging industry.
Thank you, sir. I think this topic, I mean, this is the necessity of not the day, this is the necessity for our future, for our environment. And if I start, talking on this subject, I think it will take 2 days of time, but making it short. So there are many people working on the same, and we are also, our company is also working on the same. So there are many solutions. See, by discarding the flexible packaging industry, plastics, etc., is not the solution. The solution is waste management. The solution is circular economy. So these are the two words I think this morning Dr. Subra has pointed out, and I am completely agree with with her. And also there are several innovations are going on that. One innovation is barrier coatings. Okay, that's for, I'm just giving one example. Barrier coating is that converting the multi-layer packaging to mono-layer packaging so that the waste management can be easy. And why it will be easy? Because in the multi-layer packaging, the two films or the two composites or 2 or 3 layers have different TG, glass transition temperature. So it would be very difficult to separate and convert them to other materials. Okay, so that's why the barrier coatings are used to convert these multi-layers to the monolayer. Ultimately what we require a oxygen transmission, oxygen barrier or moisture barrier, a gas barrier. So if you can apply some kind of coating and we get good moisture as well as oxygen barrier, then the packaging will be easy. The final product will be protected. So I think these are the different ways or different means to tackle with the waste management. And I mean, this is what we require for the coming future.
One more law is there actually, that is called a primary producer responsibility. So once the package is supplied by one industry, suppose the Reliance is packaging material is supplying with that package, it is now responsibility of Reliance to dispose of this materials. So maybe by someone or by themselves.
Yes.
This is the law now it is being followed. I think
there is a law.
There is some law.
That primary producer responsibility. That is called a primary producer responsibility. That is the law actually.
Yes, yeah.
Okay. Thank you. So we'll discuss it in the panel of discussion. So it's a nice thing. Once again, I thank Mr. Mahapatra. This is really an excellent message you have given to all listeners that connect and communicate. So I really appreciate.
Thank you. Thanks a lot.
Really appreciate your idea. So people should connect and communicate so that this problem will be solved. Thank you once again. Thank you. Next, please. So next is Professor Sutiranjan Parida. Professor Sutiranjan Parida. So he is, I think, from IIT Mumbai. So his lecture is on materials trends for future applications.
Can you please allow me to share? Hello, sir. Good morning. Good afternoon. And I can share.
Devasis, please go ahead. Make him co-host.
Yes.
Devasis, are you there?
Yes. Now you can share.
Okay.
So material trends for future applications. Bye.
Thank you, sir, for the introduction. And
go ahead. Your time limit is 25 minutes, please.
Okay.
Thank you.
Okay. So I have chosen a very general.
Full screen, sir.
Yeah, is it visible? I've made full screen. Yeah. Okay. So I have chosen a very broad topic, and it's nice to know that there are some students here. So very few basic things I have already given. Since yesterday, you all are listening to the different applications are spoken by different speakers, and they are range from various types of application. And you see the behind that, there is always a type of material or a process always involved, or a process that lead to a particular material or particular properties involved. And I'm just listed that. Tell me, in this society, can you just imagine a beautiful application without a specific and beautiful material? It is not the case. So, therefore, there is always a trial to get the material with a specific property, and then we do have a tool. Humans have discovered the tools to do that. We all know that while designing the material, so that two approaches are used for this. One is the top-down approach and bottom-up approach. So in both the cases, there's few things that we all must note: that design, pattern, and functionality. So even though you have for example, it's a bottom-up, top-down approach, even though you have a building blocks, you just cannot place it in any way you want. So you need to be placed in a certain aspect, in a certain design, a certain pattern, so that a functionality is generated or a functionality is enhanced. And therefore, different length cells come into picture, and this different length cells, how it is designed is really beautiful. For example, you can see a simple tree, and you can see that if you got down into different places and microscopically observe into to the down to the cellulose level, you can see how the hierarchy is arranged. So from the microstructure to the cellulose fiber, to cellulose bundles, nanofibrils, and finally a cellulose chain. And each of this situation, you will see that there is a different length scale, and there is a different design is already ingrained in the material, which gives you the kind of toughness that we all realize in this particular wood or a tree that you see everywhere. So there comes the length scale. Various length scale exist, and as you can see, I've listed few of the length scales: micromillimeter to micrometer to nanometer. And few of the speakers have passed, they have already highlighted the importance of the nanometer. And I've just listed few of the materials which have been the nanometer scale. For example, a airborne virus particle is about 50 nanometer. And the biggest particle that can pass through a surgical mask is about 100 nanometer. Therefore, mask is very essential to what we get from the nanometer scale. That is what is important, and that's what is the part of the thing that I'm going to speak here. So the size effect. So what does the size effect bring to the material property? Other than, so we all know that the specific surface area is one thing that is all increased. What that means is that you get more atom on the surface than inside. If you got more atom on the surface than inside, then the electronic level is property changed. It's not only that, any property that depends upon that kind of arrangement, that kind of electronic structure will change. Be it the physical property, magnetic property, electronics, optical, in strength also if they are arranged in such. And what I'm going to discuss today is taking the example of the energy, that renewable energy. I will give you two examples. One example is the renewable energy. Another example is the protection of material from the environmental degradation. So these two examples I will take and see that how we can designing the material are the very simple steps that we can enhance their property so that make them applicable to the real world situation. So what are the challenges? First is the energy system. So what are the challenges that we get in a low carbon energy system because of the environmental degradation to protect that? So low carbon energy is now the call of the time. So there are three things: reduce the emission and have a reliable supply, and then cost. If we combine all these things, then we'll be successful low carbon energy systems, and uh, therefore, the current research is moving towards the solar, biomass, which is also electrochemical energy, wind, water, and geothermal. So, All these things, new avenues of the energy resources have been tried just for this. But I will be speaking only on the electrochemical energy storage. These are the like supercapacitor, batteries, fuel cell, etc. So storage materials are basically supercapacitor batteries. So where they are applied, this is also one of the design, as you can see here, that you have a wind and you have a solar, and then so you get energy from the wind and solar. But then the question is that intermittency. Always you don't get to wind speed same. Always you don't get the solar amount of solar energy in same amount throughout the day, throughout the season, throughout the month. They are not the same. But then you have to supply a constant load to the users, to the consumers. Therefore, you need a storage device. So whenever there is a high speed of the wind or maybe high radiation of the solar, then it collects large amount of energy. But then we need storage to store it so that from that storage unit to the consumer, so you get the constant supply. So one time you don't get 230 volt, other time you don't get 100 volt. This is not acceptable. So that's how the two storage device comes into picture. They are supercapacitors and batteries. So I'll be speaking on the supercapacitors today. So what are the challenges and how the materials have come and how in our lab we have just what are the simple materials that we bring into the high performance. So these are the different range of the applications in which supercapacitor and batteries are applied. There, from the defense to the from the consumer electronics, healthcare, and various other space application, automobile, various applications. So if you want to know the difference between the two energy system, that actually exposes one energy gap between them. So what is that? Supercapacitor. There are two parameters which are very important in this energy storage systems. One is the power density, other is the energy density. Power density is like tells us that how fast you can go. And energy density, for example, in case of a automobile, means how far you can go. So if you have a higher energy density, that means you can go far. So that is the dilemma. Supercapacitors have large power density but small energy density. But batteries have large energy density but small power density. So this is what is the technology challenge, to get a hybrid device where you can have a higher power as well as the higher energy. So if any of the energy, renewable energy device or electrochemical energy storage device or energy conversion device can solve that, then that will be useful. Other problems also exist in terms of the application, their incorporation into the large design exist, but this is basically a challenge. And therefore, nanomaterials are being used in this case. And why nanomaterials being used? This is the example, because if you use the nanomaterial, then you have a large, because all these electrochemical energy systems operate with a electrochemical interface formation. So what is that? All of you must know that double layer formation. And in this double layer formation, all the energy is storage. For example, in this supercapacitor or capacitor, all the energy is storage electrostatically at this double layer. So this is basically called an interface. So if you have a material with a large amount of smaller structures, a very fine length scales, then you can create such a large amount of the interface, electrochemical interface, and that will give you an enhanced electrochemical response. And that electrochemical response is not only useful for renewable energy, but for sensors, environmental remediation, biological system, corrosion, and non-material coatings. So many applications will open up in this case. So just a little bit about what is supercapacitors. How does supercapacitor basically are? We all know about the capacitor. It's just if you have two conducting material in between a dielectric, and then you just apply a voltage, you just get a charge stored in that, and that's what is called the supercapacitor. capacitor. And exactly a similar type of situation forms in the metal solution interface that we all know. It's a double layer. So what is in the double layer? The charge is aligned here, but the charge that is aligned to metal side and the solution side are all the same. And this is a simple supercapacitor equation, and you can see that there are two conditions you can increase the capacitance. So one is the area, you increase the area, and you decrease the distance. So double layer, thickness of double layer is already small, which is about 1 nanometer or less. It is the area that we can play with. And that's where the different materials, different architecture of materials come into picture. And surface area is therefore considered as one of the things to design a new material so that it can enhance the property. But I will show you that it's not always directly related. You see that there is a direct relationship between, if you take a small cube and just get the capacitance of that cube volume of material, which is solid, not porous. And if you calculate the capacitance versus the specific surface area, you see a one-to-one relationship. We call it specific surface area because of the small size. So I'll just give you one example of that, how the specific area and the capacitance play. Capacitance, why we are stressing the capacitance? Because that energy is related to that. Power is also related to that. So there are three examples I've taken here. One is a ball gold, just 1 centimeter, 1 centimeter ball gold, nanoporous gold, and a single wall carbon nanotube cap. So if you see that, if you compare the surface area of that particular these three materials, a flat gold, which have a small surface area, is about 0.00012 meter square per gram. Specific surface area. Nanoporous gold, which is about 150 meters square per gram. And for single wall carbon nanotube, which is about 1,000 to 2,000 meters square per gram. But look, their capacitance, volumetric capacitance. For example, for flat gold and nanoporous gold, they match. Not one-to-one, but they match. There is an increase. But it is not exactly to the single wall carbon nanotube. It's not exactly enhanced. So here, 10 times the enhancement of surface area here, but not exactly the 10 times enhancement of volumetric capacitance. So what gives? So what basically lies there is not only the length scale of a material, it's not only the size that plays a role, but how these materials are used in real application does play a role there. And in that application, most of the surface area is missed. This is actually a challenge. You can have nanomaterial, no doubt. But two things are very important. One is how you use that so that you can expose the maximum surface area that is to be used. And another thing is that can you prefer it in a bulk amount, economically, cost, and scalability. So, but still, the research has gone beyond to design many ways to enhance the capacitance and capacity function. So electrochemical double layer is one. So where only the double layer charge accumulation at the double layer plays a role. Most of the carbon materials are used by this. So there. So what design? What is the consideration for development of this thing? Is the specific surface area and conductivity. For the pseudocapacitor are a group of materials. Those are metal oxides and conducting polymers. There, such interface do form, but what happen is that it's not the electrostatic charge accumulation that stores the energy here, but there is a very fast surface reaction happening. Remember, it's a surface reaction. In battery, lithium-ion battery, it's a volume reaction. Therefore, it is a rather slow. Diffusion comes into play. But here it's a surface reaction, where therefore it is very fast. Therefore, you can get a capacitive response here. And then hybrid materials come into picture. what gives and why what the pseudo capacitor material give? They give specific surface area definitely, large amount of charge transfer, and also conductivity. These three are advantage as also concern. For example, conductivity of the material is a concern. If you do not manage the conductivity of the oxides or conducting polymers, then they will give a lot of resistance, series resistance. They call series resistance that can lead to heating or loss of power and loss of energy. Therefore, there are other strategy which is now being followed is hybrid system. The hybrid system has many type of forms they are used. The hybrid system can be a combination of these two. Why this hybrid system is used? In one of the cases, there is a voltage range that is applied because you know that in aqueous system for water, you cannot apply more than 1 volt because water will decompose. But then if you cannot apply more than 1 volt, you see energy is half CV square. Voltage is there. So that means if you do not increase energy, voltage, then you cannot increase the total energy. So how to increase the voltage is basically using that. So use one side, imagine an electrochemical cell with the two different electrodes. At one side of electrode, you have a material which is having a water oxidation or a large potential, and then negative side you have a hydrogen evolution which are not large negative. Therefore, you can increase the voltage window and then you can have hybrid capacitance. But nowadays it has gone little further. People are using a double layer capacitance material along with a battery material, super cap battery. That is also coming and research is going on in large amount in that case. So I'll just confine to only forms of carbon materials. These are the different forms of carbon materials. And as we have discussed, orphans always do not match the surface area. For example, graphene, the surface area, specific surface area can be up to 25, more than 2,500 meters square per gram. Activated carbon, which is largely used currently in the commercial supercapacitors, has a surface area can be 1,000 meters square per gram. But then performance always do not match the surface area that we have discussed. The way the material, the way the electrode is designed, that is also important. What I will be discussing about a form of carbon that is called carbon nanoonions. Why it is called nanoonions? Because it has a rings of the pattern that we see here in the microstructure. It's like an onion-like presentation here. What are these? What are these circular things? It's basically single layer graphene layers which are aligned in the circular pattern. Therefore, it is called the carbon nanoonions or onion-like carbons. Okay, so these are the different patterns and things you can see. These are fast, scientifically fast discovered in 1980, but you see that in a traditional traditional way, the way we are making the various forms of process we do. For example, kajal preparation is one of the thing. If you remember, if you all of you must know that. I know that it gives a black powder, and we have found that this black powder is nothing but this, scientifically they might say that it's different, but it's very ancient and traditional technology in Odisha that is being used, and you will see that using this kind of materials, I can, for example, in our lab, we have prepared flexible type of material. So this material have been very nicely coated on a flexible. This is a non-conducting surface, and we have used a cloth type of cloth here, and this is a flexible supercapacitor device that we have made, and we have got a very significant type of capacitance in this case. So the kind of energy you get is basically like 58 watt hour per kg, and the cost of this is less than $1, which is about 78 rupees. So, so basically, this is fine, but then its response is just like any other nano form of carbon. Therefore, so where does it electrochemical capacity comes from? It's not only the, you see, it has a concentric structure there are defects in this graphene structure. Because of these defects in the graphene structure, chemical defects, that's what it contributes to the capacitance behavior. So S area is very moderate here. It's about 100, 120. It's about 100, 200. It's about 200 meters square per gram. But then such a high Faraday per gram capacitance comes because of the defect in this. Therefore, there is a new way of increasing this is by doping those things. So if you can dope those kind of carbon materials with your graphene sheets with a heteroatom systems like a nitrogen and sulfur, you can also increase their electrochemical capacity performance further. So what does happen if you dope with the sulfur? So look, where is the carbon and where is the sulfur? There is definitely not noticeably large electronegative difference doesn't exist here. But what happens that sulfur has a lone pair electron on it. And then this lone pair electron enhances the local reactivity. So it is not only the capacitance function that will enhance. So if such a S doping is done in any form of carbon, it also will increase the electrochemical catalysis. Also, we have done that. I'm not going to discuss that in this presentation. So that is a different line of work. What does nitrogen does is basically it has a slight difference in electronegativity. So basically, wherever it is there, it will induce a residual positive charge on the adjacent carbon atom. And that's what actually is essential for this. So what have we found? This is an N-dope CNO. We have doped nitrogen to that. And what we have found that we have got a very enhanced energy density in this case, which is about more than 40. And power density is coming, look at the kind of power density in kilowatts, more than 15 kilowatts power density. Also, the sulfur doping is also done in a similar way. And this sulfur doping also resulted in a very enhanced capacitance behavior, supercapacitor behavior. So energy density has gone beyond 170, 175, and also power density has also gone into close to the 15 kilowatts. And here, remember, this performance can be enhanced by doing a asymmetric design. This is the hybrid design. That's what we were talking about. So in one side you have a NCNO, sulfur doped CNO, SCNO. And the other side we have an oxide material. So this is a hybrid combination. This hybrid combination has resulted in enhanced capacitance and energy and power density. So if you just compare the three things, so you can see that there clearly there is increase in the capacitance, energy density, and power density in all these materials. So what we did, it is the same carbon material. So what we did, that we changes the functionality to that. So by doping structures, but there are other aspect of work that we have done. We made hybrid with the different oxide materials, and also there are another scope in there by changing the electrolyte. There's a new configuration also we have implemented that in micro supercapacitors. These are the very bulk supercapacitors. As you can see, this is a 3D type of supercapacitor, which is a very large dimension. We have applied also this material in a micro supercapacitor design where it can be applied in the on-the-chip applications. Micro supercapacitor means it's just a two electrode. There's no separator in that, and that's being applied there.
Dr. Purata, please wind up.
Yeah, so this is also, I'm not going to talk about that. Also, we have applied in oxides materials in different forms. There are different oxide materials. And also there is another thing, we also worked on the conducting polymer materials. And we have developed a method in which the conducting polymer can be applied into any non-conducting substance. And this is what we have done. And you can see that the glowing of light, basically we have prepared a series of devices in this case, and that can be used to power a watch or a LED light. There is another last aspect I'm going to talk about, the material degradation. So material degradation is nothing but the corrosion. See, we use the material. This is what the not our advanced application, any structural application. For example, you create any social infrastructure, we always expect it for the 100 to 200 years. And the loss of the material because of the interaction with the environment is basically called the cost of corrosion, which is about 4.2% of GDP in India. Look, how large is that? If you just compare that with the health spending in India, it's about 3.6% of the GDP. So, given there is a huge emphasis on the recycle, reuse, and recycle because of the environmental load, therefore, protection of the structures, and also some of the speakers have spoken about the coatings. That is what is embedded, and it's not just the coatings. It is the functionality, multifunctionality of the coatings that is what is important. So, without spending much time, I'll just go to the. So we have used many nanomaterials in these coating systems and also increase their mechanical behavior as well as their protection efficiency also. We have increased in a large amount. But what I'm going to discuss about the smart coatings. Smart coatings are basically, imagine a thing you have incorporated one type of chemical into that. You have a container, you incorporated a inhibitor, which will protect the structure into that. Suppose if there is a damage, it will come out immediately and protect that structure. These are called the smart coatings or self-healing coatings. So these are the different containers we have prepared, and also along with the small self-healing capability, you can also modify the surface, and you can create the super hydrophobicity of that. So we have demonstrated that making that paper, cotton, steel, so it can be made super hydrophobic any surface. That's what is. So just to wind up the same thing. So if we want to realize any qualitative or quantitative increase in the productivity or for new applications, we must have to go for a new material. But then to get there to a new material, a new technology or process has to be optimized. And then what is the role of that? The role of that is to control the length scale and scale chemistry. So by controlling the length scale and chemistry, then only we will reach out a new material which can help us realize a new beautiful application of that. With that, I acknowledge the contribution from the several postdocs and PhDs and master students in my group. And thank you very much for the organizers of the Odisha Chemical Society and for MSI for giving me the opportunity.
Thank you, Mr. Parida. So it's really an excellent presentation. And this your observation shows that you have done an extensive study on these novel materials. And may I invite one question for 1 minute? Anybody else from audience?
Yeah, this is Dr. Himanshu. Can I ask?
Yes.
Okay. So it's really a nice talk, Dr. Parida. I enjoyed your talk. So I'm not from materials chemistry. One thing I have a question. If you look at the nitrogen doping and the sulfur doping, okay, so in sulfur cases, you have both increase in energy density and power density. Am I correct?
Yes, yes.
Yeah. So is it because of the sulfur? It's not only the sulfur has lone pair of electron, so it has a good polarizability also. So
it has, yeah, good polarizability means what? I mean,
it's polarizable.
See, as compared to carbon, you mean?
Yeah, nitrogen. You are comparing with nitrogen and sulfur doping.
See, just if you compare the electronegativity, I don't see that.
But sulfur is highly polarizable than carbon.
That might also can be referenceable, but
Yes.
for looking at the electronegativity doesn't say that carbon sulfur burnt will be polarized more towards the sulfur. That's what you mean. So that's what I think.
It is not the bond, it's the atom itself. The sulfur atom is more polarizable.
Okay.
Yeah. So that could be one of the reasons. We have one work actually on piezoelectric effect on sulfur, oxygen, all the stuff, computational work. So we observed actually it's both a charge as well. Charge in terms of electronegativity you were talking, and
So it is between the nitrogen and sulfur, right?
yeah. So it's mostly the polarizability has a significant role actually for this energy storages and power storages.
Okay, we'll look at that.
May I interrupt a little bit?
Yes, sir.
Hello, sir. Could there be a good correlation, any correlation between polarizability and the capacity or charge capacity, whatever you are talking of? Could there be a correlation? If you are talking of carbon, nitrogen, oxygen, sulfur, phosphorus, and other things, then they have, you can correlate. What kind of correlation exists between these two parameters? That will certainly tell you about the polarizing property and the other property that you are looking for. Is there a correlation?
I can see that if there is a polarization of a bond, carbon-nitrogen bond, so probability can have lead to a charge accumulation, and also charge transfer is also important. So I see it in that way.
Himanshu, I think Himanshu was asking this question. Probably
So,
he is aiming at, see, if a molecule atom is a polarizable, it will have certain effects. So if I make a correlation between polarizability and the effect that we see in charge capacity, there should be some kind of correlation between these.
Such correlation is not there right now. No, I mean nobody has worked on that. And another thing is that to see the such effect, then you need to have a doping level. You have to increase the doping level, which is has a limitation also.
May I please interrupt for a second?
Okay.
Graphene oxide, when it is converted to sulfonated graphene, there is a comparison. Oxygen reduced with replaced by sulfonic acid groups. The material, its capacitance went up close to 600 Faraday per gram, and number of cycles of operation went up to 2000. So graphene, there probably could be some relationship as this distinguished.
That's what I'm thinking about.
Yes.
Yeah, but sulfonic group attachment is a functionalization. It's not a doping.
No, but when oxygen is removed, only then it is working. When oxygen is there, the capacitance is low. When oxygen, it is graphene sulfonic acid. So
that means it is just an additional functionality. So yes, functionalization definitely has worked, and many studies exist. Increasing.
Okay.
Okay. Thank you.
Thank you.
Thank you, Mr. Parida. So let us wind up this lecture one. And the last lecture is Dr. Pranav Kumar Patra. Dr. Pranav Kumar Patra, who happens to be the head of the process division of Cree Agro Industries of Udaipur, Rajasthan. Dr. Pranav Kumar Patra, and his title of presentation is Crop Protection Chemistry. So I must invite him to be online. So please share your feelings, Dr. Patra. And the time limit is 25 minutes.
Yes, thank you. I think, thank you very much, Dr. Das, for this introduction. I just want to share my screen now. If you can give me
co-host. Make him co-host. Are you there, Devasith?
Already he has.
Okay, now you can try.
Just a minute. You have given me already the thing.
going.
Yeah, it's coming. Can you see my screen now?
It's coming as we.
Yes. Yes.
Very good. Thank you very much, and truly I'm privileged to speak here in front of Orissa Chemical Society, and many of my friends are also have joined. That's a proud moment for me. Yesterday, Ratikanta gave a beautiful presentation on material science purification, and Rama is the convener of this program who invited me. Thank you, Rama, for this. And of course, first of all, I would also be very grateful to Professor Sasodhar Samal, who has taught me chemistry, who actually ignited the passion to think about chemistry, or otherwise I was a vagabond. And I had these talented friends, Rama, Rati, Fakira, and all, Vinod Sahu and Jugal Kishore, so many terrific guys, and late Deepak Behra, of course, I should mention his name here. And they were always protecting me, teaching me chemistry, helping me to come out of issues, problems, and motivation factor for me. So I'm extremely thankful to this group of individuals who have saved my life. They are my, friends for life. And to my teacher, revered teacher, Professor Sasdara Samal, for teaching chemistry to me for 3 years in Bhadrak College, and including Professor Rupasri Ragini Das Samal, who also taught along with other chemistry teachers whom I'm very indebted. Thank you for giving me this opportunity, and I'm sure all of you are hungry except me, because I was always eating. So let me present. Yes, another thing I would like to say that all the presentation and topics that you have chosen in for this OCS is excellent. Different types of science has been discussed, and fortunately there was nobody to speak about crop protection chemistry. So I'm there to give you an overview. I cannot speak anything about my research because this is a private industry, and we are just working for filing patents, protecting our technologies, and the world is like a big monster tiger who is just watching what you are doing, and they will like to take, buy, and consume them. So I will tell you how these crop protection industry is controlled by few companies. Okay, let me start with the history of crop protection. Agriculture started many years ago, 10,000 years ago. And pesticide use started about 4,500 years ago. And as you know, Mesopotamia civilization, the Fertile Crescent, looks like a crescent. The green map on the screen is like a crescent. And there the agriculture started because population was evolving from that area. And then less than 4,500 years ago, pesticide use started. Then from 1000 to 1850, we used all kinds of natural derivatives to grow our yields and to protect our plants. And then from 1850 to 1940, we used inorganic and industrial byproducts like phosphates, zinc, urea kind of stuffs. And then in 1940 to 70, we used synthetic organic compounds. Now, organic compounds started, getting developed. 1848, the first organic compound was discovered, which is urea. And then in 1940 to 70, all kinds of organic molecules started coming. And then 1970 to now, see, what was happening earlier is that we were using indiscriminate use of organic in the fields, but later low risk organic compounds. Now you just cannot spray your stuffs just like that. It has to be extremely regulated. Then only you can use organic compounds as a product on the field. I go to the next slide. As you see here, this graph says that the cultivable land from 1960s to 2016 remained almost the same. Okay, here. However, the crop production increased. Okay, drastically. Our population increased to 7.5 billion now, and our you know. Yield also we increased. So you can see that there is a 60% of yield increase from 1960 to 2016, and this has helped to increase the production to meet demand from a growing population without significant increase in cultivation. I will just go to very quickly into the thing topic without much elaboration of each slide. I'm sorry, and time is less. We will talk about now, about pesticides. What are the different pesticides? We all come from agriculture family, and we know uses of pesticides in our fields. While growing up, we have seen that our fields are full of natural ingredients. We used to drink even water from the field, but now unfortunately we cannot do that. Our fishes have disappeared. Our organisms, microorganisms which you do not know have disappeared, which were dependent on the balance of ecology, they have gone. And I'm working on this field to contribute something which I do not personally like, but at the same time I cannot help, because we need food. But we want to have food. We want to use pesticide without destroying the environment. That is our goal. Okay, so there are how many different types of insecticides here. Pesticides are about insecticides, fungicides, and herbicides. Insecticides are very clear, that we kill insects, okay, with chemicals. And these insecticides are mainly, acaricides and nematicides. Acaricides are arthropods, and sometimes they are visible, but there is a hugely invisible world of these organisms which you can only see by extremely powerful electron microscope. And these kind of, what the photo that I'm showing my pointer here, it's a mite. It's a dust mite kind of things which are in our quilt, which are in our body, which are killing on dead cells, which are also very very good for our environment. And also they are reaching us. They are giving us pain, so we need to do something about them, otherwise they will increase their population. So nematicides, which many of you do not know, that they are very tiny worm-like creatures which are normally infecting the roots of our plants and making the plant very weak. Okay, and then fungicides, the whole thing is all about fungus. Everywhere fungus in our body, in our plants. You can see tomatoes are infected by the fungicides, which are almost destroying the tomato plantation in this picture. And then herbicides, what to say about herbicides? Now, whole India, I see some kind of, weeds invading all our fields. And you know that very well. That is, we call it Congress grass. There is a biological name. I don't know that. But it is called Congress grass in common terms. And it has somehow come from from America with wheat. Okay, I'm not talking, this picture doesn't show that particular weed. Okay, I'm just telling something else about another weed which you all know that it is everywhere. It has come by importing weeds, and these weed seeds have come along with wheat probably, and then everywhere in India, from north to south and east to west, everywhere I see those plants everywhere, and they are quite big, robust. They can only be killed by water, means high concentration of water. If they cannot survive much in aquatic climate. So, and then there are natural origin pesticides, biopesticides, that we have been killing all those using natural oils sometimes. And we go to, now then to develop them. How do we develop these pesticides? Okay. So much research is required. One is, molecule synthesis. First of all, design mode of actions. Biology comes into picture. Structural biology, like Mona Lisa Swai, I think, was presenting about enzymes, how they are, how they are understood by NMR. So this is very important here, how when we design the mode of actions, how these enzymes are there and what enzymes we can inhibit so that we can kill the insect, okay, kill that herbs, kill that fungus. So this is very important area where we work with structural biologists, the entomologist, and then we, as a chemist, I and my other teams who are doing discovery. I'm not into discovery, okay? I'm into process. I'll tell you later what I'm exactly doing. But then the chemistry around exactly similar like pharmaceutical drug discovery. Here it is all about about pesticide discovery. So 16,000 molecules will be synthesized, screened, tested in greenhouses. Then only one molecule will go forward to become a product. 11 to 13 years of research is required to do that. More than 300 millions of dollars is required to make one molecule come to the product as pesticides. It was not before. Before it was much easier because there were no such ecotoxicology studies, efficacy studies, environmental tests were not there. So things are much easier, and we were. I will just give you a few examples of how they have affected our environment. But right now, it is not like before. It is all about safety test, efficacy test, environmental toxicology tests. So these tests require a huge amount of time. Okay, it is not about we get a vaccine and in 1 year the vaccine comes into the picture. It is not about artificial intelligence and you just make the drug discovery faster, much faster than what is happening now, because they can make this faster, but certain tests requires like environmental test requires a lot of time. You can also expedite their time schedule, reduce their time schedule, but to an extent, but not enough. And then our technology guys said there are a lot of artificial intelligence depends on data. Okay, but those data have to be very authentic. Otherwise, garbages in, garbages.
White paper দরকার এই কভারটা বন্ধ দরকার
Yeah. So how it goes now, once a molecule is synthesized, it goes to screening in greenhouses, in vitro labs.
Yaku.
Then it passes to practicality assessment, where we do all these toxicological tests. We do market survey, how this market is, because money is required while developing a molecule. And then we do all kinds of formulation study. Formulation is important because whether you want to spray it or whether you want to spray in water emulsion or in organic emulsion or in as a solid powder. So all kinds of formulation is very important to pass these molecules into the right place to kill the insects or the weeds. And then the most important area is this development. Okay. And here what we do, a large scale studies will be done in different countries. Like Brazil is the biggest producer of agriculture grains. And then India, then all other countries included. So this will be done in Brazil, France, India, in different environment, all kinds of environment. This will be studied because it is all about flora and fauna in different ecosystems. And then it goes to registration, and then approval, regulatory approval is required. Then it goes to product. After it becomes a product, further improvements, further understanding of their activities, adverse activity, and then further repurposing them. So all those things will go on after it is a product. Okay, let's come to what kind of molecules are, unfortunately, fortunately or unfortunately, unlike pharmaceutical, the mode of action here is very narrow. We do not understand much about these insects biology and how they exist, and we know little. Biology is all about heat and trial process. It is not very clear to us, but not like chemistry. So there are few mode of actions I will discuss today, and what kind of molecules have been invented to those mode of actions, and then attack those pests that are actually killing our crops and other plants. So nicotinic insecticides, we call it neonicotinoids or neonics in short terms. So these neonics, this is as you know, the name nicotine, which means it is inhibiting, it is affecting the neural cell. So this is one of the area we know that if we can hit the insect neurons, they will just die. Okay, but then there are positive effects and negative effects. These are very big market of imidacloprid. We know I'm also using imidacloprid in my small plant or in my village plants. I'm using this because this confidor, we all know confidor is imidacloprid, and this is basically to kill a lot of robust flies. However, it is already banned in the world. In India, it is still used because sometimes other country regulations are much more stringent than in India, unfortunately. And we are poor. Our farmers need to use the same thing. We are not so much educated, or our education is not getting very quickly to the farmer. So those kind of problems are always there with us, and we need to do something about it. So these are the three compounds which just a minute, I'll just put this. Okay, which is neuroactives based on the chemical structure of nicotine. It attacks the nervous system and it paralyzes the insects. However, as you have seen in this picture, honeybees, how important honeybees are, and these chemicals can kill the white flies and other flies that it is meant to be, but it is killing the most beautiful species on earth, that is honeybees. Therefore, these chemicals are banned already banned in different places. You do not know probably that how honeybees are important. 1 out of 3 bites of food that we eat are pollinated by honeybees, and 80% of our crops are pollinated by bees. So how important this is, and how important to keep this alive, the honeybees to be alive in our environment, not to be killed by this persistent bad chemicals. This is all about the chemistry, molecules surrounding neonics. You can see that we started from nicotine or anabasine, that is generation 1 insecticides. Then comes generation 2 insecticides, is acetamiprid, clothianidin, dinotefuran. And as you know that science is so powerful. If you see that something is bad for our environment, then scientists keep working on them, derivatize them, change their structures to see that they are good, but they are only killing the targeted species, not the others. So you can see in this box these two compounds, okay, flupyridifuran or dinotefuran it is called, and flupyrimine. These are extremely, honeybee toxicity is very low, and it's already registered in Japan. Can you all hear me? So you can see that these neonics are 23% of our global sale, and it's a big pesticide market, which is based on these neonics, nicotinic insecticides. Yeah, these are the flies that we target, but unfortunately, honeybees are getting killed. Then I will talk to you about another receptor, rhinodine receptor, and this is a intracellular calcium channel receptor, which are expressed in the endoplasmic reticulum, and which open in response to ligand, which is calcium 2 plus binding. So this is one of the molecules. It's called Cyzipyr, and this Cyzipyr or Rynaxypyr, Rynaxypyr probably this is a big billion-dollar sale in insecticide here. So, and adrenaliprole, etraniliprole. So all these chemicals are in the market now and killing the insects which are affecting our crops. So how it does? You see, neonics, they were attacking the nerve cell of the insects, and this is a rhinodine receptor which attacks the muscle fiber. Okay, the muscle, they once this chemical get into the site of ROIR, and it gives the muscle shivering and cramping, and then insect will die. So this is what it does. It so it is the insects become lethargy, they paralyze, and eventually they die. Then there is another mode of action, gamma aminobutyric acid, which is also a chloride ion channel inhibitor. As you see, ryanodine, you see this picture here. The left side is ryanodine receptor, which acts on the muscle, but here again GABA, which acts on the nerve. So these are the chemicals which are actually in the market: fluoxametamide and brofenilide. Brofenilide is a very new chemical which is just in January launched in Australia, and different places it will come. So it affects all those pests that have lepidopteran, like butterfly type of. We don't want to again kill butterfly, but we want to kill the moths, and we want to kill other similar like butterfly. It's a very extensive research. It is not like it is agrochemical research is extremely sensitive research. We have to be very careful, and it's not like before that. We used to spray DDT and all kinds of things which killed all our floras and faunas, and we do not see even fish. Fishes are dependent on daphnia, and daphnias are killed because they are small organisms on which fish is dependent. Fishes are getting killed because they are not having enough food. The daphnias are all killed. So these developments are extremely sensitive, and it takes quite long time, many years of research to develop a new agrochemicals nowadays. Japan, European unions are very particular. Then comes US and other countries. I told you that there are huge world of insects which are invisible, we cannot see, but they have a lot of positive things. That is, they control the arthropods, they decompose, they degrade, dead things, so that bacteria do not overpower our environment. Only the cons is that certain types are damaging our crops, they are allergens, and they are vectors of a host of diseases. What is their mode of action? Mitochondrial electron transport chain complex 2, which I will show you a picture in my next slide. And then acetyl cholinesterase carboxylase inhibitor, acaricides possess also certain novel mode of action or unknown mode of action. Certain acaricides we do not know how they actually act on the insects. So these researches keep continuing, and as you know, permethrin, one kind of chemical which you spray at home and kill all these invisible insects. These are pyrethroid compounds. And in this slide, the mitochondrial. Hello. Yeah. So this is a mitochondrial matrix on the cell membrane, and I'm now talking about fungicides. And fungicide has basically three known site of action. One is SDHI, okay, succinic dihydrogenase inhibitor. Another is QI and QII. QI is quinone outside inhibitor. QII is quinone inside inhibitor. So our biologists, have understood and made assay so that we make molecules and then screen on these assay and then see how these are active against these kind of sites. So, what kind of fungicides normally we are known? Water molds, downy mildews, powdery mildews, leaf spotting, blighting fungi, fruit rotters, rusts. So these are kind of fungus that are actually affecting our crops, affecting our fruits and our grains, and cereals, field crops, tree nuts, vegetables. These are very important for our food product, and they are basically infected by these fungicides. So there has to be research on killing these fungi. This is QI fungi. inhibitor fungi. This is now they have got resistance. Resistance is another big issue here. Like your pharmaceutical medicines also have resistance in our body. The bacteria become resistant. Similarly, the insects also become resistant.
Dr. Patel, please conclude.
Hello.
Please conclude. It's the time.
Okay, sure. I don't know how the time is passing, but there is a lot of chemistry, around these QII fungicides. Basically, I am working on process research, means the molecules which are getting synthesized in the lab and they are giving good biological activity has to be synthesized in kilogram scale and then to ton scale in the plant. So this is my area of work where I take these processes. My team makes novel processes, and then finally, we transfer technology to the plant. So we basically work as a agrochemical manufacturer or agrochemical product developer. We have to be very careful about using chemicals, and as you know, basically, we have to be highly sustainable to develop a process. It is not like just make a molecule and don't care about its after effects. These are some of the insecticides, and as you see, these pyrazole components are very common in SDHI fungicides. And how to make these pyrazole components? These are the best possible. I call agrochemicals are the real chemical synthesis, because you use extremely benign conditions to make these compounds. It is not, you cannot use whatever you want, like in catalyst.
প্রাইভেট লোক সে কি করেছে একটা বিচ এরিয়া ও নিজস্ব দখল করে নিয়েছে, হ্যাঁ, বাস যে গেল খাবার দাবার থেকে শুরু করে তারপর গাড়ি যাবে না ওই জঙ্গলের মধ্যে, তো একটা ফোর্টিংও রেখে দিয়েছে।
So, just to give you a bad effect of monocrotophos. This is, like 2-3 years back in Bihar, in this the meal program that India. Where this chemical was used, although the chemical was not there in the vessel. I'm sorry, there is a lot of noise. Although the chemical was there in the vessel, but this was used for making the mid-meal for children and in a school. And unfortunately, some residue of these chemicals were there in the container, and the children fed with the food used in that container died. 22 children died in Bihar just 2 to 3 years back. You can see that this is already banned in other countries, toxic to birds and humans. But in India, unfortunately, this is still used, and we lost a lot of children because of careless uses. And this is another having $2 billion sale of this glyphosate. It's a herbicide, and it is also a cancer suspect agent. But it is a lot of arguments still going on, because Bayer is making this molecule, and Bayer wants to keep this molecule alive in the market. It's a big billion market herbicide. How to synthesize this? As you see, there are I don't have time already. I got warning. I just want to go straight to the importance of making pure compounds, which I was asking to Dr. Vaidyanath Misra about purity, about those natural products that we are making. How can we make them exactly with the same composition next? My area of research is this. Actually, I want to use extremely pure material, and I want to see the stability of that. I want to see how they can sustain in our environment. I want to see whether I have made any other impurity along with the compound, because we are making in ton scale. So, therefore, these good GLP practices we need to use. We need to see the potency, means total component versus inactive components, how those inactive components are calculated. And then there are regulations. We have to make chemicals which are not affecting to birds, bees, fishes, and then human risk also, whether it is coming to our lungs, coming to our body, killing us through cancer. As you know, lots of pesticides uses are responsible for cancer in the body. So lot of things, and approval process is also very strict now. Money is also quite huge during research and development, so only few companies are working. Monsanto, Syngenta, DuPont, Bayer earlier, but before 2016, and now you see, now only 4 companies are actually controlling the whole world insecticides market. Then comes process research, what we do. What are the environment factors that we have to calculate by sum of mass of waste by mass of isolated product? These are important for as a process chemist, because I'm developing processes for manufacturing scale. Then how much waste we are generating, I am not getting into there much. What kind of chemistry we can use, Claisen rearrangement we have to use, not Wittig reaction, which kicks out triphenyl phosphine oxide. No way we can, as an agrochemist, use Wittig reaction kind of chemistry. We have to use, yes, we cannot use this heavy metal catalyst. We have to use enzymatic catalyst. This is a drug. Okay, I think I am almost done with my presentation.
Thank you, Dr. Parida.
Pranab Patra.
Patra, yes, sorry, Patra. So I think this is an excellent presentation, and that is the real application in our agricultural field. Actually, we are applying, we are investigating, but we don't know what is it there in the background. You have really told us the real truth and the real background of your work. Any more questions from the audience? Because it is already lunch time, so I think we'll conclude. So once again, I thank you on behalf of the organizers and myself. And now I request, this session is over. I must thank all the speakers. And excellently, I find every speaker, starting from Subhas, Dr. Subhas, or Dr. Anil Kamal, Dr. Smriti Ranjan, and last, Dr. Patra. And they have really shown the excellent strength of their work. And this will definitely give a light for the future generation, our future people, that how they can take off their research activities in industry sector. But I must honestly request all these speakers in this session, they have done an excellent work in their field, but they must motivate the people, those who are away from the field, so that the true sense of our chemistry and industry, or academic and industry interface, will be fulfilled. Thank you very much to the organizers, President OCS, who has taken this a novel initiative, and all the organizers of this event. Once again, I thank you. Bye to everybody. Thanks.
Thank you, Professor Das, for your kind gesture in addressing us for a brief moment at the end of this technical session. We also thank all the speakers in the last 2 days. All these, the speakers who have one thing in common. They all are Odia people, or they have some allegiance to Odisha. So we wanted to demonstrate that these guys who came from different background and from different corners of the state are able to excel. So we can do it. We can do it, and it is being done. So we shall discuss what went wrong in between, that in current scenario our students fail to compete nationally, globally, and why they are not employable. So to discuss these questions, we have to move on to the next technical session. That is the panel discussion session, and the session will be chaired by my esteemed friend Dr. Sushant Kumar Biswal. Dr. Biswal, if Dr. Surendra Kumar Biswal, if you are ready, we can start the technical this panel discussion session.
Thank you, Professor Samal. I thank first all 13 speakers has given the valuable addition to the seminars. The objective of the seminar is more clear why the chemistry background students, particularly in Odisha, make their career in industrial research as a career options. We have uh, 14 panelists, 9 people from the academic, 4 people from industry, 1 person, my colleague Dr. Bhatt from the R&D organization. Our main objective is why, which Professor Samal already told, why our students are not getting the employment in different sectors. Before asking to the all panelists, I will just few things I will tell. And during 2009, one task force has been made for higher education in Odisha under the chairmanship of Dr. Trilochan Pradhan. If you look after the task force conclusion, they have given the suggestion for 5 points. First is to improve the infrastructure facility, because if you look after now in Odisha, earlier we are counting 3 universities, Barampur, Sambalpur and Utkal. Now the number of university has been made by the government. Number of autonomous college has been made. In autonomous colleges, everywhere the postgraduate course is there. But if you look after the new university or autonomous college, there is the lack of facilities, the first, and lack of faculties also, because already Professor Samal told that Bhadra College, in place of 20 faculty, only 2 faculties are available, and they have to take the graduate and postgraduate classes. That's why the committee has recommended that immediately fill up the vacant post and need-based revision of curriculum. Maybe we have to look after that our curriculum, whatever we are teaching, either it is a national level competition we can do or not, reforming the examination systems, developing the skill of the faculties. And according to my experience in CSR and different committee in national level, there are 2 ways we can improve, which one is called the top-down approach, another is a bottom-up approach. No doubt those are the central university or central institutes. They are getting good amount of funds and they are able to build up the infrastructure. But still, if I take the example of my institutes, when I joined, that time also a lot of facility was not there. Because in between, government has changed the policy. We have to give the project, bring the money and create the facilities. Through project mode, now we have developed within 15 years, more than 200 crores we spent for the infrastructure facility we have created. Same thing I have observed in IITs, even also NITs. That's why the one is the institute may provide, by government may provide the funds. Another also bottom-up approach that depends upon the person, how he will present his innovative idea to bring the project to create the facilities. And teaching and research, if you don't go together in the particular the university or autonomous college, getting a new knowledge by the student is also difficult. And that is one of the reasons they could not compete in national levels. I will ask our panelists, only because of the time is short, only within the 40 minutes we have to complete. I will ask they can give their views regarding how we will improve the quality of the education in Odisha, and also our students should get the employable in different sectors. First, I will ask the senior person, Professor Ajay Kumar Patnaik. Are you available? Professor Ajay Kumar Patnaik.
Yes, available.
Yeah. Can you give? Yeah.
They may be asked to self-introduce themselves before this. So,
Yeah.
Mr. Patnaik, start with your self-introduction, please.
I am Professor Ajay Kumar Patnaik. I was in various colleges and various universities. I was in Utkal University, I was in Ravenshaw College and Ravenshaw University, and also in different colleges of government, like Holikot College and Bhadra College and Koraput College. And now I am working as a visiting professor in Ravenshaw University. So in the 2 days observation, I first thank the Vishwalu and the Samal sir and the Secretary Priyaranjan for arranging such type of webinar. And this is very essential to our students because they are not aware of about the industry. And the 2-day seminar all are very interesting. I will suggest that what Rasmi as well as the told that the students are not employable. So in order to make them employable, we have to change the curriculum from school stage, college stage, university level, as well as the PhD level. So I have suggest one thing, some few words, you have to, how it will be changed. The government is going to implement the coming year, the new system, NEP rule. Government is implementing any new education policy the coming year, where the school system will be 11 years, college system will be 4 years, and 2 years in MSc level. So in school system, we will request the government and will prepare the curriculum for the school level so that the students in the school level will be motivated towards the industry, how they can be self-employed. So it should be started from the school level in 11 system. In college level, there are 4 years they have to read 4 years. After 2 years, they will get a diploma certificate. In the second year, they will have to choose a vocational course. So we request the government through the OCS to introduce in the vocational subject the industrial chemistry as one of the subjects. So the curriculum will be prepared accordingly, and will request the higher education department to introduce the industrial chemistry as one of the vocational subjects. Second thing, so that we can introduce in the graduation level. In post-graduation level, there are 3 colleges where there are MSc Industrial Chemistry. Already the students are reading MSc in Industrial Chemistry. There are 4 colleges: SKCG College, Paralakhemdi, Videv College, Jaipur, and the Rajendra College, Bolangir. They have MSc in Industrial Chemistry, but they only have theoretical knowledge, no practical knowledge. So it is our duty, the OCS should link the industry with these colleges so that the MSc level will be more activated. MSc level will be more activated. And thirdly, I will tell few lines on the PhD level. In the PhD level, our teachers are guiding the students in different fields. Not that they are doing very good research, and these research can be put into industry in the large scale. For example, one or two example I am giving, that when I was in the university, I did a research work with a student of my PhD student. In collaboration with OUT, we have produced the biodiesel. Biodiesel from a plant, from a seed, which is available in the Rayagada as well as the Koraput, that is known as Simaru or Guamta. So we can test it and compare with the diesel, and found that it is more efficient and pollution-free diesel, but it is more costly because it is not cultivated large amount. If it will be cultivated in a large amount, then that will be more cheaper, and we can use the biodiesel. Similarly, many researchers have producing in Ravenshaw University. Dr. Bharadwaj is producing, preparing the new electrode where they can get the hydrogen as a fuel from water. Similarly, our department, the Sabita and the Sitaram, they are producing, they are synthesizing new drugs which are substitute of quinine. For these drugs are for anti-malaria drugs. They can be used in the industry for large-scale production. So many output of the research in our state may be used for the industrial production. So this is only possible if the industrial people will come forward and will collaborate with the teachers in the university. So to motivate the students towards industry or self-employment, so it should be it should be introduced in the curriculum from the school level, degree level, MSc level, as well as the PhD level, so that the students will be employable for any industry and they can be self-employed also. So this is my suggestion.
Thank you. Thank you, Professor Patnaik, because you have told that the R&D should be industry-oriented program based on the local relevance. Now I will request to Dr. Subram Mahanti. Please give your comments.
Yes, Dr. Vishwal, this was a, I think Dr. Samuel said I need to introduce. So I think in the morning I have.
You have introduced, not necessary.
Yeah.
Okay.
So I would like
some
to
new
say,
people joined, so you need to say a few words.
okay, from my background as such, I am from the Utkal University, and then I joined IIT Kharagpur for my PhD. From last 23 years, I'm working in industry in different capacity, in different industries like all Indian big companies like Reliance and Jubilant Organics. And of course, I worked for, Dow Chemicals, Romandas, and Amnova. There are few known MNCs where I had worked. So my experience is that, like, I also came from that background. Then there are two-way traffic. It is not one way. So my understanding is what I would say how we can develop or take it to the next level is teaching is always a two-way traffic. As much as teacher contributes, student also have to put that much of effort to accept it or take it that positively. Today's students, what they're learning, they are learning from a mobile phone, because I go to a this I think Somaya KG Somaya College as a faculty. So there I see as a visiting faculty, I see people are like they are learning from the mobile phone. They have to go to the books. They have to read. They have to read the literatures. They should know how to read the literatures also. That is also not there. Like currently we have so many scientific literatures. None of our students who are coming from the BSc, MSc, they are even aware about that. So that type of awareness should come. But then, as I think my this professor said earlier, we need to start from the very beginning, from the school level. What I see here in Mumbai schools, because I stay in Mumbai, I see the Mumbai schools, they have a lot of curricular different activities where they develop their personal confidence, they develop their inquisitiveness, they develop the scientific ability to make some small toys or something. So they understand it much better, how a product can be made. So in the school level, that has to be inculcated. That, okay, we need to go, it is not the academics only, we need to make some product which will go to the society. So that is how it is. The functions, that interest should be developed in the school level. Coming back from the school level, then we are coming to the college, where I think so many academicians or industrialists, everybody they are there. So in that stage, I would say there should be a very frequent interaction, either with the industry people, where they should understand, like here in different colleges of Mumbai or Gujarat, they have at least in the final year, at least 6 to 8 months or 1 year interactive course. They have to come to industry, they have to do the project, understand whatever project it is, so that at least they have a flavor of the industry, what industry does, what you need to do. Right now, Odisha students can never get an opportunity. Even when I was there as a student, we never had even idea that is something called CSIR. We have to pass through that or gate or something. We are never aware about that. Okay, we are here maybe because of some of our professors who have supported us, but that has to come from the students also. Like that, outlook. They have to be interacted. Like today, Dr. Samuel and all of you have in this arranged this conference. That should be only with the students, and only few people, the industrial people, should come and answer to their queries, and the opportunities, the plenty of opportunities what is there in different segments. These 2 days, I would thank the different segments you have chosen. So those different segments of chemistry has enough of opportunity, which even our students, they don't know. That has to be taught. And the other thing is that I would say these teachers, they are refresher courses. They also have to study. They have to go to that industrial level, not the theoretical level. Theory, when we come to the industry, we know that whatever we have learned other than the basic chemistry, nothing is useful. If you have a basic understanding of chemistry, all basic things, you will be successful, be it pharmaceutical industry, be it polymer industry, or the pesticide industries. That is what my understanding when I came through that. And these are really few things. And the industry, this intensive collaboration, that has to be again very much like, not like the person goes, somebody asks him to do the literature search. It should be product oriented. That should be a smart goal. It should be specific, it should be measurable, it should be accurate, time bound, all these things, everything should be time bound, and there should be goal to achieve. Students should know today they have to achieve something, and then industry is anyway. Teacher, students also should understand this is a hard work. This is not easy job. We will go, come back. The attitude of the students also need to be changed. That is, attitude is most important in my understanding. Whenever I go and take the interview for the students, I look at the attitude. How much the person is there. Hello.
Yeah.
Okay. Okay. Dr. Subramanian has given the lot of inputs, regarding the students'
Dr. Vishal,
confidence.
Dr. Raut wants to say something.
Okay.
Sir, ମୁଁ ଓଡ଼ିଆରେ କହୁଛି ଯେ ଆମର particularly ଆମର ଗୋଟେ analysis ଦରକାର।
ହଁ ହଁ।
ଆମର analysis ଦରକାର। ଆମର analysis ଦରକାର ଯେ କାହିଁକି ପିଲାମାନେ ଇଣ୍ଟରେଷ୍ଟ ନେଉନାହାନ୍ତି ପଢ଼ିବା ପାଇଁ, ବା ପିଲା ପଢ଼ୁନାହାନ୍ତି। ଆମେ କହୁଛେ ପିଲା ଆସୁନାହାନ୍ତି। ମୁଁ ଆଜି ଦେଖୁଥିଲି 2 ଦିନ ପ୍ରୋଗ୍ରାମରେ ଆମର ପିଲାମାନେ ପାର୍ଟିସିପେଟ ନାହିଁ। ଆମେ ଯେ ଆନାଲିସିସ୍ କରିବା, ମୋର ଯାହା ପର୍ସନାଲ ଫିଲିଂ ଦେଖୁଛି ଯେ ଆମର ଭଲ ପିଲା ଗୁଡ଼ା ସମସ୍ତେ କହୁଛନ୍ତି ଡାକ୍ତର ହେବେ ନହେଲେ ଇଞ୍ଜିନିୟର ହେବେ। ଆଉ କିଏ ଆର୍ଟରେ ଆଉ 2ଟା ସବଜେକ୍ଟରେ ଆଉ କିଛି ନାହିଁ। ତେଣୁ କୋର୍ ସବଜେକ୍ଟରେ ପାଠ ପଢ଼ିବା ପାଇଁ ଆମକୁ ଯେଉଁ ତାଙ୍କ ଭିତରେ ଯେଉଁ ଉତ୍ସାହଟା ଆଣିବା ପାଇଁ, ଏଥିରେ ଆମର ସୋସାଇଟିର ରେସ୍ପନ୍ସିବିଲିଟି ଅଛି। ବାପା ମା କମ୍ବାଇନ୍ଡ ସୋସାଇଟି ଆମକୁ ଗୋଟେ କରିବାର ଅଛି। ଆମେ ମାନେ ବର୍ତ୍ତମାନ କ'ଣ ଡିସ୍କସନ୍ କରୁଛେ, କ'ଣ ଆମେ କନକ୍ଲୁଜନ୍ ଯେଇଟା କଲେ ହେବ, ତାକୁ କ୍ଲାସରେ ପୁରେଇବା କରିକୁଲମ୍। ବର୍ତ୍ତମାନ ସବୁ କରିକୁଲମ୍ ଅଛି। ଏବେ ମୁଁ ଯାଇଥିଲି ରିସେଣ୍ଟଲି, ଆମର ଯେଉଁ ହେଇଥିଲା ଉତ୍କଳ ୟୁନିଭର୍ସିଟି ର କନଭୋକେସନ୍ ହେଇଥିଲା, ସେଥିରେ ମୁଁ ଯାଇଥିଲି ବାଣିବାର ଆମ ୟୁନିଭର୍ସିଟି ଯାଇଥିଲି। ସେଠି ସାର୍ ଆପଣ ଛୋଟ ଛୋଟ ପିଲାମାନେ କରୁନାହାନ୍ତି ବା ସାର୍ ମାନେ କରୁନାହାନ୍ତି। କାହିଁକି କରୁନାହାନ୍ତି, ତା ରିଜନ୍ ହେଉଛି ଯେଉଁ ପିଲା ଗୁଡ଼ା ଆମେ ଆଶା କରୁଛେ ସେ ପିଲା ସେଠି ଏନକରେଜ ହେଉଛି ଯାଉନାହାନ୍ତି। କାହିଁକି ନା ଏମାନେ ଭାବୁଛନ୍ତି ଡାକ୍ତର ପଢ଼ିଲେ ବେଶୀ ପଇସା ମିଳିବ, କି ଇଞ୍ଜିନିୟର ପଢ଼ିଲେ ବେଶୀ ପଇସା ମିଳିବ, ମୁଁ ଆଇଏସ୍ ହେବି, ଆଇପିଏସ୍ ହେବି, କେମିତି ଶୀଘ୍ର ପଇସା 2 ପଇସା ଇନକମ୍ କରିଦେଇକି ପଳେଇବି। କିନ୍ତୁ ଆମ ଏଇ ଯେଉଁ ଏଇଟା ଗୋଟେ କଲଚରାଲ ଇସ୍ୟୁ ଆକ୍ଚୁଆଲି। ଏଟାକୁ ଆମେ କେମିତି ଡେଭଲପ୍ କରିବା, ସେ ପୁରା ଛୋଟ ଛୁଆ ବେଳୁ ତାକୁ ଡେଭଲପ୍ କରିବାର ଅଛି। ଭାଇ ଏଇଟା ତମକୁ କରିବାକୁ ପଡ଼ିବ। ବର୍ତ୍ତମାନ ଏତେ ବଡ଼ ପାଣ୍ଡେମିକ୍ ହେଲା, ଆମେ ଗୋଟେ ଭାଇରସକୁ ଚିହ୍ନି ପାରୁନୁ, ଜାଣିପାରୁନୁ। ଆମର ଏଇ ବିକଜ ଅଫ୍ ଆମେ ମାନେ ଲାଷ୍ଟ ଷ୍ଟେଜରେ ଆକ୍ସନ କରୁଛେ। ବର୍ତ୍ତମାନ starting is why it is happening like this, why ସେ ଆନାଲିସିସଟା ଓଡ଼ିଶାରେ ଗୋଟେ ଦରକାର। ପର୍ଟିକୁଲାର ଓଡ଼ିଶା ଏତେ ଇଣ୍ଡଷ୍ଟ୍ରି, 4000 ଇଣ୍ଡଷ୍ଟ୍ରି, ବିଗ୍ ମେଗା ଇଣ୍ଡଷ୍ଟ୍ରିଜ୍ ଆର୍ ଦେୟାର ଓଡ଼ିଶାରେ। ଷ୍ଟିଲ୍ ଖାଲି ଦେଖିବେ 100+ ଷ୍ଟିଲ୍ ଇଣ୍ଡଷ୍ଟ୍ରିଜ୍ ଆର୍ ଦେୟାର। ଷ୍ଟିଲ୍ ଦେନେ ଆମ ପିଲାମାନେ ପାଉନାହାନ୍ତି। ତେଣୁ ଆମକୁ ଟିକେ ଆମମାନଙ୍କୁ ଏଜୁକେସନ ସିଷ୍ଟମଟାକୁ ଟିକେ କ'ଣ କରିବାକୁ ପଡ଼ିବ ସାର୍।
And I will add it here. If you look after the education in Odisha last 20 years, when the engineering college has opened so many, maximum what you told is correct. The maximum, those are the top grade students that are going for engineering and medicals and MBA. If you look after the 10 years back, 2010, if you Utkal University or any university, we have seen the students quality was not that much, which is earlier when you people are reading. Okay. Now the trend has been reversed. Now the engineering Private colleges almost going to the close. Now many people are now moving towards the basic science. This is the good opportunity time. We have to motivate to them because now the good students are coming to for the MSc, either physics or chemistry, math. This is the good time. We have to motivate them. Our main objective is the motivation to the students, as well as also faculty, because faculty without facility also, how much he will be encouraged? It's very difficult. That's why we have to, the society or public, we have to give a lot of support for the motivation as well as also facility creation. I may ask to Dr. Subra Manti, as Reliance is a big company, no doubt number one in the India. How much, because I know that you people are supporting a lot of ICT, Institute of Chemical Technology, Mumbai, because your R&D is mostly is going in that institutes and Bombay IIT. Is there a possibility to give same, not that amount, some fraction of the support to Utkal University or Evansa or Sambalpur University or Barampur University, if they work your related work, because unless they don't work your objectives, nobody can support. Is it possible?
See, this is possible. We do a lot of collaboration works with a lot of CSIR, lot of university, but then it has to be in that area. We
Yeah.
don't want molecule to develop which is going nowhere. So we want a target, and that too everything is product oriented. If today you see the geo, the way it has developed or anything, it is there is a like appetite for the industries to take it ahead. But then it has to be that oriented. We need a product oriented, that okay, in future just think about something little bit of this COVID era, lot of things came, lot of innovation came, but then that has to come from the faculties or students. And what you said rightly, like I think Dr. Raut said, this is student feel or the family parents feel. In my family also, when I was young, this was the same thought process. Only medical and engineering, these are the only two courses. But then right now we have to tell them, yes, there are opportunities in science, lot of science opportunities as they are. If you see today, all engineers who are sitting in the front line of the bank in the that clerical level, they're all engineers.
Yes.
See, that is the problem. That is a I don't know what I would say. But if these students, they will come to the science, they will do good research, might be we will take it ahead. So they have to give the overview. Okay, if you read science, these are the opportunities available for you, because they are not aware, even the parents are not aware. Okay, these are the opportunity, maybe some cultural revolution, what Dr. Raut said, I agree with him, that cultural revolutions has to come, parents has to change their mindset. Now all
Sir,
CSR institute and other things, that is
sir,
also open.
I would like to few points, sir. Why ICT doing good research and why donors, why the funds are coming to the ICT? Because mostly Reliance, the top man, he's the alumni of ICT. So definitely he will give the project to them. And many top companies, they don't give sir, listen to me for 2 minutes. Many big companies.
I will add it. See, the ICT quality and status is totally different because I have.
Sir, I'm just telling that same thing. Why the big companies are giving the funds to the recognized collaborators? Because they have the facilities, state-of-the-art labs, manpower, etc., and they have the right mixture of the passion towards the innovation. And for the small scale, you see the institutes what we have in our state or in our countries, only the alumni. For example, to give the funds to the Utkal University. Who can give the fund to the Utkal University? If they are taking research, that may help. So here, the important role of those are industry established professionals. They should come forward. Otherwise, it is not good. We will be talking like this. Coming 20, 100 years also, nothing is going to happen.
I will give the good example. The Kenduzar, that is a government engineering college in Kenduzar, because it is established in Kenduzar. Now the government means that is a district fund. That is a mineral fund. They call district mineral fund. we have now sanctioned around 50 crores phase 1 and phase 2. The condition was like that, not only that engineering college will utilize that facility, the Kanjurmarg district, whatever college are there, they will also utilize. Like that, we have to look after the industries, those are IOCL, which is the funding, and also Tata Steel or other mineral-based industry. We have to request to them, to interact with them, to give the funds to different university or autonomous college. Okay.
That is case to case. Okay. What we are talking here that we should
go,
convert the mass flow. All all around the state, the student has to develop and do the industrial research. That is the
you are right. You are right. Okay, we'll take the opinion of others, then we'll comment overall.
Yeah.
Professor Sukullan Das, Professor of Chemistry, BSS UT, Burla, please. 2 minutes. Within 2 minutes, please tell. Professor Das, is there?
Please move to the next speaker.
Okay. Dr. Pramod Kumar Satpathy from that is the North Odisha University. Is available?
Yes, yes.
Okay, please. 2 minutes, please. Give your suggestion because you are the faculty. What is the problem?
Myself, Professor Pramod Kumar Satpathy.
Yeah.
The Professor of Chemistry, North Odisha University.
Yeah.
I did my MSc in 1991. From 2007, I am continuing here as a faculty of chemistry.
Yeah.
So far as my point is concerned, nowadays NAC is visiting to all the big colleges or almost all colleges of the state of Odisha. In NAC, there is a academy industry collaboration. There's some marking what is there, but the concerned college or the university, they lack behind it. So my suggestion is, if in curriculum we should add something towards industry, then the people can be interested. The another thing is, in curriculum aspect of the, so far as NAC is concerned, there is some curriculum enrichment. That is, we should add some value-added courses for the students. In that value-added courses, we can add lot of things for the industry, or for the industry people come forward to teach the students in a 30-hour or to 60-hour duration, some sort of courses, so that the student after MSc can do something in industry. In another thing is, almost all MSc curriculum, there is a dissertation project work. So in that project work, you saw some sort of flexibility should be there, so that a candidate can do his dissertation or project work in the industry, so that he can know aware of the industry. Nowadays, if we compare, almost more than 50% of the students, they're only doing the project or review work. They're not doing any practical or the research work. So my suggestion is, we have to compel the students to do their project in some industry, so that they can be interested and come forward for the job prospects.
Yeah.
And another thing is what Dr. Raut and Dr. Subra told. Now, after the breakdown of the engineering colleges, a lot of good students are also coming to the general line.
Yeah.
But after the general line, what they do? Suppose a assistant professor post was advertised, we are finding that almost more than 100 people are coming with the postdoc, with the industry background, but they're not getting the job. That is the main aspect. So people are not getting suitable position after doing their MSc or after doing the academic line in general feature. Now, after only medical line is only one preferable. After all the students are now coming to the general line, so it should now is the right time to catch students for this research and academic perspective point of view. Thank you.
Thank you. Thank you. Now I will switch up to Dr. Kumar S.K. Bharadwaj, Associate Professor in Ravenshaw University. Is there? Dr. Kumar S.K. Bharadwaj. Okay, we will go to next. Mr. Nilakamal Mahapatra from, yeah, please, 2 minutes.
Yeah, so I'm Nilkamal. I have almost 24 years of R&D experience in different areas. particularly pertaining to printing inks, adhesives, and coatings for the flexible packaging industry. And regarding to this, coming to the point, so the time is now, and we have to act now for this topic. And if we wait, the government of India, government of Odisha, or political people, they will do something for us, forget about that. This is my honest opinion. I'm from my heart. What we can do is a two-way traffic. The students, the teachers, the professors, and the industrialists, they join hand immediately. In our colleges, I'm not sure, like in engineering, sure if any of the colleges that I think the professors are here, do we have a placement cell? I'm not sure. So immediately, if not, then it requires no time. We can immediately jump and create a placement cell. And nowadays, networking, I think I have already told it during my presentation, networking is more important, and it requires no time, no effort. I mean, effort is there, but now everything is connected. So we can do that. The second thing is the students, we have to motivate the student. They should come out of their inertia to not to restrict themselves inside Odisha. They have to come outside Odisha. Okay. So that's the another thing that I think we should, the moment we join our hands, we should do that.
Yeah.
And the third one is that, I mean, globalization era, and we have to give our students, our next generation, some space. So how to create the space, the industrialists or the people that are, who are working in industries like me, they should immediately come forward. So here I am very happy to announce that our company Ansefu will be very happy to get few ODI students so that we can provide them on-job training.
Thank you. Thank you, Dr.
As per our company norms, and I will connect with all of you so that immediately we can arrange maybe a few number, 5 people, 10 people, that doesn't matter, but we should initiate it so that we can do. Regarding supporting the academic, I am not in touch with the academics, but we have lots of analytical equipments in our company. So those who need support for their academic, intend to do their academic research, they can come with us, and I can rather come to them so that we can support them in the analytical research. We can provide them the FTIR, GC, GCMS, many things, OTR, WBTR, GC, Headspace, everything is available in our lab. We can provide them the support. That's one.
Thank you, Dr. Mahapatra. You have given a nice suggestion.
Yeah,
First,
overall, what
is essential in all university.
Yes.
And
overall, I'll just summarize it in only one sentence. If we restrict that, what other can do, what I can do, I can do, so that should be the concept. So instead of waiting for somebody else will do, so what I can do. So that's my suggestion, or request to all of you. Thank you.
Thanks a lot. Now, Dr. Niranjan Panda from NIT Rourkela is there. Dr. Panda? No. Now, Dr. Amresh Mishra is there. Please.
Yeah. Hello.
Yeah, please.
Am I audible?
Yeah, audible.
Yeah. So I'm from Sambalpur University, Dr. Amresh Mishra. I'm working on basically organic synthesis and on photovoltaic. And I joined Sambalpur University in 2016. And before that, I was in Germany for 11 years working on photovoltaic research. And now my group is involved in material development for photovoltaic and basically organic photovoltaic. And I feel this webinar or industry academia workshop is very informative, specifically for us and also for students who are listening to this very informative talk, starting from biomaterials to nanomaterials to organic synthesis, chiral synthesis, medicinal chemistry, material chemistry, graphene, and so on. So, and at the end also packaging industries, pesticides. So these are very informative, and I think this type of collaboration should continue in the future, and we should organize more and more collaborative research and this type of workshop time to time to make our collaboration stronger and for future generation to involve in research and motivate basically the students to involve in the research facility. Thank you.
Thank you for a suggestion. I'm requesting Dr. Himanshu Kumar Biswal. Is there? Please. He's from Naisa, Bhubaneswar. Yeah.
Yes. Before I introduce myself, I thank Sasudhar sir and Devasis sir for inviting me. So for the first time, I'm attending this panel discussion. Okay, so the 2 days talk, yesterday I missed actually. So today I attended many of the talk. They're really nice actually. It is bridging the gap between the academia and industries. So what my suggestion, okay, so my introduction, I'm Himanshu Biswal. Now currently I'm associate professor in National Institute of Science Education and Research. I'm also heading the department for last 1.5 years. For my suggestion would be to encourage the students, you call the alumni who are really established in academia and industries. So at least a few talks in each year, you can call those established alumni and ask to not to give the scientific or technical talk, as to tell them the story about their success. That will be really motivating for the students, and it is the easiest, the first step to start. So you don't need huge amount of money for that. You don't need huge infrastructure for that, but that could be really encouraging. All of us have struggled for that, to get a position, to get a job. So many of us came to science because of financial problem, some problem or other. But their success story will be really motivating than anything else. The second suggestion would be, so in Odisha, we don't have any campus selection for basic science. When I was in university, once or twice the copper sterilite came for the campus. I would request whoever here from the industry, they could suggest the company to come to the campus for the selection. So once, let's say in each year, one or two get from any university or college in industry, and that will be a really news for all others, and that will persuade others also to carry to take the career in basic science. These are the two suggestions I could suggest, which can be quickly adopted and implemented.
Yeah,
thank you. Yes.
Yeah. Next is
and I would suggest those are in academia. So Niger, we have very good facilities.
Yes.
You can bring your students actually. I'll be there. I'll help actually to show our labs. That will be also motivating. So the labs, the equipments, other facilities. That will be really motivating for them also to continue in science.
From Professor Biswal and Dr. Biswal, what I heard is very inspiring, and I would like to just talk to Dr. Biswal, if you permit me.
Yeah, please.
So we know in foreign countries there are lab visits. We do not have that kind of culture, not a part of the curriculum, so it is not there. If it is not in curriculum, if there is no specific instruction from the government, no faculty will take any student and to visit any lab, because he may be or she may be questioned why you did this, who gave you permission to do so. So that is a problem. But there is one way, a solution is that you are now head. Now suppose I want today to be Professor Himanshu Biswal's day. So that day, our camera team will visit your lab, take your interview, and then you can introduce your students, your group, and then demonstrate us some live experiments which we can record. The same thing we can do for other faculty members in IIT. So will it be possible? I mean,
yeah, it's possible. We will be happy actually to do so.
Thank you. Thank
Yeah.
you.
Thank you. The next is I request to Dr. Brajanarayan Patra. Is there? Yeah, please. For 2 minutes, please tell.
Audible?
Yeah, audible.
Sir, am I audible?
Yes, yes. Go ahead.
Hello.
Yes.
Please.
Sir, myself, Dr. Brajanan Patra, Associate Professor of Utkal University. My view in that respect is that we should start from the grassroots level. Grassroots level, I mean the school students. Actually, we are losing most of the students in plus 2 level. They are going for the engineering or medical. So we have to inspire them to read the chemistry. For that, we have to main focus on the school level or how to inspire the student for the chemistry. For that, I think we should do some scientific experiment in school level. I mean, from the 9th onwards, so that they will be motivated towards the science and particularly in chemistry. Then second point is that most of the students after BSc, they are diverting towards banking sector or some civil service like this. So why they are losing interest? Because most of the colleges in our Odisha don't have the facilities to teach as well as to do the practical in chemistry, because labs are not well equipped and we are not giving them all facilities. So at that point, I want to add that if we can make the practical subject, I mean some industrial practical in the curriculum of the chemistry in BSc level, then the students will be, I think, will be more attractive for the chemistry. And third, as Satpathy sir told already, Dr. Satpathy of North Odisha University, we should do the project in MSc. That should be in the industrial collaboration. At the same time, there should be a subject in industrial curriculum. I mean, some industrial experts will come to teach this in our syllabus, academic syllabus, so that they will be also interested for the industrial project. These are 3 suggestions from my side. Thank you.
Thank you. Thank you for your suggestion. Now, next is I request to Dr. Ramakanta Godnaik. Is there? Dr. Godnaik is there?
Yes, sir, I'm present.
Yeah, please.
Yes, sir. So, namaskar to all.
Yeah.
Thank you all. Thank you, OCS. Myself, Ramakant Godnaik, Head of the Department of Chemistry, Barampur University. Actually, I'll tell about this curriculum. Before 2010, the students were very much interested to read medical and engineering, etc. Nowadays, the students have shown their interest to read chemistry. When, from my experience, I will tell that before 2010, a very less number of students were applying to read chemistry in PG level, in a postgraduate level. Nowadays, they have shown their interest. This year, students have shown their high interest. More than 5,000 students have applied to read chemistry in science, in chemistry. So one thing, nowadays the infrastructure supplied by the government to different universities is not sufficient. Also, there are very less number of faculties in different departments. As per Professor Sasmal, he was telling that when he was a lecturer in his time, there are in Bhadra College, there were 12 lecturers. Nowadays, only 2 lecturers per +2, +3, PG level, etc. Similarly, I will tell in all universities, the faculty position is very less. So if government will show interest to increase the infrastructure, modern sophisticated instruments, and recruitment of new good faculties, then I think students will show their interest to read chemistry. Thank you.
Thank you. Thank you. Dr. Bhatt, my colleague. Yeah, please.
Yeah, I'm here. Yeah.
Yeah.
Thank you, Chairman Dr. S.K. Biswal, my senior colleague. And thank you, Sasdar sir, Debasis, for giving me this opportunity to speak here. Let me introduce myself. Myself, Bham Prasad Bag. I studied +2, +3 in Revensa College, and Sasdar sir was my teacher. Then I did my MSc in Central University, University of Hyderabad. Did my PhD at IIT Kanpur, and I had my postdoc with a Nobel laureate, Jean-Marie Len, University of Strasbourg, France, where I worked on adaptive chemistry and self-healing polymers. Here in CSR IMT, I'm working on signaling materials, particularly organic materials in detection technology. Anyway, apart from those introductory part, I mean, it's very easier to speak at the end of a discussion because most of the illustrious panelists, they have already covered up the points. But one thing I would like to highlight here, that the word was, I mean, coming back to the main discussion of this panel, the word itself, that unemployable, is not correct actually. Because see, a ODA student, I mean a student from Orissa, if he is employable in academic sector, that means he is employable in industry also. So that that American lingo, that doesn't come into the picture here. There are so many issues involved, like Dr. Biswal also highlighted those things, like the government policies, the demand supply curve, the opportunities, all those things. See, in Odisha, we don't have pharma industry. So why should the students, they will show their interest for that pharma industry, isn't it? So all those things we need to think of. Yes, we can motivate the students for this industry driven research, that is there. Second thing is, like in what CSR does to India, in Orissa also, the my institute, I mean IMMT, can do the same thing because we are part of CSR anyway. We have a Jigyasa program where we go to the schools and motivate the school children to come to the science-based education, science-based research, and we do that for school children, of course. I mean, with the help of OCS, with the help of CoreMSI, obviously we'd like to extend those kind of programs to colleges and universities of Orissa. But yes, I echo to all the panelists that there must be some government policies, there must be change in government policies, and there must be some change in the mindset of we the parents when we grow our children. Then only we can talk about this industry driven research. Thank you, sir.
Thank you. Thank you. Now I will again to request to Dr. Tapan Raut and also Dr. R.C. Raut, because they may conclude. Yeah. But you have now heard many things.
So there are many things to say and many things to do. Okay. The thing is that because we should work on a common goal, how to bring all the work which is happening in the corner. Maybe people in their individual capacity, they are mentoring the students, they are giving the training to the student, they are going to the college, university, they are doing, and they are also funding. It is happening. It's not that it is not happening. People are also, many professionals are mentoring the students to enter into the industrial sector. But how we should convert that into a flow? That is most important. For that, important thing, that should be a pool. Industrial professionals, those are established, they should pool. And the same time, the push has to come from the professor or universities and institutes. Professors should push the students, because many students, they don't know what is industry, where they will go. That is most important. That gap, that bridge, the gap has to happen. And another important point is that you see, we are talking about the academia and industry collaboration all the time. All the forum we have been talking about industry and academia collaboration. But why a industry will sponsor a project to the institute or university? They should also realize, they should analyze why someone should come to them. Do they have that certain facilities or not? If not, how they are going to make it? They should work in that direction. That is not also happening. We are blaming government has to give everything. Government is not going to give everything. That is sure. Okay, and for this, you should work on a certain model. Model in the sense, the institutes, university, they should identify the key people, and they should interact with the industry. How many times institutes come to the industry? You see, we are giving the example of ICT. I know personally Professor Jadhav, who was the vice chancellor of ICT, because he come to us directly and for the funds for the research. That mindset we should build up. We should avoid the ego. It should be ego less and trustworthy. If that soft issues that can be solved, everything is possible. For example, I can tell you this industry interface meeting we conducted first time. When we discuss myself with Dr. Sasidhar Samal, if we will ego, we will have the ego, we cannot make it success. So it should be egoless and trust. If you have built that trust, definitely everything is possible. All the what we are talking about, the employable issue, this issue, that is, nothing will be there. So my focus will be, first we should work with a theme on we should trust each other, and then we will discuss the roadmap, how we should proceed further. And Rambhai also here, and many professors are here. They can
And
also
what
give their
Tapan is
view.
saying is correct. Only thing my worry till now, why students are not participating. Sardar sir is telling 1 or 2 students if there, then that is also fruitful. That is okay, sir. But why students are not coming? Even in this meeting, we are expecting 500, 400 students.
Yes,
So question, that means there is a gap between our communicator, teacher, and student, and same gap.
Teacher and student.
Similar gap in their career also.
Yes.
That is the reason they are not coming, and we are not getting good students. First of all, I will tell you, we have to encourage the students and parents and students how to read this basic science. Maybe through government, maybe through private, maybe through NGO, maybe the DMMA fund. Funds are many now.
Many now.
Everything now coordinated, and we have made success. Somebody in America, somebody in Europe, and I am very happy. And first of all, we are discussing without physical touch, we cannot make any meeting. Now this, so let us have a positive plan, and I think Dr. Biswal and Dr. Samuel and all teachers, they can guide us, and we will move it. Thank you.
And one thing you see, we are taking the example of Bombay, taking the example of Bangalore. But why we are taking example of Bombay and Bangalore? Because they have the private labs, very big private labs. Do we have any one lab in our state? No private lab in our state. Where students will be motivated? Where students will go and take the training? So we should think with the bigger goal. It is not we should go. Sasidhar sir tell that student will go and take a training in the institute. Sir, that will happen. You sir send me as much a student to our company. We will give all the live training. So that is the that is there, but important, we should have a bigger goal. We should work towards that. Maybe you can discuss offline, sir, with you and with a team, and we should chalk out couple of plans and to work towards that. That is my view, sir. Actually, I'm happy that if anything happen in Odisha, I'll be happy. I am proud of that.
Thank you, Dr. Raut.
Sir, one more,
Samal
sir, I
already has
have one more point, sir.
uh,
Sir, Subhash here, sir.
what? Subhash.
Actually, sir, that universities and academics and even students, if they have any good ideas or any good projects, they can share with us, and we can take to the different level, to a commercial level where wealth can be generated and that can be funded for their growth as well as their institutional growth. And second thing, sir, that a global comment was made during last in 2 days. ODI students are not employable or even not recommended this is a very unfair statement. As I worked in industry for around 20 years. We have taken the student from a BSc, the diploma, and even replaced many IITian and many university students, prominent university students, and they excel. They did very fantastic job. And if you are good leader in industry or in academic or in that or R&D institution, if you are good leader, then you can train them, and they some may be fit to the production, some may be fit to the research, and some may be fit to the management. So that is very unfair. So this kind of attitude we should not develop in this kind of that open forum. So we will encourage our students and other participants so that all these will be motivated and do one course to take uplift our student at least to go to the next level, to go to industry, to universities, to in India and different parts of the country, to the world. So this, I think we should now conclude with this point. That will be a good justice to this of this today's discussion.
Okay. Now already
and the third point, if time is permit, I will third point. And the third, we are taking 7 class, 10 class. We have only one faculty. So I can suggest that there are enormous talent pool in this state, and we can very much happy to go and teach and give presentation of in entire in various organic chemistry, electrochemistry, all applied mineral processing, metal extraction, whatever may be required, or analytical chemistry. Lot of people are talent pools are there. They can add to the value to your education system. So that is how we can, if system is not there, that is how we can build, and instead of crying, we should have light a candle to move forward for the next level. So I think that will be a good idea.
Last point is that last point
discussion.
Noted.
Dr. Vishwal, good afternoon.
Yeah.
Good afternoon. So may I just add one point here? I'm being the principal of Sai International School. So I've been hearing loads of, good points been discussed. I just want to add one point. May I add, sir?
Yes.
Yeah, please.
Yeah, sir. The important, area which need to be developed is a school education. Especially, grade 6 to 10, the way we've been working across Odisha, I think that policies in the school science to be changed. Magic of, let me give an example where in Mumbai, they're doing this. In Odisha, Utkal University is doing the Maths Olympiad. No one heard about example. So how we can change, and somebody, speaking in Niger, best idea, let's take our students, especially in grade 9, 10, 11, 12, and they can go these labs, best labs, and see things over there. And for a video recording, it's a very good idea to record these videos and bring it to the.
Okay. Easy. Anybody from the participants? Anybody can tell? Anybody has any suggestion? Apart from the panelists?
ଭୋକ ଲାଗିଲାଣି।
Yeah. Okay, thank you for your suggestions, and we will again, because the what finally I may suggest one thing. It has to be led by the OCS, through OCS.
Sir, I have one suggestion. Can I get 1 minute or half a minute?
Yeah. Go ahead.
So yeah, so we need to give the opportunity to the students in all the fronts. Like in this panel discussion, if you don't take it, otherwise we should have 2-3 student representatives.
Yeah,
That way they are encouraged also. In all the fronts, the younger should be given the chances also.
Yeah, your suggestion is taken. Any
I will
students?
respond to
Any students?
Sir, I will respond to Imansu. There will be a webinar where only the students will speak.
No, sir, you mix it up. That would be far better. I feel it.
Where the students
That
are
should
only
not
the
be.
speakers.
Yeah. That shouldn't.
We are all in the audience, and there will be also a panel discussion among the students only.
ନାଇଁ ଏଟା ସାର୍ ମିକ୍ସ ହେଇକି ହବା ଦରକାର।
ଆମେ ନିଜେ ଜାଣି ସାରିଲା ପରେ ଆଗେଇବା।
Okay.
Student primarily there will be student speakers. That will be a debate about what are the ways to address this situation at that time. The debate is entirely what are the practical ways to address this situation. ଆମେ ସବୁ ସଜେସନ ଆପଣଙ୍କୁ ନେଲୁ, ତାପରେ ତାକୁ ଇଭାଲୁଏଟ୍ କରିବୁ କୋଉଟା ଡୁଏବୁଲ କୋଉଟା ନୁହେଁ, ତାପରେ ଫର୍ଦର ଡିସ୍କସନ କରିବୁ, ଗଭର୍ଣ୍ଣମେଣ୍ଟକୁ କଣ୍ଟାକ୍ଟ କରିବୁ କୋଉଟା ହବ ନ ହବ, ଆଉ ଥରେ ଗୋଟେ ପ୍ୟାନେଲ ଡିସ୍କସନ କରିବା।
Okay. Thank you all
Yeah,
the
please go ahead,
for giving your valuable suggestions. Now I request to Professor Samal to take over.
Thank you, Dr. Biswal. You have spared so much of your very, very valuable time, and it is nicely coordinated. May I just briefly read out the comments made by different members? It may be necessary just to put it in our mind. Professor Rajay Patnaik said about this new education system 4+4+2. So where at +4 graduation level, there should be vocational industrial chemistry, and at MS level, already we have industrial chemistry, but they do not have real industrial exposure. Then some innovative research work he conducted and being conducted in Ravensa and other places. Industry should show interest. Not that nothing is happening in colleges, things are happening, and industry people should come forward to see if they can commercialize their products. Then Dr. Subramanti, thank you, madam, for being so patient, and the whole day you are here. Students are not reading books. They are not reading literature. They are only interested in the mobile phones. So it is a very important point. Time is changing. With that, all these changes are happening. And this change happened within just 20 years. So school level should be starting point for the existence of literature. At school level, they should know there is something called literature in science, and they should be trained right from that stage. There should be frequent interaction, student visits to industry, and so on. Student seminars where students will speak, refresher courses, and all that. There should be a smart approach. She said very good thing that the approach should be smart. Importance of time and career management. How time is very important, how career management is very important. So some kind of management courses should go into school level and college level. So that she emphasized on attitude. And so then Dr. R.C. Raut said that we need to have a kind of cultural metamorphosis. There should be a kind of motivation for parents to have or to induce interest in their children for science. So this is again going at the same direction. Time is changing, and the kids are. I do not know how much parents do have control over their kids. Um, uh, there is there are many other suggestions, but I'll go to only most important ones. And Dr. Tapan Raut said that ICT gets funding from Reliance because some top brass know each other. So I all the time say the one thing. Suppose you remove IMMT in Niger, IIT, Central Fishery and Aquaculture, and Katak Rice Research Institute from Odisha. Where do you stand? No aims, nothing. So other states have so many institutes. Ours got the institute only very recently. And because this new, again in the new institutes, it is too much, I should not say that, but you know what I mean. When a new institute comes up and when local talents are not available or they are not supported, then this may lead to a kind of equivalent situation. It revolves around at the same point. I do not want to elaborate that point. Dr. Pramod Sarpati from North Odisha University, he said that there should be academia, industry, collaboration as a part of the curriculum. That academy and industry can collaborate, and that should be teaching. That will be curriculum in that on that subject. And very nicely, he said that industry people should be invited to teach. And in response to that, our Dr. Subhas Malik, he also said that they are ready to come forward and teach in industry. To that point, I would like to make a very humble submission. Very recently, we, the retired government college teachers, wanted to teach freely in all colleges. At least I said we shall start from Puri, Jagannath, and we shall teach in all the postgraduate colleges and to start with without any money. We went to the government for that, but the problem was that see, you are a retired guy and you are a talented guy and you go and teach there, then the students will compare your teaching with their teaching and there will be chaos. So this is not allowed. So, but this situation is different. Industrial teaching is something different. So maybe that point can be raised with the government.
Sir, why government you are?
Sorry.
Why, sir, you approach to government for giving the free teaching in the institute? Why government's permission is required?
In the government colleges,
not allowed to enter.
The government colleges, the principal will not allow anyone to enter without government permission. So
then it is this rule is wrong itself.
What to do? That's our government. We have to abide by them. Anyway, so let's move. Don't worry. We shall discuss and try to do something. Dr. Sarpati also said that nowadays good students are coming to basic science, and it is clearly seen that when there is one vacancy, there are too many students coming. So it's not that we do not have smart guys. We have well qualified students, but somehow we do not have much opening. And in this context, I may draw the attention of Dr. Himanshu Vishwal. You are having an institution and you are having your own school. And under you, how many students are employed?
As PhD student?
Yeah, all taken together.
I have currently I have 6 PhD students working under me. One
are you not paying them money? Are you paying
yes,
them?
we are paying. Yes. We are paying.
So
we have MSc students also. They are getting getting fellowships. I have 5 MSc students also.
Thank you.
Doing the thesis work with me.
Thank you. So the idea is that the academic institutes are also very good employers. I stayed for about 20 years in very advanced institutes in South Korea. Our institutes employ thousands of PhDs and postdocs, thousands with their family, and so, fully supported. So the culture there is that you don't have to be in industry or teaching, or you do research and you are paid. That's all. That's all. So that is the
That's
same.
what, sir, I said.
That is the same thing in going on in IMMT.
Alumni is the best motivator. The successful alumni are the best motivator for the
Yes,
students.
yes, 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 yes. So that is another thing you said. Okay, so then other panelists, let me say. Then Neil Kamal said that we have to act now. It's now. Time is now. We cannot delay it. There should be a placement cell. I think there is already a placement cell in colleges. Then networking is very important, and they should come outside Odisha. It's not necessary that you need to be stuck in Odisha. So his company in UNES, what is that? They are ready to give on-job training. Maybe probably after training they can absorb some students. And I had a talk with him, and maybe 10 students per year, or at least say around 5 persons can be employed there. That's a very good gesture to start with. And they have analytical equipment. So anyway, again, whenever I see this analytical equipment, I am prompted to ask that will you allow a video shooting of live demonstration of use of your analytical equipment? If you can do
Yes,
that, that will
yes,
be
Stewart,
great.
any point of time, any point of time.
Thank you, sir. Thank you. Then Dr. Amresh Mishra said there should be many such meets. Absolutely right. This is only a first step, and we do not know what step we are taking. It is a step onto the unknown, and I think from the response, I feel that we have this is a right step in the right direction. And thanks to Tapan, thanks to Rama, and they listened to my request, and they gave this concrete suggestion, and we started. So it is just a beginning, and we shall have a lot more focused, refined, and participation of students and all that. Dr. Prasad also said about, apart from alumni, say the problem again is the same. If you call the alumni to give inspirational lectures, like I have taken training in management. I have a high-level training in management course, and I can teach management at a high level. That is fine. But the question is that many people require a basic support, like a travel allowance or something. Like say, alumni, most of them are in Bhubaneswar, and they go and talk in this and that. Okay, but once they move out, whenever there is a money component, the college cannot support. College has no money. So there will be lab visits, as I told and I requested him that there will be Dr. Pedi Reddy Day, or Dr. Himangsu or Professor Ray's Day. So we shall have such kind of days, and we shall have interviews with you and your students, and then you'll have some theoretical discussion on a particular instrument, and then we shall carry one exact experiment, live recorded. If you agree to that, we shall definitely start writing letters to you. Then
no problem.
Yes, thank you.
We shall be happy. We have many analytical equipments.
Yes.
So starting from NMR to TEM to so we have plenty of equipments.
I know we are very much interested to see your latest photophysical equipments. I know.
Yeah.
So,
Okay.
then my esteemed friend Dr. Rajanaran Patra from Utkal University, he insisted that we should start from the school, catch them young, same thing. So we are losing most of our students at +2 level. It has been going on for quite a while, but there is a changing scenario now. There is a reversal. People are thinking that basic science is more, particularly chemistry. I recently went and gave a lecture in what is that? CET. Yes, CET, and that was a motivational lecture. And some students, they asked, sir, what should be our career, physics or chemistry or biology? I said, go for biology with chemistry knowledge. You will be employed immediately. Go to biology with chemistry background. Then at BSc level, people are switching over to banking. That should be, I mean, we should see how to prevent that. There is a financial compulsions, family financial compulsions mostly. I know 40 years ago, it was the same thing. Today, it remains the same. So we should have industrial practicals in curriculum. Great thing. We should have, definitely have that. And it's already there. This, what is that? Choice-based credit system is targeted at that kind of thing only. So next seminar will be on that, probably that choice-based credit system practical. Our teachers in colleges, they do not know how to handle those practicals. I was asking somebody, like, suppose there is vitamin C extraction from guava. How many of us know how to do exactly the experiment to extract say 10 milligrams of vitamin C from one guava? Himangsu ke janichanti exact procedure. Himangsu sir. So we need to find out the exact procedure. There are 30 experiments and the procedures are not very much defined. Some experiments are being done in Ravenshaw and some are done in Utkal University, and that we shall take up. It's already there in the practical. And project should be industrial project. Again, there is a money component. No parents can send their children to an industry for a 1 month project unless they are supported. And unless it is, if it is a government college, again, there should be permission from the government. Very critical situation, but excellent suggestion. And finally, okay, Dr. Bamakanta Gadnaik, he also said a beautiful thing. Before 2010, it was engineering, medical, but now things are changing. It is more chemistry. The important thing is faculty strength. Recently, there is an ad you might have seen. There are 600 college faculty positions being filled up by the government. And I said yesterday, and again, I'll maybe important to tell you that it is not government is not happy with these vacancies. They're forced to keep these vacancies. The reason is very simple. Suppose in a college 20-30 years ago, there were 10 faculty members. There was no other college in the district. Suddenly now, 9 other colleges sprang up in the same district, and these 9 colleges, they wanted government support. So the money that was allocated for 10 faculty members in one college is now being divided in 10 colleges. So in private colleges, they have one way of keeping some management posts and something, paying 5,000, 10,000. And in the government colleges, they have that, what is that, part-time, there is a name for that. Some the faculty members paid very small amount of money and they are teaching.
Ad position.
ନାଇଁ ନାଇଁ ଆଡକ ନୁହେଁ, ତା ନାଁ ଗୋଟେ କ'ଣ ଅଛି, ସେ ଯୋଉ ଯାଆନ୍ତି 10,000 ଟଙ୍କିଆ contractual. They will teach, they are paid in terms of classes they take. So it is only the money that is important. Higher education budgetary allocation is fixed. No minister, nobody can change it. So only when we increase that allocation, that what I said is GDP going up from 1.5 to 6%, and that is the vision of the present government, maybe things will change. Until then, however hard we may try, the positions will be filled up, but slowly, not at a time. Then Dr. Bama Prasad Bagh, excellent, from IMT, he said he works in organic materials for sensing purpose. So unemployability ward is American, he said. Unemployability ward is American. But we started yesterday from that word, because it's not me who is saying it, and the people who are employing our students, they said that when a student is asked what is the oxidation number of manganese in potassium permanganate, he could not answer. I often go for faculty recruitment, and you'll be sad to know that when I asked a student what is sodium hydroxide, he said sodium NaOH, he wrote, and is it acid and alkali? It's an alkali. So then can you write down methyl alcohol formulas? Methanol, he wrote. This is an acid or alkali, that is OH. He said methanol is an alkali. Got my point? Methanol is an alkali, and he's coming to be a faculty. So that's the problem of employability. And this happened because there are no faculty members in the colleges from where they graduated. They had maybe they have not attended classes because there was nobody to teach them. And they simply, okay, sorry. So sorry to keep you waiting. We have two more. Dr. Tapan Raut again said there should be a common goal. Ego must go. We must trust each other. Dr. Tapan, Dr. Ram, we trust each other like anything, you know. So that is not a question at all. And did I miss anybody? Yeah. So more or less, Dr. Prakash from Sai International, he said that again at school level there should be some training. That is again same thing. And Himanshu again reiterated that there should be student representatives in these webinars. So this is the first meeting, and I always say that no mistakes are committed all the time. You know that particular saying in I.L. Pinar book, a man does nothing until he waits so long that nobody will find fault with him. That thing is speaking my mind. So if we go on waiting, and I'll not commit any. So mistakes are coming. If you commit mistakes, then you are doing a good thing. You are moving. Yes. So with this summary, what I suggest is that to the panel members to write email to OCS and to our chairman, Dr. Biswal, SKB Biswal, your thoughts. Whatever you told, I have written down and I'll summarize and I'll put it up. And then if you have more suggestions to make, please write emails to us. And all the suggestions will be taken care of. I'm assuring you. And we shall try our best to get it implemented at any level. So with these words, I would like to thank this excellent panel discussion. And if you can allow us its time, we shall now wrap it up with a closing function. That was my closing remark about this. And then now request Secretary cum Treasurer, Dr. Priyaranjan Mahapatra, to make an announcement regarding the next OCS event. He has something coming up. Dr. Mahapatra, are you there?
Yes.
Please.
Thank you all for your patience hearing. For our next event regarding OCS Extended Lecture Series 2, along with it will be on recent advancement in material science, and it will be organized by Odisha Chemical Society in collaboration with Department of Chemistry, VSS UT Burla. It will be, I think the date will be announced very soon, but more probably the 23rd and 24th we are going to organize.
January.
And yes, and it will be mostly on different types of materials and its characterization techniques along with the synthesis method. And we are planning to have a panel discussion, but in a different way, that both the 2 days we will have that lecture series, and if possible, the third day we will include some students and more faculty members to have a panel discussion.
Right.
We are planning, but not yet decided, but the date will be announced very soon, and we'll inform with all the brochures and the list of speakers very soon. Thank you all, and we expect a lot of students from your institute along with you also join and guide us. Thank you all.
May I ask convener Dr. Tapan Raut to say a few words, closing remarks, please. Tapan.
Sir, I'm very happy.
He's our
that,
organizing secretary. I'm sorry. Yes.
This,
sorry, go ahead. Organizing secretary.
I'm very thankful to the participants, and many of our participants, they joined from abroad as well. One of my colleagues, Dr. Siba Boom, he joined from Cambridge University, and Dr. Ramanuj Narayan also, he joined from IICT Hyderabad. So I'm thankful that this particular conference with the focused theme has attracted many of them, and hope we will continue this kind of conference in the years to come. And I'm thankful to all my teachers and my seniors, and specifically thank you to Ram Bhai and Professor Samal and Professor Dr. Biswal for their continuous sincere mentorship to us, and with their guidance we are highly motivated to take up lot of challenges, and definitely with the flow we can make it happen and make it fruitful. Thank you very much, sir.
Dr. Ramachandra Raut, convener.
Sir, thank you everybody, but particularly MMSI and OCS leading members and all members. And I only thank all because they have made it success very nicely. And actually, this online present better than the physical training. So you should continue this more and more. Thank you.
Anybody present in this audience to say a few words about this meeting, not a panel discussion?
Paldu.
Yes.
You can say a few words.
Yeah, I was just listening to all these discussions, and this is very nice. I'm so proud of all of you to organize this session, and excellent presentations. I know, I have worked in different places in India and also other countries. And in India especially, I have seen our Odia students are extremely sincere, most sincere than all other state students. And only thing we lack is some kind of system. And I think this is a good starting point. I have always, my professor was having lots of Odia students in my PhD group, and other PhD groups. I have seen that once they work with Odia students, they have been very, very happy about their hard work and sincerity. We have everything. We have a good culture. Only thing that education system, I was just writing to everybody that we need passionate teachers, and it is what the teachers actually shape the system, starting from primary to PhD level. I believe the things will work. It is working already. We have best students actually from Odisha, and I thank you very much. I have nothing more to say.
Thank you.
So, Namaskar, Rasmiranjan here.
Yes, sir. Yes, sir. Please go ahead. Yes, absolutely. Make a closing remark, please.
Yes. Yeah, thank you so much, sir, actually, for really inviting me to participate. Throughout the last 2 days, I have been listening to people. It was really encouraging to see that, the people like me who are, outside Odisha, we have, at least made something for others. I want to really contribute in some way or the other if some initiative is taken at your end. And because I was in academics, certainly I would go and, interact with students. I don't mind doing that. So maybe, in a way that will encourage them, for industrial research. So I look forward to many such, interactions in coming months. So thank you so much, actually, for arranging this. This is wonderful, actually, very wonderful.
Industry
Yeah, actually, I'll be very happy, actually.
Clearance
I'll be very happy. Let us explore. I think
Typical both facility
has already contacted me.
Radio to sir,
I'm radio to Epical both फायदा दूचे अपन पकान Epical through करिया
And I really attest to what Dr. Pranav said. Frankly speaking, whenever, because we Odias, we may not be getting actually the kind of infrastructure or the facilities in Odisha, but once we are out, I know the students who are coming from Odisha, they are doing fantastic. They're so hardworking, actually. They're so hardworking, and because we have a very good culture, so people are docile and they just go on doing their best. So we have to actually, frankly, I feel so passionately about how we can start it from the school level. So maybe actually on a larger, broader, group, we need to discuss, we need to involve the government, how to make it really possible from the state level, from the school level, how we can encourage our, young minds. So maybe actually we need more such interaction and suggestion how to implement at the ground level.
Thank you.
So,
Yes.
ଆପଣ କେତେ ଜାଗା କେତେ ପାଣି କେତେ ଇଲେକ୍ଟ୍ରିସିଟି ଦରକାର କୁହନ୍ତୁ ଦିଆ ହବ immediate free.
That's me. Okay, so the point
Sir,
is,
can I say a few words, sir?
yeah, please go ahead, Dr. Tripathi.
Sir,
Banki
Bankim,
bhai
Bankim.
बहुत short रे कुहंतु
Ma'am, short coach already this international seminar was is a very successful one with able leaders like Professor Samal and Dr. S.K. Biswal. So I can see that in future we are going to have many more occasions to share our thoughts and experiences to young scholars. And I think these young dynamic teams, they can contribute a lot to take way forward. That's what I wanted.
Subhas, you want to say something? Subhas?
No, that there are plenty of but trained manpower will be available in Odisha. So any industry or any industrial support, we are there to support to build a very good infrastructure.
Very good.
So
Just do it, sir.
people are now in the ground. Very motivated team are there.
I have always a feeling that Indians are very smart because of two reasons. Our medium of communication is English, and we are very good at math. Our math is very good. So among the Indians, I also feel that Odia people are also very smart. I mean, maybe they are kind of very, very smart people. And that is amply demonstrated when Orias they go out of Orissa. Out of India, they do excellent job.
Sir,
That
India
means
ta ba egna Odia loko
there is nothing lagging in the intellect of Odia people. What is lagging is that we are not able to provide them the scope to grow. That is the only problem, I think. So yes, Ram, go ahead.
Sir, I will add one line. India is run by nine Odia people. You talk about anybody, RBI governor, Prime Minister advisor, everybody is Odia.
You can trust them. Very simple, docile people, trustworthy, and
yes, kamo karanti.
They are the qualities of
Purusartha.
So we shall be closing this session, and
Yes.
I now invite Dr. Debasis Mahan, the co-convener, to kindly propose a vote of thanks.
Thank you, Professor Samal. I am very much thankful to OCS and for MSI for arranging such a nice seminar. It's a first step, and we looking forward to many more. I am very much thankful to all the speakers, all the panelists, and all the participants from across the country and abroad. Thank you all. I think it's a very successful seminar, and it's more successful than any physical meeting. Thank you all.
Thank you.
Thank
See
you.
you.
Bye-bye.
Happy New Year.
Happy New Year.
Thank
Happy New Year
you.
to all.
Happy New Year to all. Thank you very much.